So first of all, I'm delighted to welcome Professor Ibrahim Abubakar, who's the UCL Vice Provost for Health, to do an opening welcome. Thank you.
Good morning. Thank you, Mike. So welcome to this important event. It's my real pleasure to formally open the 75th anniversary celebration for the Queen Square Institute of Neurology. It is right that we are here convened on the square to celebrate this important and landmark occasion, and I'm looking forward to an amazing day, the lineup of incredible presentations that illustrate the sort of science that happens today. It is also great that we happen to have this 75th anniversary, nearly a quarter of a century of celebration that coincides with 200 years of our university, UCL. Many of you would know that 200 years ago when UCL was founded, it was founded on the idea that not only would we create useful knowledge, but would expand the cycle of who benefits from this useful knowledge. I can't think of a better example in health of doing that than the amazing record that this institute has achieved, translating important discoveries into real impact for everybody in society, in this country, as well as globally. It is also right that we are celebrating this anniversary in the square, here in this room, but also later in the day, out actually in the square, because for me, the magic of this place, the genius of the people who created it, is actually co-locating the science in the same place as where the patients are seen. The fact that the Queen Square Institute of Neurology is co-located with the National Hospital for Neurology and Neurosurgery is part of the reason why the amazing success that we'll hear about through the day is possible. We truly are able to deliver complex clinical care for the most difficult brain conditions in this location, while also ensuring that the amazing bench work that happens next door and can be translated. So people talk about bench to bedside, right? Here it actually happens. Ideas in clinic get translated into amazing new bench work, and that bench work gets translated. So there are numerous examples of discoveries that originate in both directions, where first-in-man trials happen, where the actual evaluation of the treatments happen, pharma companies rushing in to do the evaluations of their treatments. I've seen several examples when I visit laboratories here. That therefore means national policy and global policy changes. It is that makes this place special. I'm clear when I look at, and I've been to several lectures, right, where Mike and colleagues kind of reel out the range of discoveries, whether it's our increasing understanding of the penumbra and that therefore the ischemia that happens in stroke is not irreversible and you could do something about it with thrombolysis, whether it is use of surgery to intervene in advanced epilepsy, whether it is deep brain stimulation for Parkinson's, whether it is the advances in neuroimaging that means we can more precisely understand and diagnose dementias, multiple sclerosis, all of those amazing transformation in science. For me, it's what makes this place special. It's not simply that there are lots of citations that you publish all the time in Nature, in Science, in New England Journal, etc. It is that you do that as well as transform the lives of people. It's the fact that the grandfather who has had a stroke can speak again. It is the fact that a dad can pick up their baby. It is the fact that a young person can have a better life and more longevity because you've discovered amazing new treatments that were not possible. And I'm super proud to be part of this amazing story in my leadership role and to be part of the translation of that into impact. We look back at all of those achievements, but it continues to happen. Today, we know that in the last few months, the incredible work of colleagues at Sarah Tabrizi and Ed Bowles in Huntington's, ensuring that we get lots of national headlines, but real translation to patients, right, that would now potentially benefit from first of its kind in the world reduction in the risk and impact of Huntington's. All of this is possible, not by accident, but because the individuals in this room at all levels provide incredible leadership, whether it's at the level of managing the clinical service that means that patient can be enrolled into treatment, or running laboratories and being the lab managers that enables the scientists to do these discoveries. All of us that are part of this story have a major part in the impact that has been achieved. But it also requires leadership. It requires vision. It requires that we invest in these areas and that we do better for the future. It is why I am pleased and proud to thank Mike for his incredible leadership over the last 15 years, setting that vision, but also to appreciate the contributions of our former leaders. I can see we have two of our previous provosts in the room, and I think it's important to acknowledge that, because without their foresight and realization that this is an area that UCL has incredible strength, and investing proactively in it, none of this would have been possible. So that leadership at all levels of the university, from people running services all the way to the leadership of the institution, is why we are where we are. And we continue with that story. So looking into the future, you'd be aware that we have invested into the hundreds of millions in the Queen Square DRI initiative, working together with the hospital and with national infrastructure in the form of DRI to deliver the right facilities that would mean that we remain at the cutting edge of this space, that we remain number one in the world in the neurosciences, that we leverage the fact that we win large grants, right, into the hundreds of millions in this area, that we continue to produce the right companies, the right businesses and innovation that would transform and make sure that the impact of our discoveries reach patients. It is that I'm proud of. And that is the reason when I set out at the beginning of last year to develop a strategy and a vision for health in UCL, I put neurosciences and neurology right at the heart of it. It is the reason why we are proposing to invest more to discover transformative changes in brain cancer, to ensure that when we advance in computational healthcare, in disease interception, in advanced genomic therapies, that we precisely make sure we do it in this space. I am looking forward to another 75 years of amazing discoveries, and I think the conversation today is not just a celebration of your amazing impact, but a look to the future to make sure we continue to achieve this transformative change. Thank you very much, and I'm looking forward to an amazing day. Thank you.
Thank you very much, Ibrahim, for that really nice welcome. So I'd also like to add my welcome to all of the colleagues in the room. It's so nice to see so many old and new friends. All of you in this room have contributed to the success of the Institute. I'm also really pleased to welcome two previous directors, I think Richard Frakowiak and also Roger Lemon, and also to welcome the director-elect, Erin Schumann, who will start in January. So it's great to see you all. I'm especially pleased and honoured, as Ibrahim has said, to welcome two previous provosts and presidents of UCL, Sir Malcolm Grant and Sir Michael Arthur, who have both actually contributed significantly to the story of the Institute, as they well know, including through some challenging but exciting times. For those of you who don't know, I am Mike Hanna. I'm the current director of the Institute, and I'm really pleased to be doing this introduction to give you a flavour of what's to come during the course of today's exciting plan. So we're here to celebrate an amazing institution that's a privilege to be part of. And it's really 75 years of translation of really great science into impact for patients. We're going to tell you some of the stories of how science has really affected the lives of patients globally. We're going to try and consider why has that happened here, what's special about this place, this small square in Bloomsbury. And we're also going to consider the future, particularly the next generation, and perhaps some of the challenges that society faces. I'm really delighted at the end of the day, Lord Vallance, the UK Minister of State for Science, Research and Innovation, will come and give a guest lecture. And I'm also really pleased that Professor Sir Michael Arthur has agreed to say a few words at the reception in the marquee, which we hope is being erected, and we'll be there at 6 p.m. If it isn't, we'll just have drinks in the square. So that's the plan. So it is quite an interesting story that began with two very foresighted, visionary women, Joanna and Louisa Chandler. Joanna looked after a relative who'd had a stroke or paralysis, and sort of realised there was no proper way to help people who had suffered such an event. And they managed to persuade David Wyer, who was the Lord Mayor of London at the time, to do a fundraiser. As it turned out, if you read the history of David Wyer, he probably also had a TIA, and so it had a neurological event. You don't hear much about Edward, but he was the brother who also was fairly central to this vision. They did a fundraiser, they raised £800, and they managed to purchase 24 Queen Square. So we're located in 33, they purchased 24. And they managed to open a hospital which had 10 beds, which initially was for women only, and they managed to see around 800 outpatients per year. So that was the beginning of the story. They apparently chose Queen Square because it was a beautiful, relaxing garden square, which is still the case, although there's more cars now. And they were obviously highly effective because within 40 years, a whole new hospital had opened. And by 1900, this was absolutely the major, in fact, the first neurology hospital anywhere in the world. It became really famous for the precise clinical assessment of patients, precision in diagnostics, which was really mainly clinical at that time, and still is a real feature of Queen Square today. The first neurosurgical unit opened, and it had a huge teaching program attracting doctors from all over the world. And actually, there was a bit of a disagreement with UCL at that time because the hospital wanted to open its own medical school, which UCL did not agree with. And so they went to the Royal College of Physicians, who apparently had the ability to give them a charter. So a medical school only for neurology was opened at Queen Square. And had a global reputation in teaching, which was really unsurpassed. And I think it's still the case today that this is the premier place to learn the clinical skills to be a neurologist or a neurosurgeon. In 1938, another wing was added to the hospital, and just to illustrate the central importance of research, this is before the institute, this plaque was put up that you'll all have seen where it says research, every addition to true knowledge is an addition to human power. So there was a real ethos that research was the way to develop treatments for patients right from the very beginning of the history. And actually Simon Chauven has just completed a book submitted to the publisher to recognise the 75 years celebrating the story of the Institute. And if you're interested and you email me, you can get a discount price on the book before it's published. And I think I've read it, it's a really interesting read. So the most important thing I want to say today is thank you to all the people, past and present, who have made this such an amazing place for science, but also for patients, which is really important. So all the current staff and students, all the colleagues who have served Queen Square and the Institute in the past, so we are really standing on their shoulders. And really importantly, all our professional services, current and past, without them, they underpin everything all our teachers do. All right, apparently I'm not appearing on the video. Okay. So the other thing I'd like to say is that I think Ellen and Richard are here from Lancet Neurology. They've agreed to publish a piece today on the 75 years of the Institute. So that's in the Lancet Neurology, I think, today. And I've mentioned Simon's book, which is a really interesting read. So we have great staff, and just to thank them all. And I have a great team supporting the leadership of the Institute. So Helen Crutzen, the manager, a great team of deputies illustrated here, and all the professional service staff, and particularly thank Navida and Sandra for massive work to make today actually happen. Here's the full professional services team, which are really crucial for the success of the Institute. And we're part of a faculty. We get great support from Alan Thompson and the faculty team and the leadership across the faculty. So we're part of a ecosystem of science. So this is what we're about. We really want to use the best science to deeply understand mechanisms of brain health and disease at multiple scales. And as you've heard, we really want to, where it's possible, translate that into benefit for patients, either prevention, diagnostics, or therapies. Educate the next generation and have impact. And also, importantly, develop a culture that's supportive, diverse, respectful, and where we drive partnerships. And of course, we need to be financially and carbon sustainable. So this is partly about advertising Queen Square. So if you scan the QR code, you'll see our annual review. This is produced every year by Selina, which describes some of the incredible things that happen every year at Queen Square. There's lots of different ways to measure what we do. Publications is one way to measure it. I'm always amazed how active our teams are in massive amounts of publication, including in very high, what might be considered top-tier journals. I'm pleased to say that on my watch, the money's still going up, so that's good. And it's just incredible. All these great people have transformed our ability to win major grants. So in 2015, we were at 40 million in active research grants. We're now at 440 million, which is a big chunk of all of UCL neuroscience. Of course, the wider neuroscience UCL is a powerhouse globally. We have 8 departments, which are the powerhouses of our research, led by some really inspirational men and women leaders. And we have some divisions which are important links between the hospital and the institute, led by these colleagues illustrated here. Broadly speaking, all the departments span basic science through to clinical work. And as was mentioned, we talk about bidirectional translational research. This is a very important part of the Queen Square Institute of Neurology's story, where we go from patient back to the lab, back to the patient. Our questions might be informed by observing the patient, going back to the lab. And we've been increasingly trying to think of ways to support this type of activity and develop the appropriate platforms. And 2 of them are in the next slide. So the Leonard Wolfson Experimental Medicine Trial Centre was set up with a 20 million award from the Wolfson Foundation. This has been amazing, actually, led by Vincenzo Libri, as a place where we can take early experimental drugs into patients safely. And many of the breakthrough gene therapy studies, for example, and trials have happened in this unit. The other thing we've worked hard to do is develop local in-house expertise about how do you translate take things forward towards the patient and commercialization, working closely with key support from areas of UCL, such as UCL TRO, UCL Business, and Dimitri and Susie lead on this, and we've been successful in developing spin-outs and more contacts with industry to advance our science, and you'll hear about that later. So you can't do translation without partnerships. There's some really key partnerships illustrated here with the university, with the Biomedical Research Centre, with the CRIC, crucially with the hospital, and it's great to see hospital colleagues here, and with UCL partners. So translation is totally dependent on partnership working. You cannot do it on your own in a lab. And of course, we have great charity and philanthropy partners. So just to thank OVPA, the UCL fundraising office, different charities, Epilepsy Society, National Brain Appeal, BRUK, and many other charity partners that are genuinely partners in our endeavour to use science to find treatments. I'm also really proud, actually, over the last 10, 15 years, how we've really advanced our work around EDI. This is really thanks to Helene Plun-Favreau and Dimitrios, illustrated here. We now have 12% of our entire staff volunteering to be involved in different aspects of EDI. And we've been on a journey, and we're now, I'm really proud to say, awarded a gold Athena Swan charter, which is a big, big achievement, only one of 6 in the entire UCL, which is a big place. So what about the future? So there's clearly lots of challenges. I mean, brain diseases is a very big unsolved challenge. Neurodegeneration, Alzheimer's, the biggest cause of death today, increasing in frequency. Of course, global climate change is another critical issue, human-derived CO2 warming of the climate. This map on the right, all the red dots, is just the other week, the cities that achieve the highest temperature ever. So all of these cities, particularly in Central Europe. And 5 years ago, led by Sanjay Sisodia, we decided to try and develop a strategy about what are we going to do about climate change, in particular in relation to the brain and brain health and brain disease. So with Lancet Neurology, Sanjay spearheaded the Hot Brain Conference, an annual conference looking at all aspects of climate change in the brain and disease and health. Published in the Lancet Neurology some work just showing how little we actually know. If you survey all the literature, there's not much research in this area. Sanjay's leading research, along with others at the Institute, looking at what the mechanisms of vulnerability might be. And we're working with Wellcome to develop a strategy to address this in a more concerted way. So that's one really important area for the future. Educating the next generation, as you heard, it's been a key feature of Queen Square since its inception. We have significantly expanded our PhD and BSc, MSc programs over recent years, as illustrated here, led by colleagues indicated here, and Alex is the Deputy Director for the Institute for Education. A recent development in the last couple of years is undergraduate teaching at the Institute. It was always postgraduate historically, but we've had great success with a new Human Neuroscience BSc and intercalated BScs for medical students from UCL, but medical students from other places can come and do BScs at Queen Square. So just to thank the huge number of colleagues who commit to teaching, leading in teaching. So a really key thing for the next generation. I also just wanted to make a point about neurosurgery. So there's a fantastic tradition of neurosurgery breakthroughs at Queen Square with the colleagues indicated here. But I particularly want to highlight Rob Brownstone, head of the Division of Neurosurgery's work in the last 10 years, to really build a new generation of neurosurgical scientists, how neurosurgeons work, how they work with neurologists, how they help with delivering advanced therapies, such as gene therapies, is really going to be important. And Rob's developed a cadre of fantastic trainees who are coming through now to consultant jobs, and that's really important for the future. So I can't do a talk without saying something about the building. So this was an idea in 2011. This was the first diagram we did. We thought we'd just rebuild Queen Square, straightforward, take a couple of years. Queen Square House didn't quite work out like that. It's been a long story. Many people have been involved. But we are now, as Ibrahim says, on the verge of making an unbelievably fantastic new neuroscience environment, just 7 or 8 minute walks away on Gray's Inn Road. Many people have been involved in making this happen. Ibrahim chairs the executive board. There's so many people, too many to mention, but I'll just mention 2. Alan Thompson is the sponsor of the programme, and David Howard, who's in the centre of the bottom, is the manager for the programme. And just endless amounts of work have gone on to make this happen. So on the right is the computer-generated image. And just to try and convince everybody, the one on the left, this is the real actual image. And so it is approximating to what we planned. And we're all pretty optimistic now it will be finished by the end of next year. It's going to be just an amazing environment for all of our science. And importantly, it will include clinical service as well, in that great tradition, as Ibrahim said, of the clinical work happening side by side with the science. That's really, really important. So I think it's going to be one of the world's leading ecosystems for translational neuroscience anywhere. So at the Gray's Inn Road end of the equation, it's mainly laboratory-based science with great technology, great facilities, but there will be clinical services, outpatients and MRI scan facilities. And then at this end of what we're calling the dual hub for translational neuroscience is mainly the clinical research linked to the hospital. So I think this is a fantastic new opportunity, a great and inspiring place for everybody to work. It will have core platforms at Gray's Inn Road with new technologies that are relevant that will be shared for breakthrough science. And then coming towards the end, there have been many pioneering women in neuroscience across the history of the Institute in Queen Square. The Chandler sisters obviously were the original pioneers to think up this whole thing. But Anita Harding was another example of a fantastic pioneer. She really set up the whole field of neurogenetics, as well as making several key discoveries in her own work, discovery of mitochondrial DNA linked to disease, for example. So I'm really pleased that Valda Jackson is creating a statue, a bronze sculpture of Anita, and she'll be sat just outside the main entrance to the new building in a garden area that will be there. So that's a really, I think, a nice tribute to Anita. So finally, looking back to look forward, we've heard that UCL was founded in 1826. Thanks to the Chandler sisters and the brother, that they did manage to do this amazing thing. And by 1860, they'd built this incredible hospital. It was 1950-51 that the Institute of Neurology was formed. And this building was built in 1978, which at the time was thought to be the most beautiful state-of-the-art building in Bloomsbury. I don't know how they got planning permission. I was sort of reassured slightly that from planning to finishing was from 1964 to 1978. So that's longer than our building. And it was supposed to cost 1 million, but it cost 2 million. So the overrun is even more percentage-wise. But it's just incredible, all the different things that have happened in and around this building, in this small space around Queen Square, the breakthroughs that have really impacted patients globally. And you're going to hear about these amazing stories of science during the course of today. So it leads us to reflect a little bit, and we'll talk about this later, on what are the success factors for an ecosystem of innovation that has real impact. And these are some of the things that I think are important. So, the best environment and the best technology, attracting the best people, interdisciplinary, diverse teams of people, co-locating patients, basic science, clinical researchers, clinicians, scientists, asking the right questions. The questions might be informed by science or might be informed by observations in the patient. And of course, all of this creates an inspiring environment for the next generation. And I do think that is what we're creating, I hope, in this dual hub ecosystem for the next years. Because there are major challenges. Some of the diseases you'll hear about today remain big challenges for society that we haven't solved. Major morbidity for patients and families, major economic challenge for society. So this is really, really important. And finally, this courtesy of G.P. Schiavo found this, that this is a paper in Nature that basically says you can do perfectly good collaborations at a distance, but if you really want to have transformational impact, you need to be physically in the same place as other people who have different backgrounds and you have the same goal. And I think that's a key message. And so I think 7 minutes is okay. There was lots of debate about should it all be here, but I think 7 minutes is okay, and we're going to create something amazing. So thank you for your attention. I'm now going to ask Derek to show a film which gives you a flavour of what we think is special about Queen Square. So thanks for your attention.
Everybody who works here has the same mission: to reduce the burden of neurologic disease. We're all part of one team.
75 years ago, the National Hospital for Neurology decided that they should set up a research institute. So the Institute of Neurology was actually born of the hospital. Queen Square, which is the institute and the hospital together, are absolutely now seen as a world-leading centre for innovation and development of therapies. It's bringing together the best students, the best clinicians, the best scientists to cure brain diseases.
Compared to other places, which they are doing very good preclinical science or very good clinical science, but they don't talk to each other, here is a unique environment.
We have a constant dialogue with our clinical collaborators to make sure what we do in the lab is really connected to what the patient community needs. That really helps to accelerate the research that we do.
We really do research and treat all common and rare brain diseases. So for example, in Huntington's disease, dementias, Parkinson's, Alzheimer's, motor neuron diseases, multiple sclerosis, epilepsy, and of course stroke. There are many examples of breakthroughs at the Institute over these 75 years. Queen Square has been so good at taking science to benefit patients.
One of the most important moments in dementia research was the identification of the first Alzheimer gene. That focused research to the therapies we've now got.
The major Queen Square breakthrough for stroke is the discovery of the ischemic penumbra. We've gone from a disease where there was no treatment to be offered to delivering treatments to thousands of patients every year.
I work on Huntington's disease, and we've just had a very exciting gene therapy result. For the first time, we showed we could slow the disease by 75%, which is huge.
There is such enthusiasm for innovation, and that's led to things like the emergency stroke unit, pre-hospital care, and really just shows that research and clinical work go hand in hand.
Queen Square is a really exciting place to work. We have the infrastructure, labs, the imaging centres, we have the clinical trial centre, and we have the patients who come from all over the UK to see us.
Research at Queen Square is so important. I can tell that from when I started taking part to now, huge advances have been made, like real progress.
The generosity of patients who really selflessly contribute to research is totally amazing and actually quite humbling.
So the next chapter is really exciting. The new Gray's Inn Road building, which is a new centre for translational neurosciences, will bring world-leading technological infrastructure.
That will just drive innovation, drive collaboration, and really help to accelerate treatments, which is the ultimate goal here at Queen Square.
Great. Good morning, everyone. For those of you I haven't met before, I'm Selina Wray, and together with Helene Plun-Favreau, it's my pleasure to co-chair the next session. So as we were putting together the programme for today, we really wanted to put together a series of talks that would show how much progress has been made in different disease areas, but also really highlight the Institute's contribution. And over the next session, we will hear about that in 4 different disease areas. Following the talks, we will finish with a panel discussion which will explore how innovation and enterprise in neurological disease research are really helping us translate fundamental discoveries through into patient impact. We're really excited to have such an excellent programme, and so without further delay, I would like to introduce our first speaker, who will be talking about Parkinson's disease. Professor Tony Schapira is head of Department of Clinical and Movement Neurosciences, and his pioneering work on mitochondrial dysfunction and neurodegeneration has really transformed our understanding of Parkinson's disease and helped to drive the development of new therapeutic approaches. Please join me in welcoming Professor Tony Schapira.
So thank you. Thank you very much, Selina. And thank you very much to the director and the team for the invitation to participate in this celebratory anniversary. 75 years of Parkinson's disease research in 30 minutes. So where do you go? Where do you go to get an unbiased, dispassionate, data-driven answer to the question, what has been Queen Square's contribution to Parkinson's disease research over the last 75 years? Well, where else but ChatGPT? So, and I read verbatim, if you were to nominate a single institution with the greatest overall historical influence on Parkinson's disease research, many neurologists would argue for UCL Queen Square because of its sustained contribution to. Well, I hope that over the next few minutes, I'll be able to convince you that on this occasion, ChatGPT is correct. First of all, a few disclaimers and explanations. So this is how I've put this together. I've had to focus, because of time, on a few consensus highlights and themes. I've focused on Parkinson's disease, not on Parkinsonisms. And I focused on publications and progress rather than people. And importantly, as we've already heard, there's an important focus on translation. And this little orange icon you will see on slides when there is a particular and direct translation to patient benefit. It's also important to emphasize that much of the work that I'm going to talk about has also involved collaborations across institutions and across countries. But the examples that I've selected have been driven by Queen Square, but yet incorporated all of those individuals elsewhere. So the title is fairly ambitious: Translating Discoveries to Treatment and Cure. And I thought that in the journey, we need a roadmap. And the roadmap is the basis for mapping the contribution of Queen Square to Parkinson's disease research. You need to understand the physiology of movement to understand how it goes wrong in disease. You need to understand the pathology and the clinico-pathological correlations to learn what's happening during life and disease to death. You need to understand the etiopathogenesis of the disease, involving genetic cell biology and the pathogenetic pathways. Clinical presentation, prodrome, and progression are crucial to the understanding of the development for the management of Parkinson's disease. And one of the most important aspects of that, and the tools that we use, are the patients, the clinical cohorts, and the evolution of the way in which we use clinical cohorts for genetics, biomarkers, and understanding progression. Ultimately, though, it's about clinical trials and identifying drugs and compounds that will allow us to achieve that ambition of a cure. So this is the roadmap that I'm going to take you through, and I'm going to start with the physiology. An understanding of the normal function of the brain in movement. I'm going to go back about 40 years to these gentlemen here who were from Sheffield. They were not from the Institute of Neurology, they were from Sheffield, but they had the concept of transcranial magnetic stimulation. And there you see them with their original machine, and here you see them down at the bottom. It's 40 years later. This is not a consequence of the TMS that they... 40 years later, at an international TMS meeting that Martin Kolzenberg arranged to celebrate the 40 years of TMS. But back in 1985, here was the beginning of transcranial magnetic stimulation. And here you'll see some of the individuals labeled. The person in the middle, Dr. subsequently Professor Kerry Mills, not because he responded to the TMS, but here you see the initial experiments on normal controls to understand the physiology of movement. And because it worked and there were no significant side effects on Dr. Mills, well, it was tried on an eminent neurosurgeon, Professor Lindsay Simon, with similar benefit and effects. Although it is right to say that Sheffield conceived the idea of TMS. It was Queen Square and the Institute of Neurology that incubated the benefits of TMS to the application of the understanding of physiology, and subsequently translated to the treatment of some neurological and psychiatric disorders. So a great history over a significant period of time. But during this period in the early 1980s and 90s, the physiology of Parkinson's disease began to develop exponentially, and its application to Parkinson's disease treatment also began. So the concept of deep brain stimulation was initiated by Alan Benabid and co-workers in Grenoble. But again, the incubation of the application of DBS, deep brain stimulation, to understanding normal motor movement and also to treating Parkinson's disease developed here at Queen Square. So much so, the legacy of all of that is that here you see in this corner that Queen Square Hospital dominates deep brain stimulation in the United Kingdom, doing over 40% of all procedures. So direct translation to patient benefit. But the physiology of movement also incorporates a number of different things. For instance, the illusion of the stopped clock phenomenon was explained, and also an understanding of the abnormalities and the physiology that underlay some of the symptoms of Parkinson's disease were elucidated here and have provided the basic infrastructure to developing new forms of treatment. Now, I'm going to move on to the pathology, because here is, I've explained, an important evolution of the understanding of what is normal and what is abnormal in neurophysiological terms. But what about the neuropathology? And how do you correlate the pathology with the clinical features that you see? A landmark paper was this paper which mapped the neurodegeneration of Parkinson's disease in the substantia nigra. It's difficult to underestimate the importance of this observation, which was able to map in anatomical terms the specific areas of the substantia nigra which degenerated specifically in Parkinson's disease, in contrast to other disorders and normal aging. So this set the scene for the development of the Queen Square Brain Bank criteria for Parkinson's disease. This too was a landmark development. This was the first time that Parkinson's disease was defined, clinically and pathologically. It became the gold standard for not only the clinical diagnosis of Parkinson's disease, but the recruitment of patients to research and clinical trial. The importance of this was that the patients that were recruited were much more of a homogeneous population, and therefore you could understand the results that you were getting more easily. So this dominated the Parkinson field for 30 years. And it was only subsequently with the Movement Disorders Society diagnostic criteria, not totally dissimilar from these, that we have moved on from this, which was first published in the early 90s. So a hugely important paper. The next really fascinating step change in our understanding of Parkinson's disease came almost with chance observations. Fetal grafts had been used in the 1990s to treat Parkinson's disease with variable results. And the team here at the Institute of Neurology collaborated with the team in Lund in Sweden to do this. There was in parallel a program in the United States led by Warren Alano and Jeff Cordova that were doing the same thing. 10 years later, after grafting, when some of these patients came to autopsy, there was a dramatic finding that the fetal cells that had been implanted into these patients had developed the same pathology of Parkinson's disease, of Lewy bodies. We'd all thought that Lewy bodies were simply a product of aging and time, but these cells were only 10 years old. And so from these observations, back-to-back papers from the London Swedish group and from the North American group, showing exactly the same thing, led to the idea of prion-like spread of alpha-synuclein. And this is hugely important, not only in our understanding of the pathophysiology of Parkinson's disease, but also in developing new treatments. Because if you can reduce or stop the spread, you will also reduce the symptoms and the progression of the disease. So a very important observation. Clinico-pathological correlations have been critical in our understanding of the patient during life before they come to autopsy, to be able to understand how their disease will develop, the rate of progression of the disease. And so in this study here, we've started to see that regardless of the age of onset of Parkinson's disease, the onset of complications like hallucinations and cognitive dysfunction, etc., seem to occur at the same point in time of age, indicating how age is such an important and critical factor and the most important risk factor of Parkinson's disease. This study here was able to highlight that there are different rates of progression amongst those with Parkinson's disease, and you can identify that at a relatively early stage. And this very recent paper that just came out a couple of weeks ago correlates the pathological changes with the genetics of Parkinson's disease. So putting all of this together, it helps us to build up this amazingly complex understanding of what is going on with the disease. So the pathology and the Queen Square Brain Bank have made numerous hugely important contributions to our understanding of Parkinson's disease and how it evolves. And I'm pleased to say that under its current stewardship, that continues. And the evolution of digitalization and the application of artificial intelligence to the understanding of pathology and neuropathology is now well underway. So I'm confident that the Brain Bank will continue to be at the forefront of Parkinson's disease research. Etiopathogenesis. This is really understanding what's going wrong with a cell. So I'm going to talk a bit about organelles and genes and pathways. Let me take you back a little to the late 1980s and early 1990s when we talked about the neurophysiology, the neuropathology. These things were all happening at the same time. And in the late 1980s and early 1990s, we began to develop an understanding of the biochemistry of Parkinson's disease. What was going wrong with the actual nerve cells? What were the abnormal pathways? Mitochondrial abnormalities were identified, and also increased iron, free radical damage, and oxidative stress. So these were the two planks which were evolving in the late 1980s, early 1990s. And it's interesting to note that these biochemical abnormalities preceded what was the next major step in Parkinson's disease research, which is genetics. Now, the first two genes for Parkinson's disease were identified in the United States at the NIH and in Japan, the synuclein and the Parkin genes. But soon followed two extraordinary observations and discoveries of the PIC1 gene as a cause of familial Parkinson's disease, and of the gene that was to be subsequently called the LRRK2 gene, both in 2004. So these accelerated our understanding of the genetics of Parkinson's disease, but importantly, also started to fill in a lot of the gaps in our understanding of the pathogenesis of Parkinson's disease, the biochemical cell biology of Parkinson's disease. And here's a cartoon from 2005 showing the mitochondria and the protein degradation system, and starting to plot some of the genes that were associated with this pathway. Subsequent work here at the Institute has confirmed that mitochondrial abnormalities are important. They occur early in the sequence of events in Parkinson's disease. As we moved on through the genetics, of course, the application of genome-wide association studies led to an avalanche of new genes being identified and associated with Parkinson's disease. One in particular, which is here, which is put a little earlier when the first clinical association was identified, but it wasn't until later in 2009 that the association of glucocerebrosidase, or the GBA1 gene with Parkinson's disease, was identified by Ellen Sidransky at the NIH. But this seminal discovery led to an acceleration in our understanding of Parkinson's disease. So the pathology of GBA1 positive Parkinson's disease, and by the way, GBA1 mutations are seen in 12% of UK Parkinson's disease, by far the commonest genetic association. And this is a worldwide phenomenon. They are seen more frequently in some other populations, but 12% in the UK. So the observation that the pathology of GBA1 is the same as idiopathic Parkinson's disease is really important. And perhaps they also pointed out it's a little bit worse, the Lewy bodies, the alpha-synuclein. An observation that actually in GBA positive, as well as GBA negative Parkinson's disease, you see a reduction in the enzyme activity. So there's a link now between the idiopathic and the genetic forms. Some familial things coming back, mitochondrial dysfunction, and an understanding of the observation that glucocerebrosidase, or the GBA1 product, actually is important in mitochondrial function itself. So some of the gaps are now starting to be filled in. And this lovely review, led by one of our colleagues here, has put together this map of the different cellular changes and the progression of Parkinson's disease. The mitochondrial abnormalities, the lysosomal problems due to GBA, the aggregation of alpha-synuclein, the spreading of alpha-synuclein, the pruning of synapses and the death of neurons, the accumulation of Lewy bodies, and the development of Parkinson's disease. But over the past few years, another frontier has opened up, so to speak, in Parkinson's disease. The gut, and the gut-brain axis. In particular, the content of the gut in the form of the microbiome. Billions and billions of bacteria. Evidence accumulating over the past decade or so indicates that the microbiome can have a significant impact on the development and evolution of Parkinson's disease. 2 papers in the last few months have emphasized this link. This very nice paper using a mouse model demonstrates that immunocompetent cells in the form of intestinal macrophages accumulate alpha-synuclein and can lead to the spread of the alpha-synuclein through the periphery to the mouse brain. Depletion of these macrophages in the mouse model retards and reduces the effect of the pathology. This paper here identifies a specific microbiome signature in Parkinson's disease. And importantly, shows that it is present in genetically susceptible individuals who have not yet developed the disease. And that there's a continuum of the change of this microbiome from controls to genetically susceptible to the disease, which opens up opportunities for biomarker development and, importantly, for treatment to intervene in that pathway. Cohorts and clinical trials. Well, cohorts are hugely important to understand the disease, and here I've listed just some of them: the PREDICT PD, PROBAND, and the Parkinson Families Project, which are hugely important in not only understanding the disease, but collecting patients for clinical trials. And this paper here in particular demonstrates that if you go back through the general practitioner record database, you will see the evolution of Parkinson's symptoms beginning years before the diagnosis is ever made. Really important in understanding the prodrome of the disease. Genetics, of course, are now leading into the development of cohorts, and this is a so-called PD front line, which is over 8,000 Parkinson patients, all genetically analyzed for GBA free of charge to the NHS. And the opportunity to see and follow the evolution of the disease amongst these people, now over 8 years, and to develop clinical and genetic counseling for individuals who are positive. So what I've been taking you through is a roadmap towards the ambition of a cure. And we now get to the final stage of the evolution to clinical trials. The next slide is going to show you what all of the work that I've been describing can lead to. This is a review from a couple of years ago, just showing the Parkinson's disease drugs currently in trial for symptoms and for disease modification. So these have all come from an understanding of the biochemistry, the genetics, the physiology that I've summarized in the past few minutes. And they comprise targets for the pathways I've talked about: mitochondria, lysosomes, etc. I'm going to pick up on 2 here in phase 3, the most advanced of the clinical trial catalogue. So this is the exenatide story using GPL1 agonists, based on an observation that Parkinson's disease is more common amongst people with diabetes. And this is the exenatide study, which has led from Queen Square over several years through different stages from phase 2 to phase 3. And although the most recent study published just last year was negative in not reaching its primary endpoint, subsequent post hoc analyses are now underway to identify those individuals amongst the trial population that did respond and to understand why. The next is a drug called Ambroxol, a repurposed drug which is found to enhance glucose-3 bisidase activity in fibroblasts, in mice, in cortical neurons, in non-human primates, and then in humans in a proof of principle study, leading to a phase 3 study which is already halfway through its full recruitment, and results should be out in the next couple of years. So this is an example of the evolution of basic science to clinical trial. Now the problem is that we need to do our clinical trials better, because if you remember that diagram I showed you, there's lots and lots of drugs yet to be tested. So we need a much better way to test those. And this has led to the evolution based from here in terms of its application to Parkinson's disease, but also involving Newcastle, of the multi-arm, multi-stage platform trial. Choosing drugs, choosing endpoints, and involving the public. And basically, what has been designed is a parallel study where multiple drugs are being looked at the same time in different segments of the cohort. And there are interim analyses that will be able to tell you whether any particular drug is actually bound to fail and the patients can be changed to something else. So this is a way to accelerate clinical trials. Now, I did say right at the beginning I was going to concentrate on publications and progress and not people. But it would be very remiss of me not to mention just a few names in terms of the legacy of Parkinson's disease research at Queen Square. And these are the three people that really stand out. David Marsden, Andrew Lees, and John Rothwell, who, in terms of their contribution to Parkinson's disease across decades, has been absolutely enormous. Certainly world-leading and world-beating. So those three people, I think, have had a huge impact on the different aspects of Parkinson's disease. And I'd like to thank you, not you, but these people. Because these people are taking up the banner and leading the way forward. And I'm confident that these people will find the cure for Parkinson's disease. Thank you all very much.
Thank you, Tony. For those who don't know me, I am Helene Plun-Favreau, another Deputy Director, and the next session is on dementia. But before I introduce the speaker, we are going to play a short video.
My father, Roger Pratt, was diagnosed with young onset familial Alzheimer's disease, and he died on New Year's Eve 2024. I definitely remember getting the first diagnosis of Alzheimer's and that being like, what? Alzheimer's? Like, never. And then the next thing of, oh, actually, it's this really rare type of Alzheimer's and it's genetic. I remember the feeling afterwards because I couldn't stop crying.
There are 1 million people in the United Kingdom now with dementia, and that number is set to grow as the population ages. Queen Square is integral to the story of advances in the fundamental research into dementia. Our research is built on long-term partnership with patients and families. Perhaps particularly in familial Alzheimer's disease, We're the National Centre for Families. We set up the very first familial support groups. And those families have allowed us to track the changes that are the very earliest onset.
He loved life and he loved people, and he was so interested in taking part in research from the point of diagnosis.
Yeah.
Taking part in research means a lot to me. When your family receives the diagnosis of a rare disease, to find out that it's genetic, and to watch your loved one deteriorate from the disease, I think those are loads of really good reasons to want to do something. When you come together and take part in research, straight away you feel less alone.
Most of Alzheimer's disease is not inherited and passed down through generations, but rarely, perhaps less than 1%, is. And those families have led so much in terms of what we know now. Following individuals from pre-symptomatic disease all the way through is the only way we can understand what influences progression in diseases like Alzheimer's disease, frontotemporal dementia, Huntington's, and that arc of trajectory that is so important. Brain scans, for example, have given us a window into seeing what's happening in some of these diseases, even before there's any damage to the brain. The ultimate goal, the ultimate prize, is to intervene pre-symptomatically.
The earlier we can intervene, the more effective those treatments are likely to be. In my lab, the uniqueness of our approach is because we can't easily take brain biopsies for our research. So what we do instead is we grow stem cells in the lab, which are a blank slate. We can take those stem cells and turn them into brain cells that we sometimes call mini brains. And by doing that, we can compare those cells to understand the very first things that are going wrong in the brain. The importance of that is we can then think about developing treatments that intervene at each of those stages.
Everything that we're seeing now, for example, the new therapies for Alzheimer's disease, first treatments ever to have shown the ability to slow disease, they're built on 40 years of research.
One of the most important moments in dementia research was the identification of the first Alzheimer gene. With my colleague, Dr. Martin Rosser, in the late 1980s, he and I started to work with families with Alzheimer's disease, and the largest family, the Jennings family, had mutations which we found in the amyloid protein. Finding that definitive mutation focused research, and it also guided drug company research to the therapies we've now got. When you look back on a breakthrough like that, what's been really important from that history onwards has been the building here at Queen Square of a clinical trials unit which puts these new therapies to use. So we've now got the first treatments.
I'm really optimistic about where we are in dementia research at the moment. And my hope is over the next 5 to 10 years, we will really see an acceleration in terms of our understanding of the disease and the development of new treatments for patient benefit.
I think we're in the most hopeful time since I started taking part. It feels like there's some light in the darkness.
So it's my pleasure to introduce the next two speakers. First, Nick Fox, who you've seen in the video, and Nick is the Joint Head of Research Department of Neurodegenerative Disease, and Cath Mummery, who is a Professor of Neurology in the Dementia Research Center. So both of them are leaders in dementia research and in clinical care. And as you've seen in the video, it's a really exciting time for the field with recent breakthrough in disease-modifying therapies for Alzheimer's disease, obviously offering new hope for the patients. So please join me in welcoming Nick and Cath, who are going to play a double act, I understand, in the next 25 minutes. Thank you both.
It's a real honor and a pleasure to be celebrating the Institute's 75 years of contributions and to be able to celebrate the contributions to dementia research that the Institute has made. But I have to say I feel rather daunted. As Tony said, it's trying to compress a lot into a short time. There are over 200 disorders that cause dementia. I'll be selective. I'll pick a few things, but I'll pick things that I think illustrate some of the changes in our field and some of the changes that have happened at the Institute, how we approach dementia, how those advances have contributed to the changes. And I've entitled it Ending Nihilism, because I think that is the starting point, a very nihilistic place that dementia was in. So with that, please forgive me, I'm missing things out, but I'll try and give a bit of a story of that. I will give some examples of the progress and advances of the last 75 years, the past, if you like, and then Cath will describe some of the really exciting developments happening now, particularly in the field of trials and therapies that really are helping to end that nihilism. And then I'll briefly conclude with a bit of a look to the future. So the IoN, as you'll hear today, has contributed phenomenally to a broad sweep of neurological disorders. But I think it's fair to say that dementia has been a sort of late arrival at that party. And that is more of a comment on dementia than it is on the Institute. It reflects a pervasive nihilism and stigma that has really surrounded dementia and is really only now starting to be shifted. There was a view, which I'm sorry to admit, was included neurologists, that really was nothing much you could do about dementia. What was the point of making a diagnosis? It was distressing to patients. There were no treatments anyway. There was no urgency. It was a sense that really people just had to get on with things themselves, and there wasn't really much interest either in research or in an urgency to diagnosis. Treatment and care had been woefully lacking, and there is still much to be done. But key to Queen Square has been this close collaboration between hospital and university, between clinical service and research. But it's worth noting that the very first specialist dementia clinic at Queen Square was only set up in 1988. The Institute of Neurology was approaching middle age by then. And that was monthly, one clinic. Now there are a dozen clinics across the week. There are really specialist multidisciplinary services in Huntington's, in prion, in frontotemporal dementia, dementia with Lewy bodies. A real partnership between the hospital and the Institute, between clinicians, scientists, and patients. And all of the clinics that run across the week are linked with world-leading research groups. Name just a few here: the MRC Prion Unit, Dementia Research Centre, the Huntington Centre. But the progress that has been made clearly depends upon fantastic people and ideas. But it's also fair to say it also depends upon these advances just would not have been possible without investments and funding and infrastructure. But these things are interlinked. It's a two-way street. Only with the advances is there a sense of confidence that this is a place in which it is worth awarding a grant or making an investment. But those investments then lead, and Mike's earlier slides about the new building illustrate this, the power to make breakthroughs depends upon what you're working with. And so I'm just going to mention a couple. The Leonard Wolfson Experimental Neurology Center, which was a 20 million award in 2011, which established the first dedicated clinical research facility for neurodegenerative disease. Prior to that, there hadn't been anything like that. There wasn't a facility where treatments which were risky and with side effects could actually be tested safely. That was an amazing step forward. And I think it was the largest philanthropic award the Wolfson Foundation made at the time. The Drug Discovery Institute, partnership with Alzheimer's Research UK, established in 2015, one of only 3 in the country, and then the UK Dementia Research Institute in 2017. And it's worth pausing a moment to think that there hadn't been anything like this before that. And the idea for the institute was led here at Queen Square, and it became the largest single investment in UK dementia research ever, with UCL at its hub and its headquarters. But if I go back now to where does all this begin with, go back to 75 years ago, where was dementia when the institute was being born? So textbooks at the time said very little on the key and common neurodegenerative disorders, such as Alzheimer's disease or frontotemporal dementia. And they appear in tables like this from 1949, textbook then, pretty much as a footnote. Pick's disease, now called frontotemporal dementia, Alzheimer's disease, right at the bottom. And you might notice that they are a footnote underneath dementia due to hashish. And at about the same time, MacDonald Critchley, a previous dean of the institute, published this seminal work about the parietal lobes, which was still regarded as an absolute landmark. And it's a measure of the interest, and I show this because there had been a tradition, even though that there hadn't been therapies, and there had been this nihilism, there had been this real interest here in the brain, in cognitive function, and how it went wrong. It just wasn't being translated then. And if I now go back, fast forward 25 years to 50 years ago, here we have, building on that tradition of neuropsychological research, of understanding the brain, of that understanding from following and recording and observations on patients, made by Elizabeth Warrington, the selective impairment of semantic memory, what we now know as semantic dementia or semantic variant PPA, a subtype, an important subtype of frontotemporal dementia. These were detailed observations that relied upon these links. And then, subsequently, with the advances, again led from here, on CT and MRI technology, there was able to understand the imaging changes that were underpinning these observations, these disorders. At that stage, nothing to offer people. And so that leads me on to describe some of the advances in frontotemporal dementia at the IoN. I think it's a nice illustration. I could have picked other diseases where we'd see a similar arc. A changing focus from defining syndromes, subsequently showing their association with imaging and their underlying pathology, and I think Tony picked up some of those themes too. And then, with the understanding and the explosion here about neurogenetics, understanding their genetic causes, which is key to understanding the pathology and the drivers. The IoN has been central to many of the key genetic discoveries in dementia, and frontotemporal dementia is no exception. So the MAPT, tau gene, progranulin, C9orf, these are the most common causes of what is now regarded as a very genetic disorder, frontotemporal disorder. They were all major contributions from here. And then work, I would particularly say, being now hugely driven by the Dementia Research Institute, working in cell and animal models, underlying the underlying pathophysiological mechanisms and new targets for therapies, which is really should be and is the focus of the work at the Institute. And we're finally moving from this observing of diseases, of observing of people, to modeling them in animals through cohort studies, leading hugly important worldwide cohorts, GenFi just one of them, and I think Cath will say a little bit more about that. But those observational studies are now moving into delivering therapies, going from observing to treating. And what you now see here as just one example, in the bottom right of your screen, you can see a recent intrathalamic injection of a gene therapy, AAV granulin gene therapy, into the brain of a Queen Square patient from one of the families who carry a progranulin mutation that causes frontotemporal dementia, devastatingly, typically in their 50s. This video covers a 90-minute injection. I mean, it's truly phenomenal, isn't it? It's sort of Star Trek stuff. Turning now to prion diseases. And I don't think it's an exaggeration to say that the MRC Prion Unit, led by John Collins, has delivered a series of what are truly world-leading discoveries. Just picking some. Understanding prion strain biology, linking the cryo-EM structures with strain type, establishing the genetic susceptibility and resistance, which is so important, advanced diagnostics, biomarkers and staging, including seed amplification assays, which are now absolutely the gold standard in clinical practice, making a big difference to making a diagnosis to patients over the last decade and more. And then I think the response to the mad cow disease, or BSE, and variant CJD was a model of science-led public health risk management. And extending this prion paradigm to wider generation, again, something that Tony talked about, and establishing the principles for therapies and the outcome measures that are now in clinical trials. So you've already heard John Hardy, and this is the letter that I still have in my office that was written by this amazing woman, Carol Jennings, in response to research that Martin Rosser and John Hardy were leading. We're looking for families who might have Alzheimer's disease in their family. And this person, Carol, wrote, and very prescient, she said, "I think my family could be of use." And she was very right. And it's really, I particularly put this in to acknowledge the contributions of families, which, again, in that film you saw before, I truly believe is humbling. And Carol is a phenomenal example of that. She not only gathered all her family together, and it was an extended family tree, she persuaded them all to gather in her front room and hold out their arms and give blood samples. I think there were some that were a bit recalcitrant, but she sorted them all out. And then this led to this, the first mutation that had really phenomenal consequences to our understanding of Alzheimer's disease. The recognition that amyloid had a key part to play led to the amyloid cascade hypothesis. We went from models to imaging and to pivotal clinical trials. And in all these aspects, the Institute has made major contributions. We showed, by studying these families before symptoms, as I mentioned in the film, that there is this long pre-symptomatic phase to diseases. And not just Alzheimer's disease, we know now that is the case in many of these neurodegenerative disorders. And this, one level, is terrifying, but at the other level, it opens a therapeutic window to intervene when there is still good cognitive function left and there's good functional ability, which is what really matters to patients. Hand in hand with this, we've moved on to designing the first trials, and it was a tremendous privilege to be part of helping to design and then doing the worldwide image analysis for the very first anti-amyloid vaccination study. And the Wolfson and the biomarker labs have been key to improving diagnosis, which in turn, because you've got a more secure diagnosis, means the right people are going into trials and their biomarkers supported, as well as clinical practice. And finally, we are seeing the licensing of the first ever disease-slowing drugs for Alzheimer's disease, and they are anti-amyloid therapies, only licensed in 2023. It's a start. They're not cure. They're not a prevention. There's a long way to go. But they are hope for the families, and they're an incentive to accelerate the search for therapies, which is what Cath is now going to tell you about. Cath.
Right, so as Nick said, and as you heard on the video, we have our first treatments, and yes, it does bear repetition because it has taken many years to get to this point. And they are modest in their benefit, they have side effects, but what they also have is they have proved that removing amyloid from the brain, changing pathology, changes outcome. And that is transformational in dementias. In addition, what we know from these two studies is that it matters how fast you get amyloid down and how far you get amyloid. And so these studies, which we have been integral to in terms of the design of the drugs, the design of the trials and the conduct of the trials, indeed we're still doing trials with these drugs, really have laid the foundation for where we're going. But what do we do next to improve what's happening? And there's a very exciting number of new approaches that are happening within trials. I can only, and you've heard this from speakers so far, I can only give you a few examples of the fantastic things that are happening. And I'll try and make sure that they are relevant and directed by what's happening at Queen Square. One of the new methods of delivery, brain shuttle technology, is something we are leading at Queen Square in the large phase 3 trial of trontinemab. Trontinemab is the most advanced drug in this brain shuttle category. And these drugs basically, instead of passively diffusing across the blood-brain barrier, they are actively transported. So you decrease the amount of dose, you decrease the side effects, and you significantly increase efficacy. What you see on the bottom left is the early results from the early phase studies with this drug. So those early drugs dropped amyloid by around 80 to 90 centiloids, by that amount, over 18 months. This drug does it over 3 months, 2 doses. That's a huge difference, and there's a lot of excitement about what will happen in the phase 3 trial when that reads out. In addition, this is a platform not just for anti-amyloid therapies. This method of delivery will be useful across all sorts of large molecules in diseases of the CNS. Nick mentioned the importance of prevention and that going earlier is better. He very modestly didn't mention that he was integral, as was Martin Rosser, in the setup, the conceptualization, and latterly the running of the dominantly inherited Alzheimer's network. You heard about as being a UK-wide service for genetic Alzheimer's disease. Well, this is a worldwide collaborative platform for multiple arms of trials that has been running now since 2013, and we lead it in the UK. Because it's been running since 2013, some people have been on anti-amyloid therapies here for over 10 years. And what we've seen, giving this drug to people who are at risk of genetic Alzheimer's, so they are pre-symptomatic, if you give them high-dose gantenerumab, an anti-amyloid therapy, for over 10 years, and you match controls to them, you see a 50% reduction in their risk of progression to symptoms over that 10-year period. That models to a very significant delay in onset of symptoms, maybe even up to 15 years. And that is incredibly significant in potential. Importantly, it's the first time we've seen evidence that treating early before symptoms may lead to significant delay and possibly even prevention of symptom onset. But we are no longer looking at amyloid. That was a very important initial focus, but we've now broadened the search to multiple targets in Alzheimer's disease. The secondary proteinopathy in Alzheimer's disease, as you all know, is tau, and there's a great deal of focus on trying to target tau. There has been for a number of years. Etalanetug is the most advanced tau immunotherapy that's being assessed at the moment in clinic. And this drug, Ibrahim was talking earlier about bench to bedside. This drug was developed at UCL in collaboration with ASI by Rohan de Silva. And it has been given and led in an early phase trial at UCL in genetic Alzheimer's disease population. And we've shown in small numbers that you can, using this drug, by halting propagation or delaying propagation of tau from cell to cell, that you can reduce levels of tau in the spinal fluid, and that you can halt or maybe even decrease tau accumulation in the brain. And this drug is now in a large phase 3 trial back in the DIAN-TU platform that I just mentioned, trying to use it to prevent symptom onset in combination with an anti-amyloid therapy. In addition, we have led work here in terms of the most advanced gene silencing therapy in Alzheimer's disease. And this again targets tau, it targets the mRNA from the MAPT gene. And what we've seen is that giving this drug to try and reduce tau production leads again to a dose-dependent, sustained reduction of tau, all forms of tau, in the spinal fluid. And that was associated with not just a slowing of accumulation of tau, but a reduction of tau burden on PET in those individuals on drug. And so again, by decreasing production, it seems that we can lead to downstream impact on tau deposition in the brain. The initial focus has been Alzheimer's disease, and we are way ahead of the other neurodegenerative diseases in terms of the history that has gone into and the focus that has gone into this. But you've just heard about the amazing advances that are happening with frontotemporal dementia. And John Rohrer leads the Genetic FTD Initiative, which is an international collaboration. You see the sites there in the bottom left. That's crucial for rare diseases and trials in order to get enough patients into a trial for that trial to be powered to show effect. So this is critical to the success of trials everywhere in these diseases and is led by John at the DRC. In the last 5 years, we've been leading trials across all 3 of the mutations that you heard about earlier, and you saw that video of the first gene therapy in frontotemporal dementia being given to one of our patients in one of the genetic families. So there are great advances, although there is a way to go to get to the number of trials that we have in Alzheimer's disease. Sarah Tabrizi and her team, you heard earlier about this extraordinary story, which I think is a fantastic and very exciting story about the most positive drugs so far in Huntington's disease trials. She and her team have been real pioneers across multiple trials, and this is just the most promising one so far of a number. And this is the Unicure gene therapy AMT-130, which you see being given here in a video that's not dissimilar to the one that the FTD patient had. And this drug showed, as she mentioned in the video before, a significant slowing of clinical progression over time matched to external controls. So this could change Huntington's disease patient stories dramatically, and we wait and see with interest what the regulators say. We've heard a lot about collaboration, whether it's between academic and industry or preclinical and clinical. Selina mentioned earlier the crucial join up there. The Neurogenetic Therapies program is really designed to try and maximize those joined up areas, preclinical to clinical, academic and industry collaboration to add value to trials that are in neurogenetic therapies. And Ross Patterson is leading novel methodology that is being used in one of those trials led by the Neurogenetic Therapies program to use silk, which labels proteins in the CNS in real time to look at the impact of a drug on production and clearance of different proteins. And this is really going to help us understand mechanistically not only what happens with these proteins in the CNS, but also how do we refine trial design, how do we improve dose. There's no point developing any of these drugs if we can't give them to patients. And one of the biggest issues that we have in dementia is the resource to be able to give accurate diagnoses. We're very good at Queen Square, but Queen Square is not a typical memory clinic. And what we need is to try and enhance our ability to get accurate diagnosis everywhere, not just at Queen Square, and therefore get people into trials and get them the right treatments. And that recognition that clinically we really need to better access to MRI for better diagnosis led to a pilot study in collaboration with industry where we showed that you can use an accelerated protocol of MRI, 5 minutes, not 30 minutes, and you can show non-inferior, I'll show you some scans in a second, non-inferior results on diagnosis and monitoring. And that little proof of principle pilot study has led to a huge number of collaborations across CMIC, across physics, across a number of different disease areas, and across clinical and trial applications of this process, which is a great success story of how from a little seed, a huge program of projects and collaborations can develop. These are 2 examples of the potential impact of these MRIs that are accelerated. And as you can see, the ones on the right are the accelerated, the ones on the left are the gold standard. And you can see exactly what you'll need to see for monitoring and for diagnosis. And as a result of this work, there is now a potential multi-million grant that may come from this that's going to roll this out nationally to memory services so that they are able to use what we have tried to develop at Queen Square. Our expertise in clinical trials has been recognised and led to a 50 million grant from NIHR, investing in us to develop and lead the UK Dementia Trials Network. This is coordinated and hosted at UCL and UCLH, and the aim here is to make the UK the go-to place for clinical trials, improving patient access. Already, having started 2 years ago, we have all 4 nations involved in multiple sites. We have great industry engagement and buy-in, and we have a really healthy and expanding drug pipeline in early phase trials. There are multiple collaborative projects across charitable organisations and others. What's really important is we've shown that at the UK, we can do and deliver dementia trials faster than anywhere else in the world. We couldn't say that before. And that's one example of the timeline to set up and how it's been reduced in the past couple of years from an average on one study of 187 days to 42 days. Everything I've been speaking about and everything everybody else has been speaking about has only been possible because of our patients and our families that have chosen to take part in our research. And at Queen Square, research and clinical, as you've heard from all of us, are inseparable. And our participants aren't subjects of our research. They're part of our team. They work together with us. And many of the people on this slide, including Carol, who's top front and centre, are the reason that we're here talking about these treatments. And with that, Nick, I'm going to hand back to you to talk about the next 75 years.
So, I will just speak for a couple of minutes. We've heard a bit about the past, a bit about the exciting present, and a look to the future then. And to paraphrase Niels Bohr, it's always dangerous to make predictions, particularly about the future. So I can't emphasize how different the present is to 5, 10, 15 years ago, never mind 75. What Cath has described just would not have been possible here. It wouldn't have been possible anywhere. These advances are a world away. And from that therapeutic nihilism, we're now seeing people believing that there is a future, that there is a potential for treatments not just to alleviate symptoms, but to slow diseases. And I think, hope, believe that Alzheimer's disease, frontotemporal dementia, Huntington's, these dreadful, devastating dementias, causes of dementia, will follow other incurable, untreatable diseases that first started with something that was a bit effective, had lots of side effects, and then advanced. Whether you think about HIV, the first antibiotics, or some of the first cancers. So we also need to end the diagnostic nihilism, and finding therapies helps with that. But the next frontier after that, where I think we will be going, will be putting in treatments pre-symptomatically. That if there is this window opportunity, when people still have what matters to them, their ability to function somewhat independently or cognitively normal, that is the one we want to delay onset, slow progression, and maintain quality of life. And that is addressing both risk factors and treatments. They need to go hand in hand. But with all these advances, it's very easy to forget that there are many people where this will be too late. And for not just for now, but for decades to come, we will need to be able to provide care and support for those individuals and families that are and will be affected by these devastating diseases. And we need to offer symptomatic treatments and advice and help. And it's fantastically timely, and I'm really excited to be standing here at a time when we've just had completion of the Hillary and Galen Western Rare Dementia Support Centre, with thanks to the National Brain Appeal. It's been a long journey of raising funds, the AVPA, many, many other donors, some of whom I see in the audience here, and it'd be invidious for me to name them all, but you know who you are. This is going to be the first ever, if you like, a Maggie Centre for Dementia, the first ever centre where we will be able to provide support and care for families that is not done in a clinic room, that goes hand in hand with research and education that will make a difference to so many. But the IoN will continue to deliver advances and help to shape the future of dementia. That I have no doubt about. And that will range from fundamental mechanisms. I think we will understand a lot more about modifiers of progression than we do now. It's not just about finding causes, but what changes trajectories. We'll understand a greater the effect of co-pathology, which actually is the rule rather than the exception in these late life diseases. Not just how to detect the presence of co-pathology, to understand its effects and then to intervene to benefit. And this new building, with its amazing facilities, will be a powerhouse and brings the DRI together with IoN's neuroscience strengths. But key to all this, a little bit as Cath said, is partnerships. It's a partnership to the hospital and the institute, which you've heard about before, but it's partnerships with industry, with biotech, with pharma. And it's partnerships with the people who run the marathons, the people who, my own experience is that it is actually truly amazing what people will do. Cycle around Britain, climb Everest. That is the size of the challenge, but I think with partnerships and collaborations, we will really make a difference. So I'll leave you with the words of Carol Jennings. It's marvelous. In the future, it may be possible to prevent the disorder happening. And in this year, she was awarded posthumously a UCL doctorate. Thank you so much.
Thank you, Cath and Nick. And our next speaker and last speaker before the coffee break is Olga Ciccarelli. So Olga is the former head of department of the neuroinflammation, and she's also the lead for the BRC Translational Neuroscience theme. So Olga is a world-leading expert in multiple sclerosis, and her department is really using MRI and other biomarkers to improve diagnosis, improve mechanisms of disease, and obviously develop more effective treatments for MS. So today she's going to share with us the latest advancements in MS. So please join me in welcoming Professor Olga Ciccarelli.
Thank you. So thank you for the kind introduction. It is both an honor and a privilege to stand here and present and celebrate this special day where we talk about the achievements at Queen Square for research in MS and neuroinflammatory diseases. And these are the past and present and future of the departments who led research in MS. So at Queen Square, over the years, we were early starter, I think, in terms of MS research. And during the years, we have changed from experimental demyelination and early MS research in 1960s. We moved towards studying the optic nerve as a model of demyelination and repair in the brain. Then in the early 90s, and we studied MRI. And MRI had a huge impact on diagnosis, prognosis, and monitoring of MS. Later on, we moved towards advanced imaging, adaptive trial design, and novel biomarker. And looking ahead, I agree it is always risky, but I think we're moving towards a concept of precision medicine, earlier intervention, and prevention of MS. I'm going to quickly discuss 4 areas of interconnected research. The first one is diagnosis. The second one is insight into mechanism, which obviously is linked to diagnosis and translation. Then clinical trials, and then prediction and biological stratification. So diagnosis. Queen Square has really contributed to improved and increase the accuracy of diagnosis of MS through major donation of the UK MS Society, charities that has supported our research since 1983. And here was a vision of Ian MacDonald and the MS Society to create the first ever MRI scanner dedicated to research in MS. Later on, over the years, it was upgraded from 0.25 Tesla to 0.5 Tesla, then became 1.5 Tesla, more recently 3 Tesla, which underwent a major hardware upgrade. But MS is clearly diagnosed with using imaging, and MRI has contributed to advancements in all areas of MS research. So a transformation of the diagnosis of MS was led by Ian MacDonald, who was the first person in early 2000 to think of bringing together a group of international experts to define diagnostic criteria for MS. And the vision here was to introduce MRI in diagnosing MS. Prior to Ian MacDonald, diagnosis of MS was mainly clinical, based on symptoms and relapses over the years, and Ian MacDonald and his collaborators have introduced the concept of MRI. So in 2001, MRI-based criteria which use dissemination in space and dissemination in time of white matter lesions to diagnose MS. In 2005 and 2010, there were further revisions to this criteria, to which MS consultants at Queen Square actively contributed. In 2017, our Dean, Alan Thompson, led the international expert panel that introduced further revision to the criteria, but importantly, introduced the concept that oligoclonal bands in the CSF can substitute for dissemination in time. And in 2024, Alan Thompson also led this other international panel that has moved towards diagnosing MS on the basis of the biology of the disease. So this is really a step forward to move away from symptoms and move towards a period where we really do not need clinical presentation to make a diagnosis of MS, and I'm going to explain what I mean. And in these recent revisions was also important predominance of new biomarkers. The two biomarkers now introduced in the diagnosis of MS are the central vein sign and paramagnetic rim lesions. These are MRI lesions that are seen on susceptibility-based images. For central vein sign, you see little venules that cross the white matter lesions. The paramagnetic rim lesion is this hypointense rim on susceptibility-based images, which indicates the accumulation of macrophages loaded with iron, which indicates chronic active inflammation in MS. And these advanced biomarkers have very high specificity. So if you find them, this can help to rule in MS. So they are not that sensitive as white matter lesions, which the majority all MS patients have, but a very high specificity. And the impact on changing the diagnosis of MS over the years is that we can diagnose earlier and in more individuals. So you see over the years, the box with multiple sclerosis has extended, has eaten up a bit of clinical isolated syndrome, which indicates people who present, for example, with optic neuritis or a typical syndrome suggestive of MS, so the first ever clinical onset of MS. And more recently, we have introduced into multiple sclerosis diagnosis also non-specific presentation. Because you may have a biological MS, but clinically your presentation may not be that typical of MS. Despite that, with advanced, and as I showed you, with the specific biomarkers, you can diagnose people with biological MS, even if they do not have specific symptoms. So the impact of recent revisions of the criteria is that we can diagnose biological MS. And I think for the neurology colleagues, this is probably the most interesting slide of my talk. So we move towards a mechanism-driven framework for the diagnosis of MS. So there is strong evidence that there is, prior to a symptomatic phase, we have a pre-symptomatic phase of MS. And in this pre-symptomatic phase, we have people at risk. Unfortunately, we don't have specific single genes that can identify this population. This is a group of people with a genetic predisposition to autoimmune disease, a possible exposure to EBV, which then, in some cases, develop a biological onset of MS. On MRI, we can see white matter lesions. Then they can move towards a symptomatic phase of the disease, where in some cases they have atypical symptoms, not non-suggestive of MS or non-specific symptoms, and then later on, some of those will develop clinical symptoms typical of MS. So up to 2024, we could diagnose MS only in people with typical clinical symptoms. Now, with the new diagnostic criteria, we are able to capture also biological onset of MS, a prodromal MS, and making the diagnosis earlier in the asymptomatic stage help us to intercept biological changes, hopefully reverse them. If not able to reverse them, we can perhaps modify them in order to stop the disease or slow it down before the occurrence of typical MS symptoms. So this is a very exciting moment for the MS field. Second area of research to which Queen Square has really contributed to is a new insight into mechanisms of the disease. Wallace Brownlee has followed up people who 15 years ago entered longitudinal prospective cohorts at onset of clinical isolated syndrome. 15 years later, he saw that 15% of those has secondary progressive MS, and he could identify some of the markers that at onset of atypical symptoms can help to predict who will develop a more aggressive MS. And the BBC liked this piece of work, I think, by highlighting the fact that early MRI scan can predict a long-term prognosis. We also demonstrated as early lesion accumulation is a fundamental biological landmark of MS. You see in this slide the first 5 years of the disease. People who would then develop a secondary progressive MS have the fastest increase in white matter lesion load. So this is really the window that we have to act on very early, the first years. Declan Chard has followed up cohorts for 30 years and shown that in addition to lesion accumulation, early brain atrophy is really important to predict people who will develop more disable disease. And MS, differently from other neurodegenerative disease, is a disease of the young people. So we have 185,000 people living with MS in England, and the mean age is 40 to 45. So this is young people who we can already, in this group, identify those who are more likely to develop progressive MS on the basis of early MRI changes. Claudia Wheeler-Kingshott and Amer Tusi pushed the quantitative MRI studies to identify microstructural changes outside white matter lesions. So MS is not only of the disease of lesions in the white matter, but there are important changes occurring in the tissue outside lesion, also in the gray matter, that really contribute to disability. And this can be studied with imaging. So it's a really unique opportunity to look at the microstructure with non-invasive tools. Ken Smith lab looked at the metabolic injury and hypoxia and demonstrated how preventing lesion hypoxia reduced delayed disability and gray matter atrophy in experimental models, and translated these studies on hypoxia to human studies and demonstrated long-term functional protection and reduce of chronic tissue loss. Jennifer Pocock and Jonathan Lesmy have exciting programs of research focusing on microglia and immune neuronal interaction for Jenny, and astrocyte signaling and myelinated axons for Jonathan, who are amenable of translation to human studies. The third area of research is translation and clinical trials. As I said, MS was an earlier starter. In 2000, we had the first high-efficacy monoclonal antibodies for reducing relapses and new lesion in MS. And David Miller led these international multi-center studies. And up here you see photos of people who currently lead the clinical trial unit, the MS clinical trial at Queen Square, Frederick Barkov, Ferran Prados Carrasco, and David McManus. But David Miller led this clinical trial with natalizumab, and later on this first oral therapy for MS, the dimethyl fumarate. As you see from in blue, the effect of this oral therapy on reducing lesions and therefore relapses in people with MS. The clinical trial unit has contributed as a central imaging analysis of more than 30 trials, some of which are really important trials. I wanted to stress the impact of this research. Natalizumab, around the world, more than 300,000 people have been treated with natalizumab. Dimethyl fumarate, I looked at yesterday, 600,000 people are taking oral tablet for MS. Interferon beta, all products were the first ever approved disease-modifying treatment for MS, between 1 and 2 million people. So really a massive impact, changing life of people with MS. More recently, Jeremy Chatway has worked on repurposing trials, and in particular simvastatin. Here, it was a collaboration started many years ago in collaboration with the Dementia Research Center, using imaging as primary outcome measure of a phase 2 trial for simvastatin. And these studies showed that simvastatin had an impact on reducing of about 42% the rate of brain atrophy in people with MS, then moved from phase 2 to phase 3 trial, which unfortunately was negative. But I wanted to highlight how from the beginning of phase 2 to the end of phase 3 was about 15 years research. So this opened up the need of running multi-arms, multi-stage trials in progressive MS, where we can start from stage 2 as smoothly move to stage 3 trial. And this should have the time you need to identify effective therapies. With Octopus, funded by the UK MS Society, Jeremy is testing different arms where people are treated with different treatments, and then at stage 1, a primary an interim outcome measure, which is based on MRI, will give the signal which one of these arms will proceed to stage 2 and which one will be dropped. Now, because MS was the earliest starter, we are really in an exciting time where we are not only trying to identify new treatment, but optimize treatment that we have. With new design, as multi-arms, multi-stage design, response over continuous intervention, we can now study how often to give a high efficacy therapy, for how long. Clinical trials are 2 years. Some of these patients have been on this therapy for 7 years, 8 years. How long do people need to be treated? What is the best dose? So optimization of already discovered treatments is a really hot topic, and the NHR is particularly sensitive to this because there is a feeling of over-treatment in MS, and this is really a moment when innovative trial design can help to identify the best dose, the best frequency, and the best length of treatment for people with MS. The fourth area of research is about prediction and biological stratification. Arman Eshaghi, you will hear him speaking later, has used MRI to redefine subtypes of MS. So before this work, MS was really characterized on the basis of clinical symptoms and clinical course. Using MRI, we can define MS on the basis of MRI changes, moving towards an MRI-based classification. We have hundreds of thousands of MRI scans collected in a Q-square. The challenge is how to analyze them. Some of these scans are 20 years old, so they don't have a very high quality and they would fail the most advanced imaging pipeline. Therefore, Arman has developed this new imaging analysis method which is able to extract a brain region from any single MRI contrast. And The Guardian spotted this study as a very interesting new definition of MS using biology. Future work, we have set up a new cohort with during my NHR research professorship to look at people who initiate new disease-modifying treatments and follow them up over time to identify who are people going to respond to this treatment. With AI, this is obviously a very complex problem, no? Who is going to respond to a treatment? So we have used AI and identify a method that can predict in individual cases who is like to respond. Subject 1 responded successfully, had no disease activity after 2 years, and AI has successfully identified it. Subject B did not respond, AI successfully identified it, but patient C was a wrong prediction. The interesting aspects of this method is that for each prediction, give a level of uncertainties. So you not only have a prediction, you're going to respond or not to respond, which is perhaps simplified, but this is the uncertainty of the prediction, which is really important if you're counseling people about what the next decision to make. And this is the importance of this work is that you really need to put together all the biomarkers that you have in this cohort, not only clinical grounds, not only MRI, but also OCT, serum biomarker, and so on, including genetic test. So our community, several research groups, a lot of very young and exciting, hard work PhD students and postdoc. We are really grateful to all our funders, some of whom are here today. Very grateful for charities to donate their resources into research in Queen Square. And looking ahead, this is very positive period for MS research. We're moving towards earlier intervention and prevention. Thank you to all these people who work very hard, and thank you very much for your attention.
So thank you, Olga, and thanks to all our speakers for a fantastic first session. So I think now it's the coffee break.
Okay, great. So we've got one more talk for this session before we move to the panel discussion. And we're really privileged to have another fantastic double act, this time to discuss some of our contributions in neuromuscular disease research, diagnostics, and treatment. So it's my real pleasure to introduce Professors Mary Reilly and Pietro Fratta. They bring really complementary expertise across disease domains, everything from peripheral neuropathies through to motor neuron disease. And within those domains, a similar theme to what we've heard this morning, bringing expertise and outstanding contributions in everything from fundamental biology and mechanistic understanding through to diagnosis, through to new treatments and improved clinical care. So I'm not sure who's going first. I think Mary. So welcome, Mary. Thank you.
Thank you very much, and thank you very much. Pietro and I are going to present on behalf of the Neuromuscular Department, and it's a huge honour to be able to celebrate 75 years of the Institute of Neurology. And I love to be reminded that the hospital was set up by 2 women. You know, a lot has happened since then, a lot of lessons, but I love to see that.
So we've heard a lot this morning about diseases of the central nervous system. That is diseases of the brain and spinal cord, Alzheimer's, multiple sclerosis, dementias, Parkinson's disease. But of course, if you have a brain but you don't have arms and legs, there's a lot you can't do. So we're going to move now to the peripheral nervous system. And we have nerves that take sensation from the skin to the spinal cord, sensory nerves that go from the spinal cord to the muscle and motor nerves. And those motor nerves have very important neuromuscular junctions and the muscle itself. So when we talk about neuromuscular diseases, we talk about neuropathies, nerve diseases, neuromuscular junction diseases, and muscle diseases. But of course, motor neuron disease, which Pietro Fratta is going to talk about, transcends the boundaries. It has lower motor neurons, upper motor neurons, and the brain. And we all came together into a new department in the Institute of Neurology in 2019, so the new Department of Neuromuscular Diseases, to reflect the importance of this area. So I'm going to talk about the neuropathies and the myopathies, and Pietro's going to talk about M&D. So neuropathies can be acquired or inherited, and they're diseases of the nerve, and myopathies can be acquired or inherited. But when we talk about muscle disease, we include two other groups of diseases. Now, every cell in the body has most of its DNA in the nucleus, and most of the mitochondria, these batteries, most of the influence of DNA on them is from the nucleus, but they also have their own DNA. Now, the eagle-eyed among you will see that I got this picture from a mitochondrial expert, and therefore their skewed view of the importance of the size of the mitochondria versus the nucleus is from them. The second area which is very important are channels. Channels are important to all membranes throughout our tissues. Ion channels, chloride, sodium, potassium. But because the muscle is such a huge organ, both mitochondrial disease, which is multisystem, and channelopathies tend to present or have a major part of the phenotype as muscle. And hence, we include these when we're talking about muscle diseases. Now, there's this very strong history, arguably the strongest history in Queen Square's neuromuscular diseases, such that people used to joke about the National Hospital for Peripheral Neuropathy for many years. And it's a good title, I think. So two huge figures in this were P.K. Thomas and Anita Harding. P.K. Thomas led the development of peripheral neuropathy. That is both the histology, the clinical features, the neurophysiology, and the genetics. And the exciting thing in the last century was the fact that they developed neurophysiology. They could measure nerves, and they developed pathology. And therefore, a huge number of people were attracted into the field by those advances. Anita Harding, who sadly died very young in her early 40s, has been mentioned and will be mentioned. She was a huge pioneer and really is thought of as the mother of neurogenetics. And one of the diseases they studied was Charcot-Marie-Tooth disease, which is one of my areas of interest. And although it's a rare neuromuscular disease, it's the commonest inherited neuromuscular disease. And usually presents with length-dependent wasting, weakness, and sensory loss. And PK described the original clinical features. He did a lot of the work with measuring the neurophysiology, and he and Anita really defined the clinical features of dividing them into the axonal type and the demyelinating type, and that still is the basis of the whole clinical practice today. Anita took it further then and began to look at the genetics of this, so they really had a huge and lasting impact. But Anita crossed many diseases. Her interest in neurogenetics, and her second biggest disease was probably mitochondrial disease. Now, John Morgan Hughes was one of the consultants in the hospital who really drove the research into mitochondrial disease, both clinically and pathologically. And he and Anita described the first association of an abnormality in mitochondrial DNA with human disease. And this is the basis of a huge amount of research today. So I cannot emphasize how important the history is from the hospital and from the institute in this area. There are other very important figures such as Roger Gilliatt in neurophysiology and Hugh Bostock, who invented really the whole field of excitability. So there's a lot of technical developments as well. So what have we been doing since then? Well, really, it's all about translation. You've heard that repeatedly, and unapologetically, that's what we're all about. So many of the diseases we deal with are hereditary diseases. We start with patients. We find the causative gene. We work out how that gene causes the disease. We develop candidate therapies for our preclinical models. We get trial ready, and then we do clinical trials and develop treatments. So what we've been doing for the last 20, 30 years, a lot of it is looking for genes. And of course, the genetic story has expanded. We know a huge amount more about genes than we did. So our genome is huge, and it's really interesting that only about 2% of our genome is the exome. That is, it codes for all of the proteins that make up us all. And actually, we all knew that evolutionarily it would not make sense to have 98% of our genome that wasn't doing something. But we really, until recently, didn't understand all of this non-coding part of the genome. And technology has driven this. So if we take Charcot-Marie-Tooth disease, first gene 1991. Late 90s, a few other genes, and we were doing sequential single gene sequencing. About 10 years ago, targeted panels. Then the ability to look at the whole exome, that 2%, and then the ability to look at the whole genome. This mirrored the finding of new genes, and we've been very lucky to be able to have found many of the genes here in Queen Square, such that now if we look at the neuropathy genes, we look at about 256 genes. And the NHS led, I think, worldwide in seeing could whole genome become the norm for clinical practice diagnostics. And because of a pilot study in 2018, it became the standard for diagnostic practice in 21. So how good are we at doing it? So this is my cohort of patients and the inherited neuropathy group here, and we now get a 77% diagnostic rate. Now that's pretty huge compared to 0 genes in 1990. And that's using whole genome. And the exciting thing about the whole genome, it's allowed us to begin to explore the non-coding genome. And this is a disease I've been working on for decades, called canvas. And a lot of your steadiness when you walk around is determined by the cerebellum, the sensory nerves, and the vestibular system. And we couldn't find the gene for this for about 20 years. And this work was led by Andrea Cortese, who was doing a postdoc with Henry Holden and I. And he discovered a massive expansion in the intron, that is the non-coding region of replication factor C1 gene involved in DNA repair. And as you can see, this has led to a huge amount of work which Andrea has taken forward now to work out how this causes disease. But this kind of whole genome that we use now is called short read. So what you do is you do short reads and you stitch them together to have the map of the whole genome. But with short reads, every stitch can have a mistake. So the new kid on the block is long read sequencing. Now, this is not in clinical practice, but in research practice. And that is going to revolutionize what we can see. An example of this is this gene. This gene was one we found collaboratively with many colleagues around the world in 2020. It's a recessive gene that causes loss of this enzyme and increase in sorbitol levels that causes a neuropathy. We knew we had patients. This is a recessive disease with one variant but a high sorbitol, so there was a hidden variant. We couldn't find it by whole exome genome sequencing. And what Andrea has found, amazingly, that many of our genes have got a second copy of the gene, a pseudogene, which is inactive due to variants. But in certain cases, they're very close to each other in this loop, and it's allowed an inversion to happen between the pseudogene and the real gene, and this is the third commonest cause of this disease. Now, this is only beginning to be looked at in diseases, but this long-read sequencing will really change the way we find things. We couldn't see it. There's lots of stuff that's there that we just can't see. The second area we've been doing a lot of work in is being trial ready. Now, you hear this term, but in many of the neurodegenerative diseases, change over 6 months, a year, or 2 years is too slow for our clinical measures. And companies and patients doing clinical trials, you've got to do them in a year to 2. So we've been developing this. So one of the big developments in neuromuscular diseases, which we've led since 2009, mainly funded by the MRC, is developing a way of quantifying fat in muscle. If you have a myopathy or a neuropathy, as the muscle dies, it's replaced by fat. And these are cross-sectional through patients with neuropathy, showing mild, moderate, and severe. And I always think for neurodegeneration, breed in the brain or the spinal cord. If you see that, you know why we have to treat early. You have to treat early because you cannot do anything when you get to that. We've now been able to take this into all forms of CMT and show even in the most slowly progressive forms, we can quantify and measure change in 1 year such that we can do clinical trials. And we're now leading a clinical trial here with this as a primary outcome measure. And in children who have very mild disease, we can go to their feet and do it as well. On the mitochondrial side, many of you will know that the cardinal feature of diagnosis was muscle biopsy, which is pretty invasive and can be variable. And the mitochondrial team are taking imaging with spectroscopy and advanced MRI, so they have a non-invasive way of looking at muscle energy metabolism and oxygen use to get earlier diagnosis and for monitoring in clinical trials. So it's all about bridgeable biomarkers, change in biomarker that predict clinical change over a longer period, and that's where our clinical trials are going. But what are we doing about trials? Well, I'm going to give you 2 examples of major successes. These are the channelopathy service, and this service is led by Mike Hanna with Deepa Ryan and Vino Vivekandan. And this is where patients have an NHS-funded cohort of patients nationally here. They are accurately phenotyped, they are genotyped. Functional studies are done on the channel, and this has led to trials. And in this trial of mixelatine in a rare form of myotonia, this has been successful. It's gone through EMA, MHRA, NICE into clinical practice. Similarly, lamotrigine, gone through an investigator-led trial, gone through all of the steps and is in practice. So that is the whole way around from bench to bedside in neuromuscular diseases. We don't all have genetic diseases, we also cover inflammatory diseases. And the inflammatory neuropathy service is led by Mike Lunn with Aisling Carr and Junie Quay. And Junie Quay has really taken forward the clinical trials. For inflammatory, we had steroids, IVIG and plasma exchange. But now we have complement inhibitors all just going into clinical trials. So a new era for treating these difficult inflammatory diseases. What does the future hold? The future is all about treatments. We're a translational neuroscience place in Queen Square, and it's all about developing treatments. We still have to sort out some genetics. There will be single gene disorders, and we've had some very exciting advances in the last few weeks in solving a good percentage of the 25% that's not actually solved. Completely confidential stuff. But also, I think we're at the end of the beginning of genetics. We now begin to look at the bigger picture. What modifies our gene and our phenotype? Why do you have patients with exactly the same genotype but completely different phenotypes? When you give a drug like gene therapy, what modifies your response to the drug? What modifies your chance of getting side effects? And what is the complex interaction with our immune system, particularly when we come to gene therapies? But the big change is our ability to do genomic therapies. DNA gets transcribed to messenger RNA that gets translated to proteins. And up till 10 years ago, all of the efforts were on looking at how the protein causes the disease. And there have been some successes, but they've actually been very slow at delivering. The fact that we could go in and change our DNA is revolutionary. At the DNA level, we can permanently edit it with things like CRISPR-Cas9. We can deliver replacement genes to inactivated viruses. At the mRNA level, we can change it with antisense and siRNAs. We can splice it, we can silence it, although we need to repeat treatments because DNA is still coming in. And this is in practice. For spinal muscle atrophy, this is 10 years of using antisense and gene replacement therapies. And this review is led by Francesca Montoni, who works in Great Ormond Street and ICH, but also leads our Genetic Therapy Accelerating Centre here. And he has been one of the drivers in the development of that. And we now treat the adults and the children who are becoming adults here. And in my area, my original disease I was interested in, still am, is TTR amyloidosis, a systemic disease but mainly affects the heart and the nerve. And in England, even though there's about 120 mutations, the commonest mutation arose in northwest Ireland sometime before the famine in the 1840s. And it's the commonest in England for obvious reasons because of immigrants like myself. And that used to kill people in 3 to 4 years with cardiomyopathy. But the great thing about TTR is the vast majority of it is produced in the liver. So since 2019, we can treat it. We've antisense, we've siRNA, we silence the production of TTR, patients stop progressing, and now it's all about early diagnosis. And the first phase 1 trial of a permanent treatment with CRISPR-Cas9 has just been published, again showing efficacy. So that is in practice. But we just don't do that in Queen Square. We go back to the bedside and we try to do the more difficult diseases. So in neuropathy, in a rare disease of hereditary sensory neuropathy and work led by Hai and Zui, she's developed an antisense that is allele-specific, that only knocks out the mutant allele, taken that to an animal model, and now it's being used in a humanized animal model. And in the mitochondrial field, one of the big genes is a nuclear gene that causes a disease both in a dominant and a recessive fashion. And this is what we really call personalized treatment. For the dominant one, they're doing allele-specific, and for the recessive one, they're delivering the gene. So this is genetic therapies in practice as it is now, and it is the future. But it is a continuous circle of translation. When we get to a treatment, it doesn't stop there. A new circle starts. Can we improve the treatment? What do we do? What kind of treatments work best? And it informs basic science. The second commonest cause of Charcot-Marie Tooth disease is due to connexin 32 mutations. These channels weren't known to exist in peripheral nerves before we actually had these. And there are many famous neurosciences institutes in the world, but Queen Square is unique. The combination of the basic science, which is fantastic, and no apologies for using this diagram rather than the current building, and the hospital working together with cohorts of patients, with clinical academics, I think is rivaled nowhere. I moved here because of this, and if you ask colleagues around the room, the vast majority of the reason we all moved here is because of its translational focus. It makes it unique, and it delivers in a unique way. So I'm going to hand over to Pietro to talk about motor neuron disease, but just thought this is the inherited neuropathy group. And of course, I think it's important to mention that we're funded very heavily for all our science. Thank you.
Great, yeah, so my task is, I guess, to continue, right, the journey that Mary started across muscle and nerve and take you to motor neurons. I was thinking of how to give some continuity in this presentation. I briefly considered trying to match Mary's word per minute count, but I tried under the shower, just unable to. So how I thought I could give some continuity is actually by, the good thing about giving in slides late is I could actually steal slides from Mary. And so I'm starting from this, and again, highlighting Anita Harding, and definitely she's not known for contributing to motor neuron science, but actually she did in the 80s describe and put together what at the time was the largest cohort of a rare form of motor neuron disease called SBMA. And actually this was really instrumental in identifying the genetic cause for this in 1990 with Kurt Fischbeck and Alla Spada. And this was not just yet another mutation that was found. This was actually, along with FMR1, the first expansion disorder ever to be described. The CAG expansion with then, of course, Huntington and the scars to follow. And again, continuing that circle, of course, this was 1991, but in the following decades, Linda Griesbeth on the lab side and Mike Hanna on the clinical side put the seeds to research that was actually not happening at all in the UK on this disease. And this has led to actually the development of a center where we've been actually developing biomarkers that have been adopted in trials, and there's a whole range of work on the pathogenesis and therapeutic development. But the main form of the most common and the most aggressive form of motor neuron disease is ALS. And this is where, as Mary was hinting, it's both the lower and the motor neurons that degenerate. And it's a very tragic and usually very rapidly progressive disease. It can hit, the pictures below depict Lou Gehrig, who is still active. That's his kind of farewell speech from baseball. And so the US, his name is actually the name by which the disease is called. Rugby players in the US soon after retirement. In the middle, it's Mao, in kind of one of his latest pictures, clearly affected by ALS. And this disease was a bit really, in the 20th century, especially in the kind of middle part, neurology wasn't much about following up patients. This was a disease for which nothing could be done. And so what was the point of it? And so it was actually slightly ignored. And things started to change in the 1990s when, of course, rehabilitation, multidisciplinary care, these concepts started to emerge. And it was then that Robin Howard actually started the clinic for MND here at the National Hospital. And then all these other people kind of named there took part in it and make it grow into one of the largest centers in the country. And that was on the hospital side. But on the institute side, what happened was something actually really peculiar, because a number of scientists who actually not necessarily had the main interest in neuromuscular disease coalesced around ALS. These basically scientists who had kind of wide range of interests coalesced around ALS. And this really created a quite special moment in space and time. It was definitely the largest, I'd say, European center for ALS in the early 2000s and 2010s, and I was really privileged to do my PhD in this context. And so what I'll tell you is not a chronological order, but kind of snippets of work that was done by this group of people. And I'll start by mouse modeling. So Lizzie Fisher is probably the person that has generated most mouse models in ALS in the world. And still, with this track record, probably wouldn't define herself as an M&D researcher, but as a mouse geneticist. And actually, this has really shaped how she has done work, never really following generating a phenotype, but always put an emphasis on the genetic physiology. And so her body of work really initially was this huge effort by doing random mutagenesis in mouse and uncovering new genes involved in motor neuron survival. And then, in the subsequent phase of work, doing knock-ins, but not conventional knock-ins. These are knock-ins in which she was actually putting the whole human gene in the context of the mouse locus. And although in 2026 this may sound kind of normal, maybe soon to be a drop-down menu, right, in a kind of CRO that you can request for, when this work started, this was really thought to be just the privilege of deep-pocketed pharma, and definitely not for neurodegeneration. They were just doing it for large immune-scale projects. So these were really pioneering models, but then, of course, what do you do with models? And so I'm exemplifying, giving one example of things to be done with models, and it's really work led by GP Schiavo, and again, a person whose research fame is probably more linked to neurotrophins, axonal biology, not ALS, but actually it's that having these experts working on the disease that led to him and others in IoN to actually define axonal transport defects, both using cells in vitro and actually mouse models in vivo. And so, of course, axonal transport is essential to carry mitochondrial, RNA, and organelles in these very long cells. And it was not just about defining and describing defects, but then taking things forward and generating screens to identify molecules and compounds that can actually modify axonal transport. So to understand how this can impact disease and potentially how this can create therapeutics. And it's a very large area right now in the department. James Lay is applying this not just to MND, but to many other neuromuscular diseases. And then one really important finding in 2011 in ALS, there were genetic findings coming, each explaining 0.5, 0.25, 0.1% of cases. In 2011, this mutation in C9orf came out, and it was huge because it basically explained half of the familial cases. And it was a very large expansion in an intron, and this expansion was creating these RNA foci and also giving rise to new peptides. And nobody knew what was really contributing to disease and driving it. And it was really seminal work by Adrian Isaac's lab that managed, using Drosophila and human cells, to disentangle. And so with a really clever approach, he actually showed that definitely the proteins were driving the disease. And this really led and shaped what happened in the therapeutic development in the years to come. And his lab also developed an assay that was actually able to measure these dipeptides and was used in clinical trials to show target engagement. And so, building on this kind of biomarker problem, I mean, this, ALS is no different from other diseases. It's a problem. This is the progression of patients based on clinical scales. And you can see many short disease, some have variable duration, but even within their duration, it's really wiggly lines. You can imagine what a nightmare to do a trial and actually use a shift in these measures. And so it was long known that neurofilaments, molecules coming from neurons, were actually found in the CSF of patients. But it was really work from Qinghua Lu and Andrea Malaspina's lab, joined with Linda Greensmith, to develop an assay which had really some technical challenges at the time. This is from 2015, by which they could sensitively and quantitatively measure it in peripheral biofluids. And what came out was actually now normal, but pretty stunning. And so they showed that if you had high levels, your progression was faster, and if you had low levels, your progression was lower, the blue line versus the red line. And then this assay has been evolved and basically tested it in every possible neurological condition. But in ALS, it's been really significant because 2 years ago, there's been a successful trial for a genetic form of MND, and actually it's been this assay, so the lowering of neurofilaments, that has shown the early efficacy, and now this is included as an outcome measure in all ALS trials and actually trials across neurodegeneration. So this is kind of translation on the biomarker side, but I'm actually going to highlight now 3 kind of therapeutic approaches in the preclinical development. And this is work from Barney Bryson and Linda Greensmith. And this actually is a science paper that came out in 2014, but actually sounds as cool today as it did then. And so what they showed in that paper is they engrafted optogenetically modified neurons in the nerve sheath and allowed them to actually reinnervate muscle, and were able to show that shining light in them, stimulating them, they could get the muscles to twitch. And this was kind of the proof of principle paper. And then they went on to actually try and bring it forward, properly as a therapeutic approach. And so developing this using human cells, again, optogenetically engineering them so they can fire when light is shined upon, have them re-innervate muscle, and actually how do you determine whether they fire? Well, connect them to brain-computer interfaces, which of course are these days really exploding, and this actually possibly sounds still very challenging but feasible. And just as a note of comment to this, I think there was a lot of work for them to get to that proof of principle in 2014, huge. But I think nothing compared to what it took in the next 10 years to actually solve one after the other the problems that actually one faces in clinical translation. You can't choose the best finding to show your proof of principle. There's no way around it. You have to face them one after the other. And I think it's something to think about in the next 75 years of the institutes, how one can support these efforts, because they go definitely beyond the time of current grant schemes, of current programs. And if we want to really support kind of visionary and innovative science, I think we'll have to discuss and understand how we can do it. And so I'll take the last two slides to actually show some work from my own lab. And so this is, again, another kind of story that goes from a molecular observation to an experimental therapy, and it's a story of this decade. And so at the beginning of this decade, there was this large mystery in ALS. There was this huge GWAS signal in gene called Lung13A. Nobody knew what it was caused by, and it was these three brilliant postdocs and the students in my lab that actually identified that it was all due to a novel exon being formed in this gene. And when you got this novel exon in Lung13A, you got no protein, and so you lost this crucial synaptic protein. So this was a paper we published in 2022, and then work continued by actually, given that it was a splicing defect, to develop antisense oligonucleotides that would rescue the splicing defects and actually restore the protein, as you can see in those green versus red arrows. So we got to restore the protein and got to show that actually if you do that, you get more spikes in these traces, which actually mean you get synaptic function to be recovered and active again. And so, of course, all this, developing ASOs, functional work, molecular findings, this was all done in the lab, but to take it to patients, there's need of further funding, toxicology, and other things that are beyond our skills. And so this led to the creation of a company called Trace Neuroscience. And it's actually quite timely because the first patients were dosed with these ASOs last month, meaning in June 2026. And so we're at the phase of course, the trial having started from the finding of the early 2020s. And of course, I don't want to temper excitement for this trial. This is great. This is what we're doing now in the clinic. But I think our job in the lab is to think of the next generation of treatments, think even further ahead, thinking bigger. And so I'll share an approach that is really, I think, exciting, at least we're very excited about in the lab, which is a gene therapy approach, again, for all of sporadic ALS. And so what we described is a technology that allows to actually target and get gene therapies to be expressed just in the right cells. And by right cells, I don't mean just in motor neurons, but just in motor neurons which are affected by disease. And why do we think this is crucial? It's crucial because a lot of the problem in therapeutics is toxicity. And so if your therapy is not expressed in millions of cells or billions of neurons, but is expressed just in cells that are diseased, of course, it's much easier to solve the problem of toxicity. You don't need to think of what is this going to do in all those normal cells for years. And so, our vision is to actually and you can see that it works, up in the control, the red is the therapy, there's no therapy expressed, and in the disease setting, that's the spinal cord, the red highlights just in the motor neurons that have TDP dysfunction. And so the vision is to develop therapies that we can give very early on, possibly even at a pre-symptomatic stage when people are not affected, because they stay quiet. They go all over, but they stay quiet. And as soon as cells have a dysfunction, then the gene therapies activate, and potentially, if they do their job, they shut down again. And so with this note of optimism, I'll bring up a couple of pictures that are actually indicative of the life of MND at Queen Square. We have an annual symposium now that's been running with really incredible international guests every year, and that's the photo on the left. And then we have a really important day in June in which researchers, patients, carers, healthcare professionals get together to actually discuss science. And under here, of course, this is work of many. There's no specific funder, but I'd like to, of course, thank, the institutional funders that have funded us, the UK charities, the MNDA, My Name's Doddy, LifeArc that have been funding lots of the translational projects and beyond. And this is again instead the wider department. Mary already showed these slides, and this is the neuromuscular department at its annual day. Thank you very much.
Hello everyone, my name is Susie Meyer and I am the Senior Enterprise and Translation Manager for the Queen Square Institute of Neurology, and I am delighted to be chairing this next session.
One of my patients said, I love coming here because it's the hoping machine. That's what we are. We are the hoping machine, but the hoping machine is going to find treatments, and that's what we're all focused on. Advanced therapies give hope for future treatments for previously incurable brain diseases. They're called advanced therapies because they are typically genetic therapies and they are complicated to deliver, but they make big differences to disease. Advanced therapies are no longer a distant idea. They're becoming a practical route towards treatment, and Queen Square is helping to drive that change.
Queen Square is a very innovative place for translational neuroscience because you have both the brilliant preclinical scientists that can develop therapies, but as well you have clinicians that can help to shape that research. For me, this is what Queen Square represents, bringing together science, clinical need, and collaboration to build treatments that can truly change lives.
Advanced therapies in practice for patients tend to be therapies that are delivered directly into the brain in the context of a gene therapy. A virus is used to package important information. It's injected into the brain, and the virus then releases their package, and this causes the nerve cells to produce forever a protein that switches off the gene that causes the disease with one single treatment. And they've given so much hope. I work on Huntington's disease. It is a devastating genetic dementia, and we've just had a very exciting gene therapy result. For the first time, delivering this gene therapy deep into the brains of people who have Huntington's disease, we showed we could slow the disease by 75%, which is huge. And that was a single shot of a viral gene therapy delivery. It's just the beginning, but it tells us that we can make a big difference to these previously incurable brain diseases.
Here in my lab, in my department, we developed advanced therapies to treat epilepsy. What we are doing in gene therapies is a bit different. We are not trying to correct the cause of the disease, but we are correcting the symptoms, so the seizures in the patients. The treatment options at the moment are drugs. Drugs are taken orally or given systemically, so it's affecting all body and all brain. In our case, we are treating precisely the brain cells that are responsible for the seizures. You don't have any side effects of other organs, and you can treat the millions of patients that at the moment are drug resistant, have no treatment, no options. So despite many drugs have been developed, still 30% of patients are drug resistant. We want to give an hope to these patients, so now we're going to the bedside to see if this therapy will work in clinical trial.
Bench to bedside in real terms means translating the excellent scientific advances that are discovered here into protocols that will eventually inform clinical trials and hopefully one day patient treatment. We have amazing facilities here. We can perform clinical trials to a very high standard to truly put them to the test to see if they can change people's lives. The field has come so far. Things that have built on years and years of patient research, clinician scientist discoveries are starting to bear fruit. To be part of the next generation of Queen Square research, it's a privileged position to see the patients that we see come from all over the country, hoping for change, hoping for advances, and seeing the tangible advances coming. It is really inspiring.
We are working 24/7 to try and develop therapies for all of these devastating diseases that we look after. The work that we have done in Huntington's disease, I think, has got relevance for other brain diseases like Alzheimer's, Parkinson's. I think we'll end up with therapies for all of them that will have a lifelong effect. That's the aim of advanced therapies.
So as the video has highlighted, and Mike, Hanna and Mary Reilly have wonderfully already introduced, this institute is not just a world-leading research center, but also focused on translational neuroscience and has led to the development of several spin-out companies. And I'd like to welcome our panel of academics who are involved in spin-out companies from the Institute of Neurology to kick off this discussion. So I'm going to direct the first question here as chair to Dimitri Coleman. Actually, sorry, before we get into questions, why don't we have each of them introduce themselves? If you could say your name, your title, your company, what your company does, and your role in the company, that would be great. We have to share mics because there's only two.
Thanks, Susie. So I'm Dimitri Coleman. It is on, yeah. So I'm here under two pretexts. I'm the Deputy Director for Advanced Therapies, Translation and Enterprise with the Institute of Neurology. But I'm also, together with some colleagues, including Gabriele Lignani, Stephanie Shorge and Matthew Walker, I'm the founder of a company called Epilepsy GTX. So this was spun out of UCL to develop gene therapy for epilepsy. And it recently raised a Series A funding round, which will allow a clinical trial to be undertaken starting in 2027. And I'm also the part-time Chief Scientific Officer of the company.
I'm Arman Eshaghi. I'm the co-founder of Queen Square Analytics. Queen Square Analytics is a contract research organization that provides AI-enabled services to pharmaceutical companies, to specifically multiple sclerosis. And specifically what we do, we subtype patients using AI to recruit patients into clinical trials, the trials that otherwise would be negative, by matching treatments with the underlying disease mechanism.
Thank you. Hi, I'm Alex Leff. My company I'm involved with my co-creator Jenny Crinian is a neurospeech AI. We do digital therapeutics, so practice-based apps for people with stroke and dementia. And I'm the Chief Scientific Officer.
Hi, I'm Serge Breezy. I'm a professor of neurology here, director of the UCL Huntington Centre. I work on everything Huntington's. And so I have spun out a company with Gabriel Balmas, who's a DNA repair expert in Cambridge. It's a UCL DRI Cambridge spin out. And we are developing small molecule therapies for Huntington's disease and other repeat expansion diseases. We had a very large seed fund, and we're about to start the Series A raise to take our molecules to the clinic. And I am the founder and the part-time Chief Medical Officer.
I'm Parash Kavnachev. I am involved with a company called Hologen, which is the output of a well-funded translational program that I conceived and led. Co-founder is also Geraint Rees, and we also have co-founders from KCL2. The focus of Hologen is the development of large multimodal deep generative models with a purpose in particular of being able to optimize interventional inference across an array of applications, but particularly in the context of pharma. That's me.
Hi, so I briefly kind of spoke about our work right now. I'm Pietro Fratta, based here, neurologist, and have lab working on RNA in the context of MND. And I'm a founder of a company, Trace Neuroscience, along with colleagues in Stanford. It's a US-based company that basically has one asset, which are those ASOs I spoke about, which are splice switching ASOs for ALS. And, the company's been around for now 2-3 years, and clinical trials, phase 2, have just started. So that's the phase it's at.
Hello, everyone. I'm Geraint Rees. I'm the Vice Provost for Research, Innovation and Global Engagement here at UCL. But it all started for me here back in 1994 when I came here as an SHO, and I've never really managed to leave.
So I wonder if we can start maybe, Dimitri, as Deputy Director for Translation and Enterprise, with a question about as we're celebrating 75 years of scientific and clinical breakthroughs at the Institute, looking back, what about the IoN's culture and history do you think most shaped your journey in innovation and company creation?
Thank you, Susie. So I think there's two things I'd like to pick up on. One is something we've heard about a lot during this morning, and that is the proximity between the clinicians and the benchside scientists. And the other one is the institutional environment. So proximity. So what is it about the proximity? It's not just sort of closeness geographically. It's also, I think, a number of aspects which perhaps are not represented so much in other environments that are aiming to do translational science. So, for instance, the complexity of the disease. A lot of the diseases we're talking about are actually collections of different mechanisms converging on similar phenomena. So, for instance, in epilepsy, the common theme is that people have seizures, but there are numerous different types of epilepsy. And understanding that complexity then leads to the need to step back and think about the appropriate disease model and possibly multiple disease models. Another aspect, of course, is engagement with patients. It's so much easier to do so through the clinics, and of course, involvement of patients in designing clinical trials, but also designing preclinical research is a theme that's been emerging. But it's just so much easier when you have the proximity. There's also, of course, access to databases, biomarkers, imaging biomarkers, which you might hear about more. And so this sort of two-way dialogue, I think, is absolutely crucial. Now, from the point of view of the environment, the sort of the institutional environment, something which may not have been clear from this morning's talks is that the Institute was only merged with UCL about halfway through its existence in the mid-90s. And I think at that time, I remember at that time, there was a lot of disquiet about the Institute sort of being diluted or asset stripped or whatever. The reality, of course, is that the merger has given access to some very, very important resources. For instance, the Translational Research Office, the Tech Transfer Office, which in fact I think just a couple of days ago rebranded as UCL Ventures, the expertise and also the access to venture capital, industry engagement, licensing opportunities and so on. So I think all of those come together to create an ecosystem that is really very promising.
Thanks, Dimitri. I wonder if we could move on to asking Pareshkev about when did you realize that your research could have impact beyond just a published paper and that it would require an entrepreneurial approach?
So in my case, it was not an accident. It was an inevitability because of the fundamental nature of the task. What we were building is complex models that necessarily require large-scale compute, and now the requirements of compute have truly exploded, and of course software and other infrastructure that is simply not sustainable purely within the academic domain. So actually we knew at the outset that this would need to have an enterprise dimension, and indeed that was the fundamental funding framework that was employed in the creation of the program from Wellcome Innovations. I think what is also important to realize is that this type of work, certainly at the time, could not have been pursued through the scientific domain alone, partly because there was so much skepticism about complex models and the belief that because they're hard to interpret mechanistically, then their scientific value is limited. And I think this is an intellectual prior that I think we have to consider changing, and it still remains to some degree. The reality is that complex systems require complex models if they're to be effectively controlled. And we do need to think about how we are going to bring in this type of technology across the whole of science. And this is, of course, particularly relevant, I think, to neuroscience, because if there's one thing we cannot deny is the complexity of the nervous system. A further point to make is that the natural path to translation, particularly in the context of clinical decision support, mandates a machine, a product, a device that, of course, has to be manufactured and maintained by somebody. And here, what is unusual is that you have models, essentially ideas. It's a conceptual, an algorithmic thing that nonetheless requires a machine in order to deploy and deliver in clinical practice. And the gap between the conceptualization within the academic domain and the application with the enterprise can actually be directly crossed. So direct academic enterprise relationships can be created and they can be very effective.
Well, speaking of gaps, actually, I had a next question directed at Arman, if that's all right, about many spin-out company founders, especially academic ones, describe a translational gap between academic discovery and then commercialization. So for you, and I'm happy for anyone else to sort of jump in on this if they have something to add, what was the biggest early hurdle that you had to cross to move from idea to company?
I think there's a generic issue. It's not an issue, just understanding of the fact that 99% of the experiments we do fail. They never translate, so that's just understanding that exists to start with. But specifically in case of Queen Square Analytics, which I refer to as QSA, I started at my early years as a postdoc here and also at the UCL Department of Computer Science, was attracting funding to get things off the ground, because at some point the ideas become non-academic, as Parashkev somehow mentioned, and also they're not really attractive for investors. So there is an area which many successful entrepreneurs manage to attract private investment, but that's not always the case, and sometimes government also don't fund it in a way that they find it attractive. The way I managed to pass that bridge, I was lucky. I had a customer which was a large pharmaceutical company, so we funded the company by customers. That's, I think, very organic, and it means you keep the ownership of the company, and still I own at least, I believe, 27% of the company, so still it's under our control.
Does anyone else I see a lot of nodding heads. Does anyone else want to add to how did you manage sort of the expectations of investors with your scientific
Yeah, I think
vision?
that's a really good question, because one of the things as academics is that and this is something that one has to learn, is that you spend all your time writing grants and papers and, like Ahmed said, things work and don't work. But then when you try and spin out a company, it's a whole different ballgame, because you have to persuade investors who are venture capitalists who basically, they're ex-scientists, so they're super smart, but you have to go into a room, you get 15 minutes, and it's like Dragon's Den pitch. And it's really quite an eye-opening experience, because we went and pitched to 20 plus investors. We managed to get 2 huge VC companies behind us, but it was a very interesting experience to basically selling your idea. And I think it's a bit of an anathema to academics, and then you just have to get your head round that you're persuading people to give you money to take your idea forward. So I think it was a very interesting experience for me, in that we needed a bit of coaching in how you managed to get people to give you money for an idea, which was very challenging.
I was just going to jump in there and say 25 is nothing. So what's also rather frustrating is when you get an investor finally to bite, but then they'll say, but they won't lead, they'll only follow. Yes. So there is this venture capital investment model where somebody has to put a price on the share, and so sometimes there's a lot of people willing to follow if another investor does actually make the commitment. So that can be very frustrating. Yeah.
Can I direct the next question at Alex, actually, about, keeping all of this in mind, how can our Translational Research Institute then better fuel the global ambitions, I suppose, of the next generation of spin-outs?
Yeah, well, I can really only speak from my experience with Jenny when we spun out from UCL just over 18 months ago now with these digital therapy apps. So they're practice-based apps. We know that people with brain injury can improve, even people with dementia can improve their cognition if they practice the right thing. Something that they want to practice makes a difference to them. So with sort of standard funding, we created a lot of these digital neuro interventions, as we call them, but they're basically apps. And it just got to a point where we were having, hundreds of people, maybe a few thousand people using them, but we really wanted to develop reach. Hundreds of thousands of people could potentially benefit from these. And it took me a while to realize that we weren't going to get there with standard funding. They're very happy to fund the idea. You get the paper, you do the so-called proof of concept, which is a phrase that drives me wild, because then you leave the translation up to somebody else. But the translation into clinical practice, that's the hard bit. We can all do the science. We can all have ideas in the bath like this. So that's when we had to go to external investors, and it has been a real challenge. I think, as Sarah mentioned, doing pitches. They almost accept the scientific idea. It's like, how are you going to make this work in the real world? Who are your competitors? What's your pitch deck? What's your cap table? I'm like, what's a cap table? I think helping people understand that space is probably the most helpful thing, because although we're academics, reasonable intelligence, it's just a completely different world. And people have talked about venture capitalists, but there are other people, angel investors. There's other ways of getting the money for projects that perhaps don't have this kind of 10x or hockey stick takeoff. And that's one of the things I found with UCLB, which I think has just changed its name in the last few weeks, and Geraint might want to speak to this. I think UCL needs to have a bit more ambition to help things that are not sort of physical products, so things that aren't drugs, things that aren't machines, things that aren't implantable devices, but digital technologies. I think UCL is a little bit behind the curve on how best to spin those out. We did eventually spin out with a good contract, but it took a while to organise that, so that could perhaps be streamlined. I don't know if Geraint wants to talk about that, but he's going to talk. He's going to talk later when you've all forgotten my question. Yeah.
But I think that leads nicely onto my next question, which is I'd like to first direct it at Sarah, but maybe to the rest of the group as well, as we've seen you a lot in the press this year. We know you have a very busy research group. How do you manage that with all of the challenges that you just spoke about building a company?
I think I didn't have any plans to spin out a company, but Gabriel Balmas, who I work very closely with, who was a cancer biologist, had spun out a company with Steve Jackson, who's very big in cancer. And they had spun out a company and sold it. And so Gabriel is a bit of a serial entrepreneur, and he was like, Sarah, we need to do this. And I was like, nah, business is not really my thing. But I became frustrated with this different working with biotech and pharma, often the speed of that they were working, they not really understanding the disease, always wanting a quick result. And so I thought, well, actually, I want to control what I do. So I think the way you combine it is that you've probably heard from everyone, and in like the talks this morning and from Pietro's talk, that your research in the lab is what spins out the company. So you have a finding in the lab, you think it's really interesting. Ours was a genetic modifier that had a very large effect, a bit like Pietro's, a large effect on Huntington's disease progression. It's a mismatch repair gene. And so we're developing small molecules to target that mismatch repair gene that can be delivered and distributed to the whole brain that will complement other therapies. And so I think your research, the way to combine it is that your research has to the research in the company has to be close to what you do in the lab, because otherwise you can't combine the two. There's not enough hours in the day. So I think the most important thing is when you spin out that you keep your research in your company aligned with what you do in the lab. So for example, in the lab, we do a lot of mechanistic basic science that actually supports the premises of what's happening in the company. So it's very much a two-way thing. I think UCL support this, and UCL Consulting, UCLB, have been great. The DRI have been great. And actually, there was a lot of hand-holding about how to take this through and learn. And I would say it's been a very good, with the DRI and UCL and Cambridge, a very good environment to do this. There's a lot of support, there's a lot of guidance, and there's a lot of great science and ideas. And I think that we're in a unique position to take that forward. But I think lessons learned is it has to be closely aligned to what you're doing in the lab, so you can actually make things happen faster, and they're both very much synergized.
Armin, did you want to add to
Yeah, just to add to this, how you manage your time, the other part is just understanding that business is a different ballgame. So it was mentioned many times. And the way we succeeded at the time was the UCL business leadership. So you need to pay people, so you don't expect doing anything for free. We managed to recruit, for example, a CEO who has been successful previously at UCL. So I think there's a lot of support there also. You don't need to do everything yourself.
Pietro, do you want to add anything to that? Because the next question I was going to direct to you is kind of along that vein of how did UCL's internal structures such as our TRO or UCLB or our translational ecosystem support your founder's journey?
Yeah, I mean, I think I really want to, I guess, echo what Sarah just said. And, I'm happy not to refrain to heavily criticize many aspects of UCL's life, but actually, in this context, it's really been great. I think what Sarah was saying about hand-holding, that's kind of how it's been. So, first of all, I realized that UCLB, now just rebranded, UCL Ventures, is really, we should be kind of proud of it. I've interacted with other institutions as well in the London area, and I think definitely UCLB has a significant experience, and that shows. So it's really about initially getting to work with them. So there's that hurdle, as everything, to convince them, for example, to file your patent. But once you're over that, it's been good. They kind of let you take decisions of directions, but at every decisions, they're kind of beside to guide them. And so that's been a really positive experience in my view. And when this happened, there was nothing locally. I think what Dimitri has set up now is going to make things even better. At the time, there wasn't much local support, so I think that's going to make it better. And the last comment is actually back to local support. I think many things in UCL may have a dysfunction if they're fully centralized. And UCLB or UCL Ventures is centralized, but you really develop a relation with one person who starts knowing your portfolio. We filed now 6-7 patents with the same person, and so it becomes, a text message relationship, and so that makes things easier. So as always, when there is a person and not a black box of work tribe, things are smoother. That's my message.
So I suppose I can open this up to anyone about what advice would you give to other researchers within the Queen Square Institute of Neurology who are curious about commercialization but don't know where to start?
So again, I was we started QSA when I was a very early postdoctoral fellow. So I would say, as a student, if you're a PhD student, if you are a postdoctoral fellow, is UCL Innovation Enterprise, which is geared towards education. It's different from UCL Ventures, but that's the area you want to start with, just to understand the process, how it works. But if you do have an idea, a patent, then that's a UCL Ventures, but you can work with them on an IP. So that's the work you've done. You finished the work. You think it's going to appeal to the customers. You define the customer. That's the time to go to UCL Business.
Well, I was going to say that the obvious thing is to come and talk to you, because so you offer an enormous amount of signposting and initial advice, and I'm very happy to be contacted as well. Of course, just to put things in context, of course, we sitting here are a few of the people who have embarked on this sort of entrepreneurial trajectory with some success. I'm sure there are people at the Institute of Neurology who have been frustrated that their idea did not lead to a patent, or the patent was not picked up by someone. At the same time, filing a patent is only the first stage, and sometimes you don't need to file a patent in the first place. I think the first patent that we filed, we assumed that industry would come knocking on the door. That wasn't the case. We realized that industry was not attuned to what we were doing. We were sort of ahead of what they were thinking about. We knew more about the disease. We knew more about the mechanisms, and that's why we had to go down the company creation route in order to advance the project with I think some success. Although, yeah.
I think just to echo that, I think that's why we did as well, because it was realizing that your disease better than anyone. But I think the UCL also, they've mentioned UCL Ventures, UCLB, the Translational Research Office here, led by Jane Kinghorn and her team, have been amazing. They have helped with MHRA, they've helped with so many different aspects, and they are very available. They have huge expertise, huge industry expertise, and they've helped with applications that are academic industry applications to help move company research along. I would say that in my discussions with lots of VCs, if you've got an idea, I think, personally, I think if you have, and David Altshuler from Vertex wrote a very nice review about this, and I quote it, but I think if you have an idea and it's supported by human genetics, then the chances, when Mother Nature's done the experiment and the date your experiment is supported by human genetics, the chances of the drug target and the development, you have to develop a drug, but there's a much higher chance of the actual drug mechanism working because human genetics supports that target. And that, for me, has been a very driving, we've heard a lot about genetics, but that kind of gene to target is really important. If you've got genetic data supporting your target, you're in a very good position. And I would say we had an idea, and it was an idea that managed to raise over $30 million. But that's, I think, the power behind that, is if you've got good human genetics with an idea, that's the place to start. And there's so many people at UCL who can advise you on how you take your idea forward.
I wonder if we could hear from Geraint to say a bit more about maybe what would you change or strengthen within UCL or within the Institute to accelerate the next generation of spin-outs, and then maybe a bit more about changes in the ecosystem that could accelerate the next generation of spin-outs out of UCL.
Sure, I'll have a go. I mean, I'd start by saying how inspiring I find it to hear all my colleagues on this panel talk about their experiences, about how they're transforming the world, and also saying nice things about how people interact around UCL. I think reflecting on that, those of us who've been here as long as I have in Queen Square would say that is a complete transformation from the 90s, where a panel like this would be literally inconceivable. So I think it's hard to understate the cultural transformation in being able to talk not just about basic science, not just about clinical translation, but also about entrepreneurship. So what would I change? In a sense, nothing. Please carry on. But I think, to try answering that question seriously, what I'd say, reflecting upon, say, some of the lectures I just heard, and you've heard this morning and will hear this afternoon, is for this institute, science isn't the limiting factor. It's deal flow or flow of those deals. That is, there is so much great science here. There is so much potential here, and it is getting even stronger over the years. So what I mean is we need to make deal flow happen. So we need to make it easier for people to declare their invention, to have those conversations we've talked about, to start down that entrepreneurial journey to being a founder. And we need to make that virtuous flywheel of people like my colleagues on this panel saying positive things in the community work its magic. Deal flow is important for a second reason, which is everything I've learned about entrepreneurship in my own entrepreneurial journey is it is really difficult to pick winners. And of course, many, many companies fail after founding. The mark of an entrepreneur is somebody who picks themselves up after that failure and just co-founds the next company, and then the next company, and then the next company. And so, at a university level, what we'd like to do is create as many deals as possible, because that's the best way that we are going to find the most successful companies that transform our world in the biggest way. So that's the first thing I'd do differently, if you like, but I'd just encourage it. Second thing I'd do differently, or I'd encourage, is permeability. I'm going to talk a little bit about ecosystem in a second in the closing remarks you've asked me to give. But permeability means allowing founders, like we've heard from on this panel, to be able to perhaps put their lab a little bit on pause, step out, be that chief scientific officer for a couple of years or however long or whatever, to de-risk that going across the boundary. And that works the other way too. Having scientists here, having people like Jane Kinghorn, who was mentioned, who of course had a long career at GSK before she came to work for UCL, is a way of bringing commercial expertise into the organisation and making that part of the culture. So I think permeability is really, really, really important. And then the third thing, which is more my team's jobs, is risk capital. Everyone on this panel has talked about pitching and money and VCs. And that is a rate limiting step, a really good rate limiting step for us, because the companies that UCL is creating are attracting around, roughly speaking, £3.5 billion of inward investment from VCs every 4 or 5 years. So we're having Series A's, Series B, Series A's that are routinely above $100 million, Series C's that are even larger, all over the place. So for us, capital and access to capital, particularly at those scaling stages, is really important. So that's my 3 fairy godmother wishes for the Institute of Neurology. It's about deal flow, it's about permeability, and it's about capital for the great ideas we've heard. But perhaps to your second question, which is more about the ecosystem, and perhaps to say a few things about what we're doing to help founders. I would say innovation doesn't happen in a laboratory or a clinic. It happens in an ecosystem. And an ecosystem is made up of two really important things, people and places. The people we've heard about, and we've heard a lot on this panel about the interactions between people that facilitate this kind of entrepreneurial journey. Because not everyone is an entrepreneur. Not everyone wants to be an entrepreneur. And to be frank, that's a good thing. It's about the population here having all sorts of people, just as we talk about having translational scientists and clinicians working alongside basic scientists, and that being a virtue. Having entrepreneurs in the mix is the third leg to that. So the culture has been transformed, but the place, I think, is really important to think about. And what I want you to think about, or what I'd love the Institute to think about as you near the move to Gray's Inn Road, is to think not in terms of your global collaborations and your global partners, important though they are to delivering scientific advances, but what's happening right in our own backyard. And that is, I don't think it's an understatement to say, one of the biggest transitions in the world at the moment is happening right around us in terms of what's happening with real estate around the north of the Euston Road, roughly speaking from Warren Street all the way down to King's Cross. Why is that happening? Well, it's a few things. Thing one, I'll again root it back in this institute. It's the founding in 2010 of DeepMind by Demis Hassabis, who was, of course, Eleanor Maguire's PhD student. I was second supervisor, but it was Eleanor whose PhD student it was. Got to claim that. And Demis founding DeepMind in Russell Square, then subsequently moving to premises just by the Russell Square tube, and then up to King's Cross, has totally transformed that district. Because that company has created a lot of wealth, and those co-founders have seeded other companies. Isomorphic is the most obvious one, and they all are staying in the local ecosystem. That in turn has attracted developers. Those of us who are long lived at Queen Square will remember how scuzzy King's Cross was back in the day. It's not now, it's all Sweaty Betty and Gales and that kind of stuff. But that transformation has given premises for those companies. And so what have we seen in the last 6 months? We've seen Anthropic take a big lease down the road from King's Cross. We've seen OpenAI open their first office outside, 500-seat office. So all the frontier AI companies are now coming to within 20 minutes walk of this lecture theatre. And that's breeding a second generation of AI native life sciences companies, of which Hologen is one, but there are many others. I've mentioned Isomorphic, Relation, and others. And that is all happening, not just because of Demis, not just because of DeepMind, but because of UCLH and UCL and the Francis Crick Institute. That's what these companies tell you why they've come to this particular part of the world, this particular part of Europe, because of the density of talent and the ecosystem that we're helping create. Now, I'm going to shut up shortly, but I think that's what all of this patient work over many decades, patient clinical academic work, coupled with the resources of a large university, is helping create what I hope will be the world's biggest and best ecosystem for doing this kind of entrepreneurship. My teams are super active. UCL is super active now in curating that ecosystem. So we'd say, what have we done at UCL? Well, we've created high growth, high value companies. We've heard from some of them on this panel, and there are many others in other areas of UCL. We're also moving on to creating entrepreneurs. So Alex did talk about support for computer science. So let's talk about the last two computer science professors who did a raise. That would be Tim Rocktaschel, who raised $500 million, and David Silver, who raised $1.1 billion, as seed rounds, by the way. So we are working on that and creating the kind of entrepreneurs who will power the future. But the final thing we're doing is curating just that ecosystem. We're learning a new skill at UCL, which is how not just to, as it were, figure out our own estate and our own relationships, but to help curate and orchestrate and conduct all of the many people in the ecosystem that need to interact together. Commercial partners, VCs, the local council, train companies, central government, all of the entrepreneurs, and many more besides. That's a new skill that UCL is going to learn, and I hope as IoN DRI moves into its new premises, really close to OpenAI, that unleashes the next wave of creative entrepreneurship in this institute, which has done so much for UCL and for patients.
Thank you, Geraint. We have time for maybe a couple of quick questions from the audience. Unfortunately, we don't have roving microphones, so I'll have to repeat them. But did anyone want to ask a question to the panel? No.
Stunned silence.
Ready for lunch?
Richard Fakovia.
Okay, one question here. Sorry.
I guess, as you mentioned, all the companies are moving in. Are there ideas to foster these collaborations between UCL?
Sorry, go ahead. The question was, do we have any ideas to foster collaborations with AI companies that are moving to the area?
Yes, we do. So I think the first thing to say is collaborations like that, in my view, are never corporate things that somebody in a leadership position like me dreams up and then comes to a room like this and says, who wants to collaborate? They're things that are investigator-led. So what we need to do is create the conditions under which the right investigators can create the right projects that the right companies might be interested in. And we heard about the Translational Research Office as one example in the translational arena of people whose job is to do that. So one example, for example, of a company we are working with, and I'll mention it because I was looking at Pareshkev, is NVIDIA. They don't yet have premises, but we do have a strategic collaboration with them. And that's investigator-led. In Pareshkev's case, I'm sure he won't mind me saying, is working on digital twins with Peter Coveney. But there are other investigators who are doing a series of projects there. The importance of that kind of a collaboration, that leads on to more things. So NVIDIA providing free access to CUDA, their development environment, for example. They're providing some compute. But they're also interacting at multiple levels with UCL, including at my level, but also at many other levels, to just build that kind of interrelationship and understanding that helps a large company understand the capabilities of a large university. So investigator-led, but university-supported is the model. And in terms of OpenAI and Anthropic, yeah, absolutely. But also Ineffable Intelligence, Recursive, which are the companies founded by David Silver and Tim Rocktaschel that I talked about, and many more. We haven't even talked about Quantum, for example. Quantum Motion are moving their premises to very close to Euston. That's John Morton and Toby Cubitt as co-founders. But they bring with them a host of quantum companies around them as well that we will build relationships with. Thank you.
That'll have to be the final question. Yes, in the back. Yes.
So
So we heard from the panel about going out to VCs and talking to 1 million VCs, and then they come in and do their due diligence on the company, and then they say, okay, we'll fund you for X million dollars or whatever it is. And that's great. And we're so desperate to take the money that we don't necessarily do our due diligence on the investors. So how do you do the due diligence on the investors to make sure that you're getting in bed with the right people?
The question was, I just need to...
I can answer that really simply. Of course, you're right. We do due diligence on every single investor, and that's partly what UCL Ventures is there to do.
Sorry, can I just repeat the question for the people online who won't be able to hear, which was how do we, from an academic perspective, do due diligence on the investors that we may be working with?
So the answer is we do due diligence on every single investor. And that would be part of the service that UCL Ventures provides to that. In fact, you might not realise that we do due diligence not just on investors, on every external partner. So somebody talked about the infernal black box of Work Tribe. So even at that pre-competitive stage within Work Tribe, we now do due diligence on every single new partnership in a structured way. It happens invisibly behind the scenes because nobody really wants to get involved in the bureaucracy of due diligence. But we do that because if we're going to work with external partners, whether they're investors or partners, we need to take, well, all sorts of things, including security, very responsibly. And that's the responsibility of UCL as an organisation, but we do it to allow people to be free to just realise their ideas and create great companies that change the world.
I just wanted to make a small low-level director comment, which is since Susie arrived and her predecessor, it has transformed our capability locally to really think about these issues and to get all of our PIs who are interested to engage. And I think it's a good model because the TRO is serving a massive population of biomedicine. And I think my suggestion would be more colleagues like Susie in different parts of the faculties that are relevant would be a really positive way to catalyse the ecosystem all around them. Because I think scientists, PIs don't always see the ecosystem. It has to be sort of shown to them. And that works best at a local level. So that would just be a comment to thank Susie for her great work.
Thanks, Mike. I think with that, we should also thank our panelists for a great discussion and head to lunch. So thank you very much.
So this session is being chaired by Dimitri and myself, and we're going to try and keep everybody to time, so some short introductions.
Well, it gives me great pleasure to introduce the next speaker, who's Gabriele Lignani, who will tell us about the Research Department of Epilepsy.
So, hello everyone. Thank you very much for inviting me. It's a pleasure. It's an honor to represent the Research Department of Epilepsy. I have strange feelings that probably is the first time in my academic career, also if it's very short, is that until this morning I was sure that this talk was 20 minutes talk, and just realized this morning is a 30 minutes talk. So I have more time than was I thought. That means that I will be on time. So it's a big honor. I'm part of this journey in this department in the recent history of the department. So I learned a lot during last weeks about the history of epilepsy in the Institute. And I want to thank, first of all, everyone in the department that helped me to put together this presentation. So, yeah, this presentation is divided in 3 parts, in 3 acts. The history, and you will hear about foundations that change epilepsy surgery, the monitor on epilepsy, new imaging, clinical trials, genomics, and global health. Then the second part will be about the present of the department that I decided are the last 15 years because I didn't know how to decide what is present. And he's about this world-leading epilepsy department that is integrating very well the clinical research and the experimental research. And then also you will hear from me a bit about the future, where we are going, how we are building the translational strategy of the department. So the sentence below is quite important, is the take-home message of this presentation. That is, for 75 years, the department has repeatedly changed how epilepsy is understood, and I will show how. our ambition for the next phase, for the future, is to change the natural history of the disease. So before I go a bit more chronologically, I want to say that in the history of the department, you will hear a lot about first discovery on epilepsy. And there are three patterns that run through all the 75 years of epilepsy in the Institute. Clinical standard, and these are new guidelines for clinical standard worldwide around surgery, video EEG, trials, long-term outcomes. The building of a technology platform dedicated for epilepsy imaging, EEG, fMRI, genomics, gene therapies, but also global responsibility and how this department changed the global, I would say, the global knowledge about epilepsy worldwide. So, actually, the history of the epilepsy in the Institute started 100 years before the Institute. So, as you heard us today, the National Hospital started in around 1859, 1860, and it was called National Hospital for the Relief and Cure of Paralyzed and Epileptic. And then in the late 19th century, the Queen Square clinical tradition was based, and one of the original clinical problems was epilepsy. And there was early stages studies, including people also like Euclid Jackson, that helped to shape the modern concept of epilepsy and focal brain function. Then another important time point in the history of epilepsy at the National Hospital is the establishment of the Chalfont Center, called the National Society of Employment of Epileptics, was founded by the Queen Square neurologist, and is the core still now of the partnership between our institute, the hospital, and the Chalfont, and now is the House of Epilepsy Society. So after the institute was established, one of the first important milestones in epilepsy was in 1952, where while a mix of it's the first one that showed the first British series of temporal lobectomy. That is the foundation of the most perform effective cure epilepsy surgery at the moment. And this was basically the starting of the epilepsy surgery history in the Institute and the UK. The second important milestone was 1972, where here the Institute has been funded the first and established the first video EEG telemetry unit in Britain. And that was a unique moment in which neurologists here were able to study patients, study the electrical activity of the brain and see also the patients and the behavior of the patients. This era between 1952 and 1977 was the establishment of modern clinical epileptology, how we know now, and is for Queen Square is where it was linked to the careful clinical observation, surgical intervention, and physiological measurement, and is a model of current epilepsy research. And it was really the building of the specialist hospital, specialist center around epilepsy. The next area of the history of the department in the Institute is an area that I call it from patient to population, because was not just about the clinical center and the patients in the center, but also was looking out on the population and worldwide, epilepsy worldwide. And was also an area that where Simon Shorvon, I think is here in the audience, was appointed by the Institute to start an epilepsy program here. And in 1983, was started the National General Practice Study of Epilepsy, that is one of the largest prospective cohort of newly diagnosed epilepsy of its time, and is a follow-up of more than 25 years and help define the prognosis and the outcome of newly onset epilepsy. So this, as you can see, is shifting from a specialistic hospital to a population perspective. In 1988, here started there was the introduction and also the early evaluation of epilepsy treatment gap, that is what is reframe a bit the access to care as global challenge. So basically say that epilepsy basically is comparing how many people have epilepsy, how many people are treated with epilepsy, and showing that there was a global challenge, defined as global challenge. And so this was the first paper showing with Shorvon and Farmer, a review about this concept, and this concept was picked up by the World Health Organization, the International League Against Epilepsy, and the International Board of Epilepsy in a campaign out of the shadow that is the most important global public health initiative in epilepsy so far. At the end of this period, 1993, also a series of publication, and that, as you can see here, is involving other countries like Kenya and Ecuador, shift the idea of seizure, biggest seizure. So if you have seizure, you continue to have seizures, and change basically the fatalism of having epilepsy to a treatable disease, that you can treat epilepsy with anti-epileptic drugs. And this was a change, probably a global change in thinking of this disease that started here in the institute. So this area was broader the department impact from a specialty care hospital to a population and global center for public health, mortality, remission, access, and equity. And also this area reinforced the three-part model that is Queen Square, the hospital, the institute, and the Child Fund, also because Simon Shorvon was also appointed director of a Child Fund at the time. And this is still present at the moment. Another part, this is more late 90s, beginning of 2000, another part of the history of the department is around imaging, precision diagnostic, and also clinical trial. And this sense, making the invisible visible is what is showing here. So in 1992, between 1992 and 1996, was created the world first MRI unit dedicated for epilepsy research in Child Fund. And this was the first publication of the measurement of hippocampal volumes from epilepsy MRI group at IoN in 1992. And this is a very nice picture of David Cobley, that is a very famous artist with a portrait also at the National Portrait Gallery. And this is called hippocampal flip, and it's an MRI machine. And the people there was the radiographer, and then Simon Shorvon, David Fish, and Leigh Sanders, and John Duncan. It's the first MRI worldwide dedicated for epilepsy. Then in 2000, we also led the first clinical trial for levetiracetam that led to the licensing of this drug in Europe. And this was a multi-center, double-blind, randomized controlled trial led by IoN and Child Fund. And this is a medicine that still now is helping millions of people with epilepsy. Imaging in this period was the core of the research in the department. And we had new tools and new imaging approaches. Was the first integration of fMRI and diffusion imaging in 2003. We also created the first and analyzed the first intracranial EEG fMRI in humans, and also the first human PET scan for NMDA receptor function. So this program of imaging in this period helped place neuroimaging at the center of diagnosis, prognosis, and mechanism understanding of epilepsy. Then in early 2000, I believe that this is when Richard Frakowiak started to create the research department and put together different people in the research department of epilepsy that was called clinical and experimental epilepsy. And there was the starting of also the discovery science in the department, and not just the clinical, but also the discovery science. And between 1999 and 2013, I just highlighted 4 of the key time points of this this beginning of the discovery science area. In 1999, it was novel voltage-gated potassium channel mutations. I put this paper here on the bottom left, not as because it was led by the director, Mike Hanna, but it's also very important to understand the next slides about therapy after, because this inspire, the starting of this research inspire some of the translational work that we are still doing in the department. Then in 2001, there was the first evidence linking human calcium mutation, calcium channel mutation to epilepsy. In 2010, also the key role of astroglia in memory formation mechanism was discovered in our department. Dmitry Rusakov discovered this. And then very important, in 2013, the department as a whole, led by Dmitry Kuhlman, was awarded a Wellcome Strategy Award to work on synaptopathies with many people also from other departments, and was also the time in which, thanks to this grant, we appointed James Rothman a Nobel laureate for working in the department on synaptopathies. So that for me is the history of the department. Obviously, it's still also part of the present, and the things that I will say in the present are part of the history. It's difficult to define exactly history and present. So if, as I say, arbitrarily I decide that more or less the last 15 years are the present of the department, and what I will explain now is a bit what we are all doing in the department. And this is really a world-leading department with, as I say here, with a translational spine. We are working on all the translational pipeline, from a really basic mechanism, basic science, discovery science, to the clinical trial and the population science as well. So, and the strength, I believe, of this department, from my point of view, is the ability to connect both the clinical cohorts, the advanced measurements that I showed on the history, but now also human tissue, mechanismic model, and therapeutic development. And they create a very nice synergy between clinical science, imaging, genetic, circuit, and system neuroscience, and therapies. And I will try to explain how. So I want to thank all the people, my predecessor as head of the department. As I said before, the department was called Department of Clinical Experimental Epilepsy. And the first head of the department was John Duncan, that was the imaging area. He's developed all the imaging part of the department. Then Dmitry Kuhlman, that as you see more or less with the time, is where the discovery science started the most in the department. And then Matthew Walker, that is the one probably that is pushed more for the translation, right, to put together the clinical and the discovery science. And then Leigh Sanders, that made sure that our excellence, individual excellence, was put together and was recognized worldwide. When I was appointed last year, I decided to change the name, it's not just cosmetic change, because I thought that the Department of Clinical Experimental Epilepsy was a bit divisive, like it was showing that we have two different things, clinical and experimental, but I thought that the link between them, chatting and speaking between clinical and preclinical scientists was the key of success for translational strategy. So I call it a research department of epilepsy. So I want to give some numbers, just on overall numbers of the department at the moment as present. This is the last 5 years, not just because the numbers are important, but it's just to show that it's not a single person, it's the all together, all the PIs in the department is making this translational endeavor possible. So we are, it's like I think 15 years that we are first worldwide for epilepsy research as a department that is most recognized worldwide. In the last 5 years, we published more than 50 peer-reviewed publications, and we published more than 40% of all epilepsy research in all UK, including more than 18,000 citation. We are 28 group leaders. I count here not just senior group leaders, but also junior PIs. We are a very large number of junior PIs, around 10, probably 12 junior PIs with independent fellowship, not tenure track, but they have their own money, and we needed to work to make sure that they have a future. We have 170 people between staff, student, professional services, and quite a lot of active grants. So what we are doing in the department at the moment, present. So, as I say, we start from discovery science, basic science. This is the work mostly of, this slide is mostly work of Kirin Volinsky, James Rothman, and Dmitry Rusakov, where we study the single synapses, and we study how synaptic transmission can be affected in epilepsy. These are very important studies because these can tell us what are the mechanisms that are wrong in epilepsy and how we can treat them. We also study astrocytes, not just neurons, and we have a lot of breakthrough on revealing how synaptic organization is structured and how the role of astrocytes, plasticity, and physiology, both in memory and circular excitability linked to epilepsy. This is the core of our discovery science in the department. Without knowing how the brain works, we cannot really find treatment for the brain. The other important research that we are doing in discovery science part is the circuit dynamics in epilepsy. And these are works that is a bit done by my lab, but also Dmitry Rusakov and Dmitry Kuhlmann, is understanding how the brain and how excitation inhibition works in the brain and what's going wrong in epilepsy. One important study that we are doing is not just see how more excitable, less inhibition there is in the brain, but trying to understand how, because the brain is a dynamic organ, how these dynamics change in epilepsy. And this is work mostly of Dmitry Kuhlmann that is on different cell types, the impact of different cell types in epilepsy, and as well as how seizures spread in the brain and how the GABA, the inhibitory transmitters, and the glutamate, the excitatory transmitter, are balanced during the seizures and during the spread of the seizures. This is fundamental to then develop therapies. Understanding the complexity of the pathology and the circuit dynamic changing in epilepsy is what brought us to think about how we can treat the brain and develop new therapies. The other aspect in the department on more basic science is genetics and actually the genomic architecture of epilepsy. So the work mostly driven by Sanjay Sisodi and colleagues is now going behind the single epilepsy causing gene, and it's not just the single gene that causing the disease. There are other things around the genes, including also the impact of, for example, climate change and other environmental impact on gene expression. Plus, we use a lot of different models, genetic models, to study epilepsy, and also not genetic models. We study, as many other departments, rodents, human cells, iPS-derived neurons. We have human tissue from surgery. We do post-mortem as well. But uniquely in the department, we study also we model diseases with the Drosophila. And this is obviously is not just for our department. And James Jepson, that is the Fly Lab in the department, is collaborating with across all departments for this incredible genetic model to study neurological diseases. And then this is where we are going a bit more on the translational, still preclinical science, and is about advanced therapies. We actually pioneer all these gene therapies worldwide for epilepsy. So the Kv1.1, the potassium channel, is the one that I showed before that Mike and Dmitry found. And actually for serendipity, Dmitry studying this mutation for Mike, or with Mike, found out that in a wild-type context, upregulating these channels control for studying actually ataxia, not even epilepsy, he saw that upregulating this channel was decreasing the excitability of the neurons. Not silencing them, decreasing a bit the excitability. And basically in 2012, Rob Wykes and Dmitry and Stephanie Schorge, Matthew Walker, described the first gene therapy for epilepsy upregulating Kv1.1. The idea is you have a potassium channel that decreases excitability, you express that in excitatory neurons, decrease the excitability, and you rescue the seizures. This was the first time using this channel for gene therapy. In the same paper also they use optogenetics, so light and light on neurons to modulate their activity, and show that it was possible to treat epilepsy and decrease epileptic seizures. It was the first time that optogenetic was used for epilepsy. As well, in 2014, there was the first paper showing that chemogenetics, so an engineered channels that binds only to synthetic drugs, was able to decrease seizures as well. And was the first time that it was used for epilepsy. And then gene editing in my lab was actually just after CRISPR was born. We decided to test this discovery. We decided to test this approach, gene editing for epilepsy. And actually, we published the first 2 papers, evidence that you can use CRISPR-A in this case to upregulate the gene expression for epileptic encephalopathy and for acquired epilepsy, showing that we were able to modify the endogenous genes to stop seizures. And finally, the most innovative and new ways of doing therapy, that is something that also Pietro Fratta mentions today, is this closed-loop therapy, like therapies that can sense the pathology and treat the neurons only when it's needed, where it's needed. So Dimitri in 2018 and me and Dimitri in 2022 discovered 2 ways of doing this closed-loop therapy for epilepsy. One at the level of synapses, in which is a biochemistry closed loop, in which when there is too much glutamate for seizures, there is a closed loop that decreases the stability of the neurons. In the other paper, we show that we can use promoters that sense activity. And when sense activity, we express the potassium channel only in those neurons and decrease the seizures, only targeting the real important neurons to treat. I'm telling all of this because all of this invention together brought to the establishment of Epilepsy GTX that Dimitri just mentioned. So Dimitri, Matthew Walker, Stephanie George, and myself founded this gene therapy company in 2021 that we just got Series A to start the first clinical trial in 2027. And 3 patents from the one that I showed before are now part of this company. And actually the leading program is the KV1.1 channel that starts from the study of MyCan, etc., etc. That one is the leading program. We, as a department, we have more than 9 or 10 patents of original gene therapies for epilepsy. So we put 3 together in a new company, but we have other 6 or 7 that are potential for other companies. So this was all the preclinicals, less translational. Obviously, we need also to record the activity and understand where the seizures start. So we do a lot of neurophysiology, both in rodents but also in human. For example, use a new graphene-based array to just not just record seizure, but also spreading depolarization. That is a big phenomenon after seizure that is quite important for the pathology. We also investigate neurophysiological investigation in patients. I don't have to explain the OPMEG because I think Martin explained quite well. And we are also working on measurement, for example, of cognitive measurements with these tools. These works have been done by different people, the graphene-based array, the Rob Wikes, and all this part of neurophysiological investigation is Matthew Walker, Umesh Vivekananda, that is here in the image with the helmet, and Neil Berges as well. But we need neurophysiology also to try to prevent and help people for prognosis. So we use stereo-EEG, also DC recordings, infra-slow waves, as you can see here, to see post-spreading depolarization linked to the graphene array before. We also mapping network with direct brain stimulation, and we study seizure severity and pseudo risk. This is Beate Dill and colleagues. Then we also work in something a bit tangential, but it's quite important for epilepsy, that is functional dissociative seizures. So are seizures not linked to epilepsy? They are similar to seizures behaviorally, but don't have the electrical discharge as seizures. And we were the first group and first department to describe heartbeat evoked response in functional seizures. almost the end of the present, and as I say, the imaging was very important. There was the imaging area driven by John Duncan and colleagues like Meneka Sidhu and also Davide Giampiccola, still working in image-guided surgery. And all of these slides is to show that imaging is very important to define surgery, surgical outcome, and also help neurosurgeon to do surgery, but it's also redefined a bit, and I think it is a concept that goes through all the department from discovery to clinical, is the concept that the epilepsy is a phenomenon of a brain network phenomenon. It's not just a lesions, but it's a change in all the network in the brain, and this image-guided surgery is helping that. And then also we do non-invasive prognosis with the Lemieux still going on the EEG fMRI that I presented before. We don't do only things related strictly with epilepsy, but also around and comorbidities. We're doing quite a lot of epilepsy neurodegeneration with Matthias Kopp and Maria Toms, and this is a big team that is also now is creating like a consortium between the Dementia Research Institute, the Epilepsy Research Institute, to try to understand really the link between epilepsy and dementia. We're doing clinical neuropsychology, so Sally Baxendale, and also the work on clinical trials, for example, drug retention, and we are studying for 3 decades of anti-seizure medication retention study at Queen Square of Leigh Sanders that is doing a lot of these clinical studies of anti-seizure medication, but also population and global health. So this is the model that we have at the present, is one continuum. It's from patient cohort, deep phenotyping, to measurement, video EEG, as I say, many different ways of measurements. We study the mechanism from a single gene, synapses, glia circuit. We use several different models, organoids, Drosophila, human tissue, and we develop different therapies, not just gene therapy, but also neuromodulation. And obviously, we create also a spin-out company. So this is why the department can lead internationally in epilepsy research. And we have everything from patient phenotype to mechanism experiment and therapeutic testing. And this is a new book edited by Leigh and John Duncan, Leigh Sanders, John Duncan, about epilepsy at Queen Square, just published now. The future, in the next 2 minutes, I will say, you saw it, I knew that I was going late. 20 minutes was not enough, probably. So the next 75 years, and I believe that is our aim, as I said before, is not only to understand epilepsy that we have done so far, but also to change its natural history. So what we want to do is create a translational pipeline that we are trying to do, from discovery, human models, therapeutic design, delivery. We're doing a lot of different ways of delivering gene therapy at the moment, also clinical testing. And the 3 main topics will be advanced therapeutic pipeline, therapeutic repurposing, and also clinical trial readiness. We want us to create a precision platform in the department. We are improving all the different aspects in the individual brain map, like 7 Tesla, as Martina was saying before, neuropixel, stereo gene in patients, predictive biomarker, precision intervention. We are working quite a lot on genomics, climate and global equity as well, and we are really fostering and trying to foster the next generation of leaders, as I say. So the future, the department becomes a platform, so measuring better, predicting earlier, intervening more precisely. Finally, the strategy, the 3 main strategy in the department will be to rebuild the clinical trial capacity linked with the Chalfont. We have more than 5 clinical trials at the moment in the department, and we're expanding it. We are working with UCLH to expand the academic neurosurgery program, and we really want to build a discovery to translational pipeline even better than it is now. So this is the final thing. So I think that the next challenge is to unite everything that we created so far into a faster path from discovery to disease-modifying treatments. I want to thank everyone, all the PhD students, postdocs, professional services, everyone is working in the department, all the leadership board, and everyone that helped to put together this presentation. Thank you very much.
Thank you so much, Gabriele. I think we've got a short film before I introduce David.
I woke up the normal time, but didn't feel right. I knew there was something wrong, but I wasn't sure what it was. I thought I'd laid on my arm after I'd sort of tried to move it, and I think I realised at that point that I was having a stroke.
Stroke's the second biggest cause of death globally. About 11 million strokes every year. It's also a leading cause of disability in adults. Stroke is one of the most important medical emergencies we face.
I couldn't speak and my face had dropped. My granddaughter, she knew instantly that I was having a stroke and she phoned the ambulance straight away. The ambulance came and within no time whipped me off to Queen Square.
Queen Square is an important place for stroke research and care. Co-location between the hospital and the institute is critical, and what makes the Queen Square story so significant is that research done here, because of the unique combination of scientific expertise together with clinicians, help lay the foundations for the treatments we now use every day. The major Queen Square breakthrough is the discovery of the ischemic penumbra, which found this idea that when an artery blocks, which is the cause of an ischemic stroke, there is a bit of the brain that is irreversibly damaged, but there's also an area of the brain which can potentially be salvaged, and we call this the penumbra. If this area can be reperfused, if we can open up the artery and restore the flow of blood quickly enough, then that will allow a full recovery of function to that area of the brain.
The next thing I remember, I was sitting up in bed like this, and I said, I'm fine, you know. It was a miracle. It was an absolute miracle from being completely, couldn't do anything, couldn't speak, to being back to normal within hours.
Up until the discovery of the penumbra, stroke was considered to be a sudden, irreversible injury to the brain. So we've gone from a disease where there was really no treatment to be offered to where we are delivering treatments to thousands of patients. Thrombolysis, for example, which is a clot-busting drug given intravenously, improves the chances of a functional recovery from about 1 in 3 to 1 in 2. And thrombectomy, which is to actually remove the clot from a large blocked artery, it increases the proportion of people who achieve an excellent functional outcome from about 1 in 5 to about 1 in 2. So this is one of the biggest treatment effects in the whole of medicine. It's really important that the hospital and the institute are close to one another, because the treatments that we're evaluating need to be offered within hours of the onset of a stroke. Bringing these treatments into practice was a huge team effort. We developed these hyperacute stroke units that allowed us to assess and diagnose people really quickly in order to deliver these time-sensitive critical interventions.
Research from Queen Square had global international recognition and reputation. When I started doing my fellowship at Queen Square, I joined forces with the colleagues here, and through that, that's when we started setting up the first stroke unit in Malawi. The international leadership of Queen Square really helped us with the blueprint, the clinical, the research, academic side of things, the training. It has been really foundational in helping us drive what we have been able to achieve. And now we have a service that's running for the last 3 years, adopted by the Ministry of Health. We've built local expertise, and it has significant impact on outcome already.
The transformation in stroke care has been absolutely astonishing, and the work is not finished. The next frontier is to make the same kind of progress for brain hemorrhage, the most devastating form of stroke. Our aspiration is that every person who suffers a stroke will have the opportunity to take part in stroke research.
I am eternally grateful to the team. I know what the consequences would have been if I hadn't got there in time and got to this particular hospital.
So, yeah, it's a great pleasure. You saw him on the video there. Great pleasure to introduce Professor Dave Waring, who's the head of the Department of Translational Neuroscience and Stroke. Dave and I were SHOs together here, so we share tenacity, I think, and a bit of a sense of taste in music as well. Anyway, I'll let you get on, David. Thank you.
Thanks, Alex. Yes, well, we're still alive anyway, so that's good. I'm really pleased that stroke has been selected as one of the diseases that we're going to talk about today. I've been really inspired by the talks that we've had during the day, and I'm aware this is the last disease area that we're going to be looking at. I must admit, I feel a certain amount of pressure. I know it's not a competition, but it is pretty inspiring what we've heard so far. So what I'm going to do is, first of all, remind us why stroke is such an important disease. I'm going to go through very, very briefly a little bit of history that Mike touched upon earlier on, actually, that stroke is part of the origin story of Queen Square. And then I've just selected a few breakthroughs in various aspects of the stroke care pathway. So acute stroke, I'm going to talk a bit more about the ischemic penumbra and its impact. Then I'm going to talk about prevention of stroke. It's one of the key things that we need to do in the clinical service. Then I'm going to talk a bit about brain small vessel disease and brain hemorrhage that we heard a little bit about on the video. And then finally, but very importantly, stroke rehabilitation and life after stroke, which is a particular strength here at Queen Square. And then I'll just touch on some of the future plans. So the importance of stroke. I don't need to tell you all how common stroke is. It's the commonest neurological disorder. It's the second biggest cause of death globally. It's the third largest cause of death and disability, and it's the largest cause of complex adult disability. There are about 12 million strokes every year. And in fact, in the UK alone, there's 100,000 strokes every year. So that means during the course of this symposium, there will have been about 100 strokes in the UK. So it's a very common disease. These statistics don't really do justice to the personal impact of stroke, though. And I've just put up Robert McCrum here, who was the Observer literary editor. Many of you will have read his work. And he wrote this book, My Year Off, which describes the profound impact of stroke on his life and his re-evaluation of his life after that. It's very eloquent. It's a wonderful book. He had his rehabilitation at Queen Square, actually, which is some interesting descriptions of that as well from a previous era. So I commend that to look at the personal impact of stroke. And incidentally, his great-grandfather invented the penalty kick, apparently, in football, which is a random fact, but slightly appropriate at this time of year. So just to go back to basics then, about 4 out of 5 strokes are due to ischemia. They're due to a blocked blood vessel. And then the other 1 in 5 are due to hemorrhage into the brain substance, much rarer hemorrhage around the brain substance. And this is usually due to a rupture of a small blood vessel. And the thing I want to get across here is that it's a clinical syndrome. So there are very many causes. Stroke in itself is not a diagnosis. It's an acute syndrome due to ischemia or hemorrhage, but there are many causes. So the historical perspective, as we heard from Mike earlier on, the Chandler sisters, and not forgetting their brother Edward, were instrumental in setting up the National Hospital, because they were cared for by an elderly carer who had a stroke, unfortunately, Mrs. Pinnock, and they realised there were no hospitals in London to care for somebody with a hemiparesis or with the sort of deficits that we see after stroke. And as we also heard, David Wire, the Mayor of London, joined their campaign, their fundraising campaign, to build a hospital for people with paralysis and epilepsy. And by a quirk of fate, he actually had a minor stroke during the fundraising campaign, which really gave it a huge lift. And I think we all know the value of philanthropy and the importance of that. But that probably indirectly led to the foundation of the National Hospital and subsequently the Institute of Neurology. I didn't expect that to get a laugh, actually. But so it's quite interesting going back to the early case records of the National Hospital, and this is Hugh Links-Jackson, one of the fathers of neurology, and this is one of his own case books where he describes, interestingly, transient attacks and then leading on to a cerebral hemorrhage, which of course could only be diagnosed after death in those days. But stroke was really, the case books were full of strokes. But then, for one reason or another, I'm not quite clear why, but stroke really wasn't practiced at Queen Square for probably another 100 years, actually, until there was really when Martin Brown got the Foundation Professor of Stroke in 2000, and as we heard, Richard Verkowiak established the Functional Imaging Lab, which was interested in stroke, and then Nick Ward and others. Really, stroke wasn't on the radar very much in terms of clinical applied research at Queen Square until much later. So now I'm going to talk about a few breakthroughs in stroke. That was what I was asked to do. The remit was breakthroughs in stroke at Queen Square. And this will necessarily be very selective, and I do apologise in advance for missing out lots of important work. Although I should say we're a much smaller department than a lot of the other departments that have spoken so far today. But what I'm going to talk about then is acute stroke, and this is just to bring this to life. This is somebody who comes to the hospital with a blocked left middle cerebral artery. You can see the red arrow there on the CT angiogram, and many of you will be aware of the face-arm speech test. So this person comes in with a hemiparesis. They can't move their right side. They're unable to speak. This is now recognised to be a real medical emergency. But it wasn't a medical emergency really before the foundational work done by Lindsay Simons, Jan Astrup and others on the ischemic penumbra. This is the Neurosurgical Experimental Laboratory at the Institute, taken during the mid-70s. And you can see Lindsay Simons on the right. These were experimental studies in animals, and they were looking at thresholds of brain ischemia. Simon was actually a neurosurgeon. And what he discovered was that there were thresholds of ischemia whereby tissue can undergo a reduced blood flow leading to neuronal dysfunction, but without death of the tissue, opening up the possibility of treatment if the ischemia can be reversed. So this is just a simple example here, a diagram showing the blocked artery here. The blood flow, if it's reduced to below about 10 mils per 100 grams per minute, that tissue will infarct. It can't be revived. But there is this penumbral zone, which I think he named after the sort of half light around an eclipse, or the light around a candle, I think, over dinner was one of the rumours. But the penumbra is this tissue where it's not working, so there's a neurological deficit. But if the blood flow can be restored, that tissue is potentially salvageable. And we now know that the duration of the penumbra in humans is typically pretty short, only a few hours, although in rarer cases it can extend a bit longer. So every minute of a stroke, you're losing 2 million neurons and 14 billion synapses and about 12 kilometres or 7.5 miles of myelinated fibres. Which is, this is why this is now the clinical mantra, time is brain. And I think it's fair to say that the ischemic penumbra concept, which was here at Queen Square under Lindsay Simons' laboratory, has led directly to the treatments that we now use to restore blood flow. So we've got thrombolysis and thrombectomy. And if we go back to this case, we can now image the penumbra in life, and this is part of our regular acute stroke service. And here we can see CT perfusion. The red zone is the ischemic core that I was talking about, and the green zone is the potentially salvageable tissue. This person underwent a mechanical thrombectomy, which is a non-invasive, minimally invasive neuroradiological procedure to remove the clot. And we can see just a tiny little infarct here after the mechanical thrombectomy. And I think I said in the video, we've got this massive increase in people reaching functional independence from about 1 in 5 to about 1 in 2 to achieve. This is from one of the many meta-analyses. And I should say we did one of the first, the first trial of thrombectomy in the UK was co-led from University College Hospital, and we included most of the patients into that study that confirmed that it could be delivered in the NHS. So just very briefly on acute stroke, I just want to mention some important observations that were made during the COVID pandemic by Richard Perry and others that you can see listed here. So we were able, we've got such a busy service now, we're seeing around 2,000 people with suspected stroke a year. We very rapidly realised that something was going on with the COVID virus, and we were seeing these large vessel occlusions that were surprisingly refractory to our usual treatments. And Richard Perry very rapidly assembled a UK multi-centre study that provided the very first description of venous sinus thrombosis in association with vaccination, which has been important in obviously moving on to design safer vaccines after the early phases of the pandemic. Okay, so that was acute stroke, and now I'm going to turn to stroke prevention and just go through a couple of relevant aspects. So first of all then, about 15 to 20% of strokes are caused by large artery disease. And this is a magnetic resonance angiogram showing the arteries in the neck going up to the brain here. And at the front of the neck, we've got the carotid arteries, which supply the brain hemispheres. So this is a substantial proportion of ischemic strokes that are due to carotid stenosis. And many of you will know Professor Martin Brown, who was appointed as a chair of stroke here, I think 1999 or 2000. He established the stroke unit here at Queen Square. And he's probably best known for his work on carotid stenosis, doing large international trials. There are two ways of dealing with carotid stenosis. One is putting a stent in, and the other one is removing the atherosclerotic plaque using carotid endarterectomy. And we didn't know how these two techniques would compare until a very large study that Martin led showed that there was an excess of largely minor strokes in the people undergoing stenting, although the long-term outcomes turned out to be very similar in the follow-up of this international carotid stenosis trial, surgery trial. So this was hugely influential on national, international guidelines and still guides our clinical practice. More recently, just last year, Martin published the data on the European carotid surgery trial comparing medical treatment for atherosclerosis in people of lower risk, showing that it was largely equivalent to surgery in the ECST2 trial. And Rolf Jaeger and others have looked at the brain imaging of patients undergoing carotid revascularization and have shown the importance of brain imaging changes such as white matter hyperintensities, looking at heterogeneity of treatment effects. You can see all the Lancet papers. I haven't had a chance to put them all up there, but a lot of papers which have directly transformed the field and still inform our clinical practice on a daily basis. So the other type of stroke that I want to mention is cardiac embolism from atrial fibrillation. So this is about 1 in 4 strokes. And we've got really good drugs for this. We've got anticoagulants. We've now got the direct oral anticoagulants, which reduce the risk of stroke by about 3/4. So it's one of the best interventions we have for stroke prevention. Massively used drugs, really effective. But one of the problems we've got on the stroke unit is somebody comes in with a stroke like this. This is a very large one. And we've got a massive risk of recurrence early on, probably about 1% per day, something like that. And we want to treat people with anticoagulants to stop those early stroke recurrences. But there's a really scary potential effect of anticoagulants is to cause bleeding into the infarct. So this was a position of clinical uncertainty, and we used to wait for about 2 weeks to do this anticoagulation. But we've led some trials now that have definitively shown that you can start the anticoagulants early after ischemic stroke. So one trial was led from UCL, and then we've combined with other international trialists to show that there is a reduction in early stroke recurrence, about the same sort of effect as giving aspirin for acute stroke, without an increase in intracranial bleeding. So it's safe and it stops early strokes. And this is beginning now to transform practice and guidelines quite quickly. So a few words on small vessel disease, and then I'm going to go on to life after stroke. So small vessel disease, a group of diseases, the most common brain disease, causes around 25% of ischemic strokes, most hemorrhages, contributes to about half of dementias, and it's by far the most prevalent brain condition that we know of. Of the consequences of small vessel disease, brain hemorrhage is probably the most consequential in terms of disability and mortality. So there are about 3.3 million deaths from brain hemorrhage every year, which I think is more than all other neurological conditions put together, something like that. I mean, it's a very lethal disease with a 40% mortality at 3 months. So it's a big challenge. And one of the things that we've worked on at Queen Square over the years is looking at neuroimaging biomarkers to try and diagnose the arteriopathy that causes hemorrhage in life to allow us to assess the prognosis and ultimately to tailor treatments. And I will just briefly say that one of the things that was one of the breakthroughs from Queen Square is that we've developed these non-hemorrhagic markers of a condition called amyloid angiopathy, which causes a proportion of brain hemorrhages, and it's got the highest recurrence rate of any stroke subtype. And there are no treatments for this condition at the moment. So it is important to identify patients with the condition for prognosis, but also for clinical trials. And then the final thing I just want to mention on breakthroughs relevant to small vessel disease is around about 2000 when Rolf Jaeger actually implemented this new technique which showed these black dots on scans called microbleeds. And we were terrified of putting people on anticoagulants or antiplatelet drugs because of these microbleeds. And they discouraged the use of these drugs, which are hugely effective, obviously. And this is a consequence of anticoagulant-related hemorrhage, which is even more lethal than brain hemorrhage not associated with anticoagulants. And I'll just summarise this work very quickly. I thought this would be an easy thing to do. I thought it would take a few years. It ended up taking 10 to 15 years to actually get some answers to this. But the answer is microbleeds essentially, and perhaps paradoxically, predicted ischemic stroke. And this was always a lot more common than hemorrhage. So we got more ischemic strokes than hemorrhages. And we now basically don't withhold treatment because of microbleeds. So this was very reassuring clinically and a helpful result. So now I'd like to talk about stroke rehabilitation. This is really one of the key strengths in Queen Square. And of course, most of the burden of stroke is in people who have survived an acute stroke and may have had hyperacute treatments. They may have benefited from those treatments. But there remains a huge burden of complex disability, including cognitive, language, and motor effects. And I'm just going to highlight two groups that have worked on this. Firstly, Alex Leff, who we've heard from earlier, and he has really, his work has shown the importance of dose with respect to treatment of language disorders after stroke. And these disorders are among the most consequential disabilities in stroke survivors. And Alex has recognised the importance of dose, and he's implemented that in hospital-based therapy delivery with this comprehensive aphasia service at Queen Square. And this is generously funded by the National Brain Appeal. And the evidence from observational data very clearly seems to show that this is a hugely effective intervention, both in terms of speech production and speech perception. I haven't got time to go through all the neuroscientific contributions that Alex has made into understanding language recovery in this context, but this is now delivering a scientifically based, neuroscience-based rational therapy for aphasia, recognising the huge importance of dose, which is, of course, not delivered sufficiently in the NHS. And then the other thing Alex has pioneered is innovative technology-based solutions, including gamification apps to allow people to practice word generation in order to improve their speech and communication. And you can see this is the iTalk Better app, and it's delivered. You can see the sort of gamification, and you can see the clear benefit of this app on language performance. And he's been incredibly prolific, actually, at delivering these technology-led solutions for rehabilitation after stroke. And these allow things to be developed, to be delivered at huge scale, including treatments for hemianopia as well as language. And he's also worked on dementia. I mean, he's been incredibly productive in terms of delivering these interventions and putting patients in control of their own recovery, which is so important, the sense of agency in people who've had a stroke. And then, secondly, I want to mention the work of Nick Ward, and of course with Richard Frakowiak initially, looking at the neural basis of recovery after ischemic stroke, and making the most of these emerging functional brain imaging techniques to show that there is a massive reorganization of the brain after a static insult from a stroke. Here we've got one in the brainstem, one in the subcortical region here. And this shows us that there is adaptation in surviving pathways, which again opens up the possibility of neuroscience-based neurorehabilitation to make the most of that adaptive process and support motor recovery. And Nick has recognized the importance of the upper limb as one of the most important factors influencing daily life. It's one of the things that a huge proportion of stroke survivors will have difficulty with. And again, this concept of the massive importance of dose, which has only recently been recognized. And what Nick did was implemented this high-intensity stroke rehabilitation, probably the first in the world for chronic stroke patients. So this is beyond that acute phase, often months or years after the stroke. And he's shown a clear benefit in upper limb function with intensive neurorehabilitation for the upper limb. And this has been critical not only for informing a randomized trial to definitively prove that this works, but also to show us that stroke patients can recover beyond this kind of 6 months. I don't know where it came from, but they're always told you've got to get better for 6 months and then nothing's going to happen. So it's really good that that Nick and Alex indeed have shown that neuroplasticity is not time limited, although obviously it is rather faster in the early stages. And the other innovation that I want to mention is the neurorehabilitation online. This was another super rapid response to the COVID pandemic. And this was an online group-based neurorehabilitation system, which was deployed incredibly rapidly during the pandemic. And it's now actually been rolled out to NHS centres throughout the country, or certainly in some areas of the country. And it seems to be an incredibly powerful way to deliver, again, at scale, neurorehabilitation for survivors of stroke. And then just finally, I want to mention our stroke registry. So it's taken a while, but thanks to BRC funding, we've established a registry which is now around 4,000 patients with detailed imaging and 6-month follow-up, and we're now undertaking the 10-year follow-up. And what this registry has shown us already is the huge burden of non-motor outcomes that are the hidden face of stroke. So we know about motor deficits, we know about language deficits, which are usually reasonably apparent to outsiders, but then there's this hidden burden of things like sleep disturbance, anxiety, sexual dysfunction, integration into society, these things that are completely hidden. And this is one of the first outputs of the Signal Stroke Registry, and we've also got embedded within that a young stroke registry. We're just beginning the 10-year follow-up now to see the long-term impact of stroke in this well-phenotyped registry. And then just finally, this is Parashkev, who leads the high-dimensional neurology group. And this is one of his foundational papers, which tells us that if we do traditional lesion symptom mapping, if we don't take into account underlying patterns within the data, so in this case the arterial tree, we're going to mislocalize. This is a map of the mislocalization of brain function. And I think this has now set Parashkev on this track of massively scaled, high-dimensional generative modeling to understand the organization of the brain and its disruption by lesions. But I don't understand a word of it, to be honest. And this is another paper that is quite recent, really pointing out that this high dimensionality is fundamental to the heterogeneity of treatment responses in all of these domains after a stroke. So who knows, one day we may not need to do randomized controlled trials that take 10 years to get the answers. We might be able to derive from observational data and counterfactual modelling whether treatments are going to work in particular patients. And just super briefly, I'll just outline a few things that are on the horizon for us, in the short-term horizon. The next frontier, I think, is preventing brain hemorrhage, that hugely disabling condition. And we're getting some really exciting data now in this condition of cerebral amyloid angiopathy that has the highest recurrence risk of any stroke, very disabling. The hemorrhages cluster in time and space, and we're getting some evidence this may be due to inflammation. We've got evidence of inflammation sometimes preceding hemorrhage, and we're hoping to work towards trials of the first preventive strategies in this condition. And then this is just a handful of some of the future projects. We're going to be doing randomized trials to see if we can safely treat people taking anticoagulants at the time of their stroke. This excludes currently a large proportion of people from thrombolysis. We're going to be working, as I said, on stroke and dementia prevention in CAA, randomized trials and rehabilitation, including one that Alex is leading on language recovery, and Nick will be continuing his upper limb work. We're going to be looking at the 10-year hidden impacts of stroke, high-dimensional modelling, and we're going to be hopefully finding some new treatments for small vessel disease, including small vessel occlusion. So these are too small to do a thrombectomy, but we've got some ideas for drugs that may be able to open up the arteries, such as vasodilation, endothelium blockers, and so on. So I should have said right at the beginning, this is hugely selective. There are many, many funders and people that have supported this work for whom I'm extremely grateful, and I thank you for your attention.
So, good afternoon, everybody, both in presence and on other side. So, my name is Pietro and I'm Deputy Director Strategy and Dual Hub of the Institute. Today, I'm not here to talk about a research core facility or the new institute, the Dual Hub, or other initiative. And I think that I can see a sign of relief in some of your eyes. But I'm here to talk about a very important matter. So every discovery, every new therapeutic indication or advance, or any of the commercial enterprises that we heard today are the result of many factors. The great engagement with and partnership with our hospital, the leadership of basic science and clinical research, and of course the support, the enduring support of our patrons and funders. However, the discovery and all this achievement would not be possible without the incredible support of an exceptional number of early career scientists. And these career scientists devoted their best experimental year, both in clinic and in basic science, to the Institute and their success. And as shown by several of the speakers that occupied this podium before, this their brilliance allowed their recruitment that further enhance the delivery of exceptional research in the recent history and before of the Institute. So, celebrating their work and the commitment and valuing their dreams and aspirations is one of the main goals of this short session this afternoon. And for this reason, I would like to ask you to join me, both in presence and in the other side, in a round of applause for all the ECR that operate at the Queen Square. Thank you. So, celebrating the ECR is important, but as important is listening to their preoccupation and understanding their needs. This is the reason why the presentation of Alia, that is a PhD student, one of the many excellent PhD students at the Institute, I'm delighted to open a panel discussion with 6 early career scientists at different stages of their career. Their faces and names are present in this slide, but I will call them. So, Alia Masood Abdel Hafid is a PhD student in neuromuscular and is a basic researcher. Francisco Moreira is a research assistant, a PhD student across the department, but affiliated with epilepsy. Jasmine Donaldson is a postdoctoral fellow in D&D. Francesca Magrinelli is a senior clinical research fellow, clinical in movement neuroscience, and Felipe Nascimento is a senior research fellow in DMD. And I would like to call on the podium also Professor Rob Brownstone, who is a professor and clinic and neurosurgeon in DMD. And there is a big prize if you can spot the oddball in this.
I've been called worse.
So this is a question, is a panel that is open to the audience, to all the CR and also the senior, to ask questions so that we can start a discussion to bring forward in the Institute. I have prepared a couple of questions just as a warm-up for those of you to think about your own question. But the first question that I wanted to ask the panel is that we are living in unprecedented time of change. And many of you have experienced the feeling that the ground is shifting under our feet. And under these circumstances, it is absolutely essential to provide the best support and training for the next generation. And the question for the panel to this group of early career scientists is which kind of support is urgently needed? I don't want to avoid a long list. I ask you to pick one of the most urgent need that you think that is necessary. And this can be enterprise, comms, or any other scientific training or clinical.
So to get us started, I think as early career researchers, one of the most important traits that we have to develop is our ability to communicate. One thing that always isolates a lot of scientists is that we're not always able to speak the same language as the people listening to us, whether that be in enterprise, whether that be people living with the diseases we study. It's being able to understand how to make everything as simple, and that oftentimes doesn't get incorporated in our research as much as we think it should be. And that's a skill that I think all of us should start focusing on a little bit more of.
Okay, good start. I'm gonna say maybe it's something a little bit more specific to science, and I think sometimes it will be good to have to learn skills of what's after research or going into industry and pharmaceuticals, and what do they will need from us back there. And I think we should be incorporating our designs and experiments, thinking not just sometimes we have good ideas and but what's going to be needed to take it afterwards.
So Francisco, as a feedback to your answer, so is UCL providing enough or not courses or area of forums to discuss this point?
Good question. I think in the past 2-3 years, Dimitri Kuhlmann, as a translation deputy director, started with Eleonora Lugara and now Susie Mayers, creating these interactions with venture capitals. They do a lot of seminars on how venture capital works, what pharmaceuticals want, what's the discussions happening, and things like that. Other symposiums like advanced therapies and things like that. I learned most of my things through those things, and those initiatives were definitely very helpful.
So what will be necessary to make those action more effective, in your view?
I think we're definitely going in the right direction. There's a lot more happening. It's just now, like the institute. I think the institute is taking a very translational, strategic move, and I think as early career researchers need to be incentivized to come to these things and attend them more often. Yeah.
Thank you.
I sort of agree with both of those points. I think collaboration is really the way to move our science forward. So between diseases as well. So we have an amazing HD center where we meet fairly frequently. But we also have, say, the Department of Neurodegenerative Diseases weekly meetings. And I think attending those and kind of having an incentive to attend those in terms of collaboration and seeing friendly faces week on week, then sort of build your confidence to approach other people within the department to then kind of share skills. And I think that's going to be really, really critical in kind of facing these challenges of kind of bench to clinic and vice versa, where we're meeting more clinicians and we're kind of hearing the struggles in clinic and then how we can sort of address those in the laboratory, I suppose. So I think really just promoting collaboration, which I really do think IoN does well. But I actually do think, myself included, we're not all amazing at attending these things weekly, which I think potentially is the fall of us in a way.
Thank you.
I'll try to bring the perspective of a senior clinical research fellow. I think the biggest challenge for us is the transition to independence. So that's the most vulnerable time of a clinician scientist academic career. And although I believe early independent fellowships are invaluable, the opportunities are getting smaller and smaller. And so I think we should rethink and create new opportunities and pathways towards early independence. And in this regard, thinking about possible solutions, in my recent experience, it has been instrumental to participate in meetings with my senior colleagues, with my PIs, with my mentors, when drafting grant applications, sometimes participating in the nighttime marathon the night before a deadline. I think it has really changed my way to think about how to make any results from my clinical research, clinician scientist research, a valuable question for the future.
Thank you. Felipe.
I really don't have that much to add. I'll just echo some bits that were said. I think nowadays the modern scientist needs to be quite versatile and have skills that go just beyond science. And this can involve something related with, being proficient at governance, admin, IP, etc. So support on those areas, I think, would be really important. And to do proper science, we need an ecosystem in which, we're surrounded and supported in all these areas. And we obviously learn by trial and error. So this is a way to mitigate and obviously save some time by learning other skills that down the line will support the output of our research.
So I think that is from a moderator perspective is quite interesting that one of the requests is in addition to versatility, in addition to opening to other career stages, is also sharing the pain of the last deadline, and you don't think that sharing the pain is putting you off to a career in science as you wish to entertain?
Well, not as much as finding yourself in front of work tribe to cost a research grant for the first time. So I really enjoyed.
We are talking about a different level of pain here.
Yes.
So I open the session to the audience. If you can speak loudly so I can repeat the question. Dimitri.
It's likely that science is going to be totally transformed by large language models. Anthropic AI released Claude Science very recently. Do you think UCL should be providing you with access to powerful large language models and training in how to use them?
Absolutely.
You might need to repeat the question.
Yeah, so the question is about, thank you, Dimitri. The question is about the need from UCL to provide AI solution, in particular software that provide a new and large language model to all ECR and all the UCL personnel.
In the past year, there's been an explosion of access to these models, and the distinction is going to be who uses them.
Can you talk closer to the microphone?
I think the big distinction is going to be who uses them correctly and right, or like taking the full potential of these models. It's very easy to ask ChatGPT like Google like questions, but the power of these models goes far beyond this. And I guess as scientists we can teach ourselves a lot of skills, but it will be great to have like more formal guidance too. Like especially like I've heard I was at a seminar the other day and they were saying talking about like confidential pieces of your research could actually be a problem with these models, and these things, you know, like it will be great to have good guidelines, good skills training about for this.
We really shouldn't strain away from the world of AI because it's coming towards us, and it is being incorporated in every other institution. So as part of UCL and as a pioneer in the space, it's something that we should be really excited about and encouraging our scientists to learn how to use effectively, especially since it can increase our workflow efficiency by 100%, especially if we know how to use it correctly, because now high school students are the ones learning how to write the right prompt, but we're the ones who don't know how to prompt ChatGPT or Claude correctly and get left over with answers that are just not the answer you're looking for. So it's a whole new way of thinking that we should embrace.
Yeah, I think that with the new tools comes also new responsibility. I think that at the level of the institute, we should really think about SOPs on how to use AI, the language model, SOPs that are not only our initiative of single labs or department, but that goes at the level of the institute and across the site. Sarah.
So I wanted to ask, because I think this is a kind of bigger existential question that with the kind of MRC and Welcome and NIHR, and I know this because I talk to lots of juniors about applying for fellowships, but the whole path towards an academic career with career development fellowships, then senior fellowships, has changed. And there are there are very few. The path with career development fellowships and senior fellowships with MRC and Welcome has disappeared. And so it's a kind of bigger question to the ECRs in that, A, do you feel supported enough at the IoN and UCL with input on potential careers and paths, and do you feel that there is enough of an infrastructure here to help support you and navigate that? But also it's a kind of existential question for the university in that we have to support the next generation. It's really important. And so with the change in funders and funders' drive for clinician scientists or non-clinical scientists, it's how we fill and plug that gap to make sure that we keep focusing on the next generation, the people who will all be in our jobs in 15 years' time or 20 years' time. And so I was interested in how supported you feel, but also a wider UCL question about what do we do as a university to make sure that we keep people in fellowships and funded.
Okay, I give a non-AI version of the question of Sarah is, do the ECR feel supported in the shifting environment of funding for their career progression, and should UCL do something different or more in this area?
Okay, so I personally do feel very supported in terms of, I guess I'm sort of early career postdoc currently. And I think there is constantly information being fed to us, at least through our department, of opportunities. And I think we have a lot of kind of champions, particularly my boss, who is Sarah, but I know that there are a lot of... And I'm not just saying that because she's there, but I do really believe that she is a champion of kind of early career researchers, and I really believe that so many of the kind of professors or doctors and people that I come into contact with really do champion their students to achieve the best that they can. And so I feel extremely fortunate to be in the IoN for those reasons. The thing that I potentially think, the only thing that I would change is that it's very academic focused trajectory. And so I do have kind of, I've only ever been in academia, and so I am really curious about what industry looks like. And there are definitely industry kind of collaborations that are available, but I am still very curious about that aspect. And I'm wondering how that can, I mean, probably the best way is to go and try it, but I don't necessarily want to leave academia to try something that I might not like. And so it's wondering how we can try and teach us about industry more informatively, so that we can actually make a really structured decision prior to leaving academia and jeopardizing like the academic career that we have, I suppose.
Yeah, I echo you about this. So I think I am receiving support, although I think there are more resources, and for some of them, I realize I'm not entirely aware of how they can be exploited in a way. So I think one of the aspects which is improvable might be to expose earlier to the possibility of alternative funding from industry, from developing idea, because of course in our previous path we are driven by research questions, but then the question change, and this is particularly true when you have a clinical background and you realize that it's not only about making the experiment working, you want to produce something which is meaningful and can go back to your clinical practice. And so that's my main point. So I was chatting with Susie recently about this, and she kind of changes my view about some of the research we are doing and the possibility, the potential of them. I would have probably not thought about before that interaction. So, yeah.
Can I jump in there? I don't know if Susie flagged up Royal Society Industry Fellowships. This is an opportunity to go and experience industry.
Yeah. And then to bring again the experience of a clinical fellow, I think I've received a great mentorship over the past few years, although it's quite frustrating to hear that the alternative to an early independent fellowship is going for a higher percentage of clinical job, because of course, I mean, I love being a doctor, I love doing clinical activities, but I'm also aware that that would represent a break of the flow in the period which is possibly the most prolific period in the career of an early clinician researcher, clinician scientist. So that's why as I mentioned before, I think we need to structure differently the transition stage.
Thank you, Jasmine and Francesca. Given the fact that Francesca introduced the point, a very important point about the clinical scientists and the crises that have been detected, I would like to involve the former ECR and the leader of our neurosurgery division, Rob, because Rob had a pivotal role in reshaping neurosurgery, academic neurosurgery within the square, and it has at heart the future of clinical scientists. Rob?
Yeah, so I think our job is to listen to everybody and to respond to them, and this is where the ideas and the needs come from, and that's really key. I think Sarah asked an absolutely key question, and particularly pertinent, I think, when it comes to clinician scientists, is we need to be able to support clinician scientists throughout their career, but in particular through training and early career. And that is becoming more and more difficult with time, as you say, the disappearance of various fellowships, the fantastic clinical lectureship program of the NIHR, but there aren't enough of those positions. There are far too few of those. I mean, it is an amazing thing. It was a real highlight when I moved to this country that they have the integrated academic training program, but it needs to be doubled or tripled or quadrupled. And then what happens once you finish, and how do we support you once you finish? And there aren't, it's not easy. And now, you know, with the BRC funding going to be halved at best in the next round, how do we support people? And so I think this is where the discussions between our institute and UCL and the NHS, our hospital trust, and how are we going to ultimately come up with treatments to treat the next generation of patients? It's by doing research, and somebody's got to step up to the plate to support these young people in their successful careers so that we can come up with new treatments.
Thank you, Rob.
Does that answer your question, Sarah?
No, it's absolutely true. It's exactly what I was driving at.
So, question all over the place. First, Mary.
I just wanted to ask one question related to musicians. So some universities in the UK have got systems in place, so they'll do transition funding for a year depending on the person and what's their likelihood of getting a fellowship, so it's competitive. The UCL doesn't have that, although individual departments do, you know, have that. Do you think there should be a more official system in UCL? Do you think that would give, you know, do you think that would be helpful?
I repeated the question, non-AI, of course. The question is about the provision or not of more support for clinical interface, if I correct.
No, it's not necessarily clinical, it's just someone that's going for fellowships.
So more support to prepare for a transition to independence and the fellowship preparation.
Transitional funding, so up to a year transitional funding by the university, if competitively.
Please.
Maybe.
Felipe.
Can you hear me? Yeah. So obviously one of the small issues or large issues with being an early career researcher regards job security and long-term investment. And obviously this affects the scientific output because funding can be short, also the contracts, and this obviously constrains the intellectual risk of some of the research that we're trying to do. So obviously having the possibility of covering some of those gaps will probably contribute to obviously better science, more collaborations, and obviously more freedom and more relaxation to be able to do the science that we like to do.
I think it would be very helpful, particularly if I look at real case scenarios and the fact that most often a postdoctoral fellowship has a duration of around 2 years, and this is more or less well, it's close to the time between submission and acceptance of a paper in a high-impact journal. So 1 year of transition could make the difference for outputs to become visible and to lay the foundations for the next stage.
Okay. Okay, so please.
Thanks. So I come at this from the angle of a clinician scientist that's only recently.
Yeah, the AI doesn't work.
Yeah.
It's better this way.
So, I come at this from the angle of someone, a clinician scientist who's only recently established independence. And I completely agree with the points that Francesca was making, that the pots of money are really drying up and fellowship opportunities are shrinking. One of the things that I found incredibly daunting was navigating fellowship applications. It's almost like writing another PhD thesis at times. So my question to you is a practical one, that would it be helpful if the IoN had some kind of mentoring or matching system where you are matched with someone who has recently completed a fellowship application to help you navigate the writing of it and the reviews that you receive back from the funding bodies?
Sorry, I just stole the microphone. So I'm going to give you a good example that was started by the head of my department, neuromuscular diseases. we established a career development initiative specifically for that to help with grant applications. So peer review, mock interviews, knowing which fundings are available, the deadlines, so all this type of support so that obviously we can guarantee the highest level of support for both clinical and non-clinical fellowship applications. So this is something that started. I'm not sure if it's widespread at UCL. I know there are some departments at UCL elsewhere where there's this type of support, but that's exactly the need that this initiative is trying to tackle, and we hope that will be very successful.
I'm very early in this stage, so like I'm not thinking about fellowships anytime soon, but I think it's just important to say, like, in my department, it was recently created an ECR forum specifically to do this kind of thing. So people that just got fellowships to like interact with people applying for fellowships and just discuss what were their struggles, the things that went right, the things that went wrong. This is a department thing, but maybe it will be interesting to see like an institute level forum like this.
I think that for mentoring perspective, I think that knowing what can go right is very important, but also there are pitfalls in the application that I repeated over and over again. Sitting on several panels for fellowship evaluation, you can see it as a referee. So having the courage to say, okay, I have failed for some reason, very specific reason, is important. Kailash. Okay.
Thanks, GP. Just a comment to say about the support for the fellowship, etc., that there are opportunities, that's true in IoN departments at IoN, but don't forget to look at the faculty level as well, because with our Vice Dean Research, for example, now I think there is an ECR lead, lots of opportunities are going to come out, so it's important as well. IoN is our DNA, but it's also important to look out at the faculty and at the UCL level where there is a huge amount of support. We are often just not necessarily aware of it. And then to come back to your point, Rob, about the clinical scientist, I agree it's really hard, but I would like to add that it's equally hard for the non-clinician academic, if not harder. I just wanted to make the point. Okay, I just wanted to make the point.
Okay.
Thanks. Given your perspective, and it's an international marketplace for science and neuroscience, is the UK still a good place to be an ECR in neuroscience? And is it getting better or worse? And where does it better?
I think this is a really topical question, especially given the current ecosystems and how much they're changing. The Institute of Neurology is in a great position because we collaborate globally across the world. So it means that even if we're based in the UK, we're consistently liaising with people outside of the UK. And that means our research is world-renowned, it's world-known. Given recent changes in the political scene of the US, it means that from an ECR and from a lot of my peers, a lot of the conversations of maybe going to Harvard, who competes with the ranks of the IoN, is no longer as amenable to staying here in the UK and actually building and being able to see your trajectory as you choose to either remain in academia or perhaps go into industry if you fancy. So the UK is only getting better, and especially now that we have a lot more of an opportunity to keep building Queen Square House and the IoN at Grayson Road and continuously collaborate with people, we remain as competitive as, if not more, than what's happening in our competitor universities and institutions.
Thank you. Yeah, so as GP said, obviously we are in times of unprecedented change, I think both in momentum in the field and in funding streams. And I thought, when we think about if the traditional funding is changing, I just wondered if you, as ECRs, are thinking about alternative ways to build that agility to find alternative streams or look for entrepreneurship opportunities. And I know this is probably directed at you a little bit, Alya, because you've built a massive movement in the UK with Nucleate UK, and I wonder if we could learn from some of those things about how to find new ways to fund research and move things forward to make sure we are getting the best innovations to people that are waiting for them.
And it's a fantastic question. It will be a great lead up to my talk after this. And not to spoil anything too much, but Nucleate UK is designed to support academic trainees who are interested in spinning out their technologies. And this is very much a question that I think a lot of us on this panel has thought about our primer conversation around IP and how can we make sure that we're licensing the IP that we're developing. I definitely think that within the IoN, we've started having fantastic conversations about translation. Those conversations can easily be amplified and focused on a little bit more. Understanding the world of enterprise is really important because commercializing your technology could be a great idea, but it could also be a really bad idea depending on the technology you're planning to commercialize. Some technologies don't deserve or don't need VC backable funding. Instead, they actually would benefit a lot more from having grant funding or going through the industry pipeline. So having open conversations about that, I think, should be our next steps and should continue to be our next steps since we're already having them. But stay tuned because Nucleate UK is also one to keep an eye on if any of you guys are interested in learning a bit more.
So I think that the time is up for this taster about a new way to engage ECR and the faculty. I think that this is a beginning of a conversation that we really would like to bring forward and start. And remember, you have champions in your department and your head of department, all the deputy director, all the head of division are there for you, and you have an important ambassador that you can contact. And just a comment to the observation done by Helene. We know that the comms sometimes fails, and we needed to pay attention to comms and lack of thereof, because sometimes the resources are there and we don't know about them. So please join me in thanking this brave ECR and Professor Branson for being on the podium.
Okay, so I thought that was a fantastic ECR session, and thank Alia and all the ECRs. I think we should give them another round of applause, actually, for a fantastic session. Yes, it's not easy for them to get up and do that, so thank you very much. So if you want to take your seats for the main event, I'm really very pleased and grateful to Lord Valens for making time to join us this evening for the guest lecture. He was appointed the Minister of Science, Innovation, Research and Nuclear in the Department of Science, Innovation and Technology and the Department of Energy Security and Net Zero on the 6th of September. So sounds like it's quite busy. He was previously Minister of Science in DSIT from the 5th of July 24 to September 25. And of course, you will all remember that he was Government Chief Scientific Advisor from April 2018 to April 23 during the COVID pandemic. And all of you, I'm sure, will remember the very clear advice that he gave along with Chris Whitty and also at the time Boris Johnson. His career outside government is extremely impressive. Lord Valens was President and Director of R&D at GSK from 2012 until 2017. Prior to that, he was Senior Vice President, Medicines and Discovery and Development. And during his time there, he was directly involved in developing many new medicines which were approved for use worldwide for a range of diseases, including cancer, rare diseases in children, and asthma and HIV. Many of you will know that prior to joining GSK, he was a clinical academic at UCL. He was Professor of Medicine and led the Division of Medicine at UCL. He was also a consultant physician. I actually remember him referring patients to me at the Middlesex Hospital a long time ago. And he's got many years of experience in basic science, clinical research, which has spanned from medicinal chemistry, structural biology, cellular work, and animal work. He was awarded his F.Med.Sci in 1999 and became FRS in 2017. So it's really a great privilege and honour to welcome Lord Valens to give his guest lecture. Thank you.
Well, thanks very much, Mike, and it's a great pleasure to be here on this day to celebrate 75 years of the Institute of Neurology. And looking back at what's been achieved, it's remarkable. I mean, if you think about the origins of how we think about treating stroke and the demonstration of there being a penumbra around the stroke that was actually recoverable, to the work on the genetics of various neurological diseases, Parkinson's and others, to the Huntington's work, to the epilepsy work from ions now through to gene therapy, prions, MRI. I mean, it's an extraordinary roll call of achievement. And not only is it a roll call of achievement, but what I think is fascinating is to have seen Queen Square go from predominantly a clinical institution to a clinical and world-class research institution. And increasingly, and it was very evident in listening to the early career researchers, to go to that next stage now to somewhere that actually creates companies and starts to turn those into products. So that evolution is, I think, of great importance. And I was reflecting that 40 years ago, when I was applying for SHO jobs, the one I didn't get was the one at Queen Square. And they've since invited me back 3 times to speak. And it feels like it's a very prolonged interview process, which one day I might get some success in. But what I want to talk about is exactly this point about taking science and innovation through into impact. And I want to talk about it in science broadly, rather than just focusing on biomedical science or neuroscience. But I think what I'm going to say, I hope, is relevant to how we think about that as well. So I'm going to start here. And the purpose of this slide is to say, I can't think of a single area of government activity or operations or policy that doesn't need science and technology and wouldn't be better if we had more science and technology. So that's obviously true in health. But it's equally true in how we think about building our houses or designing our cities. It's true in terms of how we think about our food supply and what we want to do about the future ability to get food in a changing environment, or our transport, which is changing in front of our eyes in terms of how we move from A to B. Communications, of course, depend on that. Our energy supply depends on it. So every single department, including education and how we think about the impact of science and technology on the ability to make sure everyone gets an education. It's not just, though, that sort of general departmental point. If you go to resilience and national security, science and technology is at the heart of that. It's obviously at the heart of resilience to natural events and accidents. So in my first weeks as Government Chief Scientific Advisor, I was involved in the Novichok poisonings in Salisbury and Amesbury. That Christmas after that, and some of you may remember that, I got a phone call about 2 days before Christmas saying, what do we know about drones flying over civilian airports? And the answer was not very much, as it happened at the time. And it's still quite a difficult problem. What do you do in a situation where you just can't go and shoot them down? The following year, and the middle slide at the bottom there shows this, a dam was due to collapse, Toddbrook Dam, you may remember that, and all sorts of questions about where the water would flow, what that would do to underlying infrastructure for surrounding areas, disease-borne problems and so on. And of course then, having gone to be Government Chief Scientific Advisor to slightly broaden beyond biomedicine, COVID came along and I spent most of my time dealing with COVID. So clearly important for natural disasters, but also absolutely central to science and technology, to defense and security. So you've only got to look around the world. I mean, China puts this absolutely top of its priorities across departments and for its defense. And we're in an era where we have to think very seriously about defense, and therefore this becomes a major part of what we should think about. So those are reasons why government should care about science and technology. But beyond that is another one, which is, of course, central to any government, which is economic growth. You can't actually get much greater than average economic growth if you don't have science and technology companies. And I think it's been like that for a very long time, but it's strikingly true now. And Mario Draghi, who wrote the report for the EU, basically said, if you don't have science and tech companies, then you can't grow as well, and if you compare the US with the EU, the outsized growth is all driven by a small number of tech companies. And the bottom part of the slide shows the same thing from the Tony Blair Institute, showing the price to earnings ratio as a sort of surrogate of potential growth for the S&P 500 in the US and the FTSE 100. And so if you say at the beginning you start at the same place, at the end in 2024-5, the FTSE is well below the S&P 500 in terms of its growth potential. And if you strip out the tech companies, basically they look identical. So you have to have science and tech for every departmental activity. You have to have it for defence, security and resilience. And if you want to get growth, you've got to be good at this and getting it through into companies and growth. So that leads to the obvious question, which is, are we any good at it? And I'm going to start with the government itself. And when I joined government as a Chief Scientific Advisor in 2018, together with the then Cabinet Secretary, we asked a question which was, well, what did it look like in government in terms of the ability of government itself to deal with science and technology? And I'm not going to go through the details of this, but I do want to pick out what was one really striking set of numbers, which is when we looked at the FAST stream, so that's the graduate entry scheme, which is almost certainly going to determine what the future of the civil service looks like, because these are the bright young things who come in and get fast-tracked through. We found that there were about 1,200 people on fast stream schemes, of which 24 were in science and engineering fast streams, but even more striking, out of the totality of 1,200, 45 had a STEM degree. So the whole of the civil service was skewed towards not having science and technology knowledge. And these were the numbers which got the permanent secretaries to sit up and take notice and say, we've got to do something about it. And I'm pleased to say that the answer to how many there are now is 50% of the fast stream have a STEM degree. So it's definitely moving in the right direction. There are definitely some pockets of excellence in science. There are some public sector research establishments. The Met Office would be an example. The National Physical Laboratory would be another example, where there is really world-class science, and that is part of the government process. So there are definitely pockets of excellence. There's definitely a much greater awareness of the need to have science and technology, and there's a greater skill set coming up through government. But if you're going to mark this from a school report, you wouldn't be giving it an A. You'd be giving it a sort of B with a sort of try harder and you'll get there. And I think that's exactly what we need to do. But of course, what happens inside government is not by any means the answer to how good are we at science, because we know that when we go outside and we look across the universities and research institutes, we're really good. And we're good in terms of number of universities rated highly, in terms of number of academic publications, the world's most highly cited publications. We do well here. And I think it's important to recognise that we do well, but we should not and cannot rest on our laurels. We cannot assume that we will always do well. And whereas 20 years ago, there were not that many countries that could really be at the cutting edge of science and technology, there are many more now. The competitive landscape is really quite different. And so we have to make sure not only do we support this, but we continue to strive to be the best we can. And that's going to require funding. And at the last spending review, which is the time at which money is given out to departments for the subsequent number of years, and in this case it was more than 1 year, it was a 3-year period, you find we managed to make that argument and got £86 billion overall for R&D and science and tech across government. And that's the biggest amount that was ever given. And it was done at a time when there were enormous financial pressures. So if anyone doubts that this is a topic which is important in government and government worries about it, just look at that, because many other things were cut at that time. Now, everyone's always going to want more, of course, and that isn't all money that goes to academic research. But there is a big slug of it that does. And one of the things that I think is important is to make sure that we use that money in a way that really delivers the sorts of benefits, the impact that we're talking about. And that's why with Ian Chapman as head of UKRI, we've set about trying to define more clearly where the money does go. And one of the unintended consequences of creating UKRI was that when the money went to 9 different places, different research councils and so on, it wasn't big enough for anyone to care about. When it's all in one place, it's very politically visible. So you better be really clear that you're using that money well, because suddenly it looks like something that competes with other things like defence or education or social care that matters to the population rightly and matters to society. So what we said was there's a chunk of money which must be the bulk and must be the thing that you care about as a funding agency to go on basic curiosity-driven investigator-led research, the sort of thing that I've been clear, no politician should be interfering saying where that goes because nobody knows. It should be going to academics and investigators based on peer review and the ability for others to demonstrate one thing to ministers, which is this is world class. But the subject matter shouldn't be for politicians to interfere with because they'll get it badly wrong. And we all get it badly wrong because by definition we don't know what's going to be important, and the thing that looks slightly bonkers today turns out to be unbelievably important in 20 years' time. So that's a key bit that needs to be protected. But then there's a bit, often on the more applied side, which is relevant to answering some of the big questions that society has, that governments have, that we want answers to. And that is a legitimate area for government to say, these are the things we care about most. These are the things that society needs. And in that funding, we need to be much clearer about what the outcome is and when the outcome is going to be given. The timelines become important, and the ability to leverage private sector investment alongside that to make that happen. And then the third bucket is companies, and I'll come back to companies in a minute, where the public spend in R&D can leverage private sector investment, can enable things to happen that otherwise wouldn't happen, but shouldn't be something that private sector would do on its own. It should be money that will catalyse the ability for things to happen either at a start-up stage or growth stage, or indeed in R&D intensive businesses. And then of course underlying that there's the need to have infrastructure and skills and all the things that matter across all of those. So that's why UKRI is doing what it's doing. And I think it's incredibly important, not least because you don't know, I don't know, what colour of government we have in the future. And people will ask questions about this, and the more we can demonstrate exactly how this money benefits the things that government cares about, the more we're going to get growth in that budget, which is what we need to assure. And already several of the early career researchers mentioned ARIA, and in the PhD talk just now. I mean, ARIA is another way of making sure that we don't only fund pure curiosity, but we think about, well, what might that turn into and how might we be able to translate that? And that's all very high-risk stuff in ARIA, but that's important and it's already having an effect. So this, I think, is an area that we must keep focusing on and must make sure we get right. But I want to spend a little bit of time talking about the third part of what a country needs, as well as a competent government in science and technology, a very strong independent academic and research base. It needs companies to be starting, growing and staying in the country, and indeed it needs research-intensive companies at a big level. We do pretty well in some of that, and the big level companies are important because they're the ones that often train people who are in biotechs and tech companies. And if you look in any company, whenever I go to visit a small biotech, I meet people absolutely guaranteed who trained at either AstraZeneca or GSK, because those are the companies that do most of the training. So the big companies are important. We do extremely well now on startups. And I was really impressed and amazed by listening to the early career researchers, how many referred to companies, startups, engaging with companies, careers in industry. That definitely was not the case when I was an early career researcher. It was definitely not the case when I was here as Professor of Medicine at UCL. It was unusual. And it was also seen as slightly not the done thing if you were a credible academic. That has changed, and thank goodness it's changed. It's incredibly important. So companies are crucial, and we are doing quite well. Number of startups in the UK is greater than anywhere else in Europe, and just in the first quarter of this year, we've got the biggest income of venture funding ever in the UK, and it's about 48% of all money coming into Europe for venture came into the UK. So that's a pretty good position to be in. And that's helping us get this flourishing startup scene. I might still argue that some of them are a bit undercapitalised, and that's a problem because you then spend all your time raising capital. But the real challenge we've got, and it's a long-term challenge, is scaling those companies. How do we make sure they get to a scale where they can really be credible? They should displace the companies that are at the top of the FTSE 100 now. And we're not doing that enough because we're not scaling. And that's what I want to spend just a little bit of time talking about, what government's doing to try to get companies more able to scale in the UK. And the first question is about funding. It's about release of capital, because we're talking about funding rounds of 300-400 million plus. That's where we're falling down. And this was some work done a few years ago, but it sort of normalised the US and the UK to GDP, and then asked what's happening in the venture space. And at the angel and seed level, the US and the UK were roughly the same, US a bit better. Early venture capital, US was better. That gap's sort of still there, but the UK is doing well. At late venture capital and scale-up funding, we're much worse. Actually, it's even worse than this. It's about an 8-fold difference. So we are missing capital in terms of being able to scale our companies. Now, in a way, a weird way, that probably didn't matter so much 15 years ago because we didn't have many startups to scale. It matters enormously today because we've got lots of startups. Every day we don't get this right, we're missing an opportunity. And every day we don't get it right, we're offering a very, very nice selection of things at a relatively reduced price to be bought by somebody else. And that's the issue that we're trying to tackle. I want to show you one other piece of data about this as well, which is the money also comes from different places. So at the very early seed stage, a lot of the money comes from the UK. We've got seed and angel investors who fund in the UK. But as you go through the rounds of funding and you get up to the very big rounds, the UK proportion of funding drops quite dramatically. So that's the blue bar. The amount coming from the US goes up enormously, and indeed from the rest of the world. Now, I don't mind the fact we get lots of incoming capital. That's good. But this, again, is a missed opportunity and a problem. And the missed opportunity is that when you look at what's funding some of these bigger rounds, there are pension funds in there. They're Canadian and Australian pension funds. So pensioners in Australia and Canada are doing very well out of UK science. It would be quite nice if pensioners here did very well out of it. So there's a missed opportunity for pension funds, and there's a missed opportunity for releasing large amounts of capital into this space. It's also a problem because it means that we're not actually getting enough capital in at the right stage with investors who really understand it. So this is a big focus, and it's why the government has been looking at, well, it has made changes to make pension funds really clear that the expectation is that they should put money into these areas. That was the Mansion House Accord and Mansion House Compact, and it's happening. It's beginning to happen. And this year, I think we're going to see much more of a release of capital from pension funds. And I'm pretty sure, having spoken to lots of people in the industry, that once this starts, there'll be a FOMO effect and everyone will crowd in, and we're going to start seeing a lot. And then we'll see specialist investors, people who understand the risk profile when you're doing this scale up. So although I've said it's the big problem, it's one that I think is looking better. And there were some other measures in the budget last time for allow companies as they scale to give more incentives to their employees without incurring a tax burden, so that there are changes which are pro scale up, and I think we will do well. And just today, actually, I was reading that we're now the 3rd country in the world for new unicorns, so companies valued at over $1 billion from this area. So I think things are definitely moving in the right direction. But having laboured a bit the point about money, I do want to say it's absolutely not all about money. And when I was GCSA, we tried to put together a science and technology framework to say, how should you think about this as a government? And that then got changed a bit and published under the last government. And one of the things I was very keen to do is not change it when I came in, because chopping and changing is not a good thing in this area where you've got to have a long-term consistency. And the ideas are pretty simple, which are say what you think you want to be good at. And that's where the industrial strategy and making some choices on technologies matter. Make sure that you think about skills development, and we heard about that earlier. There's work to be done not only in the overall skills base, but also in training people to think in an entrepreneurial way and to have the opportunities to do that. And I'm pleased actually that there are some PhD studentships which UKRI is putting together, where people will deliberately do a PhD where there's an aim also to perhaps commercialise the work as well, so that we start to see some of that happening. So skills, infrastructure, we know we need, a point I'm going to touch on right at the end. But there's a couple of others, and the first is regulation. You can kill companies easily with regulation, and big companies actually quite like regulation because it excludes others from being able to enter the field. Small companies, it's really difficult. And that's why we set up the Regulatory Innovation Office, which is there not to think theoretically about regulation, but to go out, speak to biotechs and tech companies and say, what's getting in your way, and then try and unblock that. And that, I think, is having an effect. But the approach, I think, is important, which is to go and find real problems and solve them. So let me move to the other one I wanted to touch on, which is procurement. And procurement is, I think, more important than most people realise in this space. If you're a company that has got venture funding to start, you're beginning to show that you've got a potential product, your first customer signals something very important to the market. And the government spends £400 billion a year on procurement, and it's not really targeted towards SMEs, towards innovative companies, towards higher risk procurement. That's not true in the US, where government money is often used to support the early stages of a company through a procurement contract. And it's true in other places as well. And so what we're trying to do is to get this notion that using government procurement money to get early contracts or advanced market commitments out into our own domestic companies will be an unlocking feature for then getting large amounts of private sector capital to come in, because that company now looks as though it's got value because it has a customer. And I just want to give an illustration of two and then a ambition for two. So a couple of months ago, 3 months ago, we've been puzzling for a while. We've got a very thriving quantum computing sector in this country and quantum science generally. But it's clear that our companies are at a stage where they're looking quite juicy to buy. And what I wanted to do was to put down a marker to say the government will buy a quantum computer. And so we got a commitment from Treasury to put over £1 billion to say, we'll buy a computer if you can make one that does this, this, and this. That competition launched with lots of companies being able to enter for it. Lots of companies are going to get a provisional yes, or several will get a provisional yes because they can potentially do it. And then obviously there'll be a release of some money along the way and milestones. But the moment we did that, companies from overseas said, I want to move to the UK. UK companies started unlocking large amounts of private sector capital, and our base looks much more secure. We actually got a call from the US to say, oh, that's a good idea. We wish we'd done that. So, you know, these sorts of procurement signals really matter to the market. And the same sort of things going on with nuclear fusion, where we said we'll build a nuclear fusion plant. That's already happening. Is it going to be the only one that makes it? No. Will it make it? I still don't know. But I do know that around that, because of the supply chain and all the small companies associated with it, we're already seeing economic benefit. The two that absolutely require attention and have big opportunities are the NHS, where we're currently not great at using the NHS to give early contracts to innovative things and saying we would buy a British product from a UK company which nonetheless carries a bit of risk but might be really good. So there's a big task force working on this now to be innovative procurement in the NHS, which I really hope is going to deliver, because I think it will make a big difference. And we've got lots of examples of UK biotech companies and tech companies, med tech companies, who make a product and find they can't access the NHS, and they sell in Germany or France or the US, and the next thing that's going to happen is they'll move their headquarters. So this is an aim to try and get that growth part of the NHS really working. And the same story for defence, and that's why the defence investment plan that was published this week is really important in terms of not just, are you going to buy tanks and are you going to buy warships, but are you going to do things that actually stimulate innovation in the UK, innovative companies, very often actually dual use that have other potential outcomes as well. There's a big opportunity in those two areas. Now, none of this happens by chance in terms of innovation, and it's worth looking that, although it's easy to assume it's all happening in the Golden Triangle, as people have said, it's not. I mean, a lot is happening, and I was asked by the Chancellor about 18 months ago, would I try and work on the Oxford-Cambridge Growth Corridor to make sure we maximise the potential of what is an internationally competitive opportunity. But it's happening right the way across the country. And what we're doing in DCIT is trying to map these much more effectively by sector, by area, to enable to explain that to investors, but also to make sure that we do promote this agglomeration that's so important, and the movement between academia, industry, small companies, big companies, back into academia, to get all of that working effectively in those areas. And part of that was to create something called the Local Innovation Partnership Fund. And the idea there, with £500 million, was to give money directly just to strategic mayoral authorities, so that the mayor, who knows the area better than anyone in Whitehall can know it, can start making choices on his or her knowledge of the system and do things that might not be done centrally. So that, I think, is going to be a theme on this sort of innovation spend, particularly on the company side. How do we do that in a way that actually benefits growth elsewhere? And I will finish with something, and people will be sitting there thinking, what the hell, he's not mentioned AI at all. Well, he is going to now, because it's where all the questions, of course, come. And I think that we still underestimate the potential change that's coming upon us with AI in all sorts of areas. And when we think about AI, it's not just the algorithms, it's the data. And we've got some data-rich resources in the UK, particularly in biomedicine. And it's the hardware. And we have underinvested in significant compute infrastructure in this country for a very long time. That is being reversed very rapidly now, with commitments to increase the compute resource by 20-fold, which have already actually been achieved, and there'll be another 30-fold beyond that. And it goes back again to the early career researchers point. The question that Dimitri asked is, do you need to access AI? Yes, you do, and that means being able to access not just the algorithms, but the compute infrastructure as well to be able to do that. And there are a number of measures being taken place, ranging from creating the Health Data Research Service to have a much better way of getting safe health data securely looked after, available for people to do research on and to develop products, through to growth zones, compute infrastructure, new initiatives to try and get data out in a way that industry can use, investment schemes. But what I wanted to finish on really was the notion that although AI is going to affect everything, most people at the sort of cutting edge of AI now that I speak to in different companies are completely obsessed with the effect that AI is going to have on science. And therefore, we need to be really on top of this, because AlphaFold, and of course Demis is an alumnus of IoN and UCL, AlphaFold completely changed our ability to look at protein structure rapidly. That is the start. And people, including at DeepMind and isomorphic are ranging from everything from can it be the hypothesis generator through to can it be the robotic experimenter through to where do you go beyond, for example, in the drug discovery space, beyond just protein structure to being able to predict what molecules to grow, and how do you then design the clinical trials with the best patient population and the right outcomes. This is going to completely change science, and I think therefore it's incumbent upon us to make sure that we do put enough resources into making this enabled for researchers and academics to do what they need to do. And there is a large chunk of money going into AI from UKRI and others. That chunk is going to have to grow if we're really going to get on top of this and understand what the future of scientific research looks like. I have one sort of slight, because people may be sitting there thinking, oh goodness me, my job's at risk. I think there is something quite profound about the fact that this is a human-AI interaction that we're talking about. And the other thing that perhaps is less profound, but I've become slightly obsessed with, is the number of things that happen in science which are massive accidents, and what that means for AI and science. And I don't mean things that are unpredictable and unseen, because AI is going to be quite good at getting that. I mean, and an example from an area that I was involved in years ago, where one technician was very careful with a blood vessel and one technician was not very careful with a blood vessel. And as a result of that, Robert Furchgott noticed that the problem was that one was rubbing off the endothelial layer, and therefore that blood vessel didn't behave normally, and he discovered endothelium-derived relaxing factor and got the Nobel Prize for that. I don't know how AI would ever make that mistake of being clumsy and not clumsy. It would probably spot there was a difference, but I think there's plenty of human stuff still to be done, and part of that is our ability to make mistakes as well. And with that, I will leave you, but thank you and well done for the 75th.
Well, it's a pleasure for me to make some closing remarks, and particularly to thank Lord Valens for his giving up his time to come here. I know he's an alum of UCL, but that's quite a long time ago. I also remember getting advice from him in his office. You probably don't remember, but I can't remember what you said. But anyway, I'm sure it was very good. Anyway, I shouldn't have mentioned that. I just also want, I mean, the presentation, the talk was incredibly insightful and informative, and actually quite encouraging, even though some of the areas are a bit challenging. There is opportunity. Just reflecting, this is really, this is a year of birthdays for UCL. So we've got our 200, which UCL thinks is very old. It's not that old compared to some other universities, but we also, within the faculty, we have the Year Institute, 20 years, ICN, 30 years. But it's a particular pleasure for me to be here to talk about IoN 75th. I had the privilege of being the director here. People always say the privilege, and it was a privilege for a short period before Mike took over. Today's programme, I think, gave us a real sense of the breadth and depth of what goes on at Queen Square, and it is really, really impressive. And I think emphasised the relationship with the hospital, which is so critical and which has been so strong for so long. I mean, that's been incredibly important. And also the ECR, I mean, I really like the early career discussions. And it's really fortuitous you were here to hear them, Patrick, because, I mean, they said a lot of the things that you hoped they might say, and they did that unprompted. So that's really even better. When the Faculty of Brain Sciences was set up only 15 years ago, the Institute of Neurology was one of the key participants. And going back to Mike's slides, which I understand he did in 20 minutes, when I looked at them this morning, it took me about 40 minutes to go through. So it must have been at some speed. You get a sense of the trajectory, which is quite incredible in terms of academic output, particularly grants, papers, student numbers, and we even now have an undergraduate BSc, which would have many of our august predecessors turning in their graves, children at Queen Square. Anyway, fortunately, that's in the past, and this is really a great achievement, and I'm delighted. It also helps with the challenges around financial sustainability, which I have to mention, given my role. But of course, when you build up your research as you do, it's often really challenging to keep that sustainability for the future. This is a work in progress. And now we're moving into a different era, which is quite challenging. Again, as came out when we were talking to the early career researchers, we need to be more fleet of foot. We clearly need to work a lot more with industry, and everyone is telling us that, and we certainly need to do that. We need to make sure we have impact. We need to focus on the areas that government is interested in, that wants answers to, and that's entirely appropriate. And I think we're well placed to do that. And again, that's what we're about here. It's certainly what Queen Square is about, and it's what the young people are about. So I think that's all actually really encouraging. And if we look to the future, the short term, we've got a lot to look forward to. We have this extraordinary new building. I mean, putting up new buildings in UCL is not something that happens very often. And it's not likely to happen very often at the rate things are going. So I think we are in a very, very fortunate position to have this building. And we need to demonstrate the value of it. So there'll be a lot of pressure on us to show that this is going to achieve what we set out to achieve by bringing everybody together and being so productive. And that it will have a real impact on our patients and on society. I mean, it's incredibly important. And of course, we need to mention we're going to have a new director of the Institute, Professor Erin Schumann, and she comes from Max Planck Center in Frankfurt, as many of you will know, and has just been the recipient of the Kavli Prize, which is incredible and really a marvelous achievement. I can't see Erin, but there I do see her. She will be the first female director of the Institute in 75 years. So that's pretty good. So to end, I'd like to thank Mike and Helene for arranging this extraordinary day and for all the work they and the team have done over the years. I'd like to wish IoN all the best for the next 75 years. I hope to be around for some of it. But particularly, I'd like to thank Patrick for a really fantastic lecture, and thank you for coming. I think we really, really enjoyed it. So thank you very much. So now I think we have to do something with the plaque and the purple thing. I think Helene is joining us as well.
You okay? Okay, here we go. Come on, take it over. Oh, there we go. It's trapped. Let me have your favorite.
So just to add my thanks to Lord Valence, and that's the end of proceedings in here today. So you're all now welcome to join us in the marquee. So if you would like to go there, make your way there now. Thank you very much.